Hello, hello, hello. Welcome, everybody. Welcome to the stream. Welcome, welcome. Hello, hello. I am so excited to be talking about this particular essay today. The end of the genetic paradigm of cancer. A very, very uh, strongly worded essay that is going to be so much fun to read. Before we get into it, let me say hello to everybody. Hello, Astro. Welcome, welcome. Hello, Marlon. Welcome. Hello, Moon and Archie and Nicholas. Hello, welcome, welcome. Who else we got in chat? Diamond, welcome, welcome. Gluon, hello, hello. Arcady and Lettuce Bird, welcome. Lunkil, Nicholas, Jirier, welcome. Clockwork, hello, hello. Aiden, welcome. Welcome to the freaking stream, y'all. I have had way too much coffee, as you can tell by my I don't know, difference in volume. I'm really, this is too much caffeine. How much caffeine is too caffeine? Too much caffeine? In my case, it's exactly one coffee. I am ready to go. Welcome, Aingi. Yeah, let's go. Oh, and Silas, welcome. So this is the end of the genetic paradigm of cancer, and this paper is pretty insane, I would say. Give me 1 second to... All right, we're all good. Welcome. Okay, let's get into the paper, shall we? Oh my goodness. So this is a paper that was published just last week and has been causing a bit of a stir on science social media right now. And so I knew I wanted to cover this paper when I read the very first sentence of the actual introduction, which I will do right here. We'll come back to reading the abstract for sure, but first let's just read the introduction here. It is said that the wise only believe in what they can see, and the fools only see what they can believe in. And that is a very strong way to start an essay. You can immediately see the tonal contrast between this opening statement and the language that Charles Darwin used in The Origin of Species, right? Because if you've seen my stream on The Origin of Species, you would know that Darwin tried really hard not to upset anybody in his writing. He was very careful to be like, well, we have evolution, and this might imply that there is some universal ancestor. But if you want to believe in a supernatural origin of life, you are certainly welcome to. He's very diplomatic in that regard. And this paper starts off just by calling everybody who disagrees with you an idiot. Or which I think is really it's an interesting strategy, and we will see very shortly whether or not this strategy pays off. And so what is this paper actually about? So as the title implies, this is about the end of the genetic paradigm of cancer. This is an essay. This is not a research paper. So this is not a paper that is claiming to have made a new discovery, but rather one scientist talking to other scientists being like, hey, I think we've been thinking about cancer all wrong. I can't stand statements like this in academic papers. I don't know if you would call this an academic paper. This is an essay, right? So this isn't an actual research paper. This is more of a casual format, a persuasive essay, if you will. And to me, that allows this paper to be a lot more flexible, a lot more loose in its language. Clockwork says, this paper rules, loved reading it. Love reading the intro. Every paper that name drops Philip Ball is an instant 10 out of 10 for me. Yeah, I have not finished reading this paper. I took a look over it just to make sure that this wasn't some kind of woo paper, some kind of, shouldn't say anti-science, but rather some questionable science. And it turns out that for reasons that I will get into the paper, I actually think that this is quite a noteworthy attempt to get us to rethink how we think about cancer, not just cancer research scientists, but how even the public thinks about how cancer operates. And I'm very, very excited to get into this with all of you. But yeah, this beginning, if you've seen my social media posts, to me, this first sentence screams, that meme from Invincible, like, think, biologist, think, like, and I'll go over why I think that I'm not angry about the way that this paper starts. I think it's really awesome, and I think that it's worth noting, and will be very interesting to look through. All right. So let's talk about the abstract here, just to give ourselves an idea of what this paper is trying to say. So, genome sequencing of cancer and normal tissue, alongside single-cell transcriptomics, continues to produce findings that challenge the idea that cancer is a genetic disease as posited by the somatic mutation theory, or SMT. In this prevailing paradigm, tumorigenesis is caused by cancer-driving somatic mutations and clonal expansion. You can do exclamation point jargon 1, 2, 3, and 4 to catch up on the acronyms if you were not aware. However, results from tumor sequencing motivated by the genetic paradigm itself create apparent paradoxes that are not conducive to a pure somatic mutation theory. But beyond genetic causation, the new results lend credence to old ideas from organismal biology. To resolve inconsistencies between the genetic paradigm of cancer and biological reality, we must complement deep sequencing with deep thinking, embrace formal theory and historicity of biological entities, and reconsider non-genetic plasticity of cells and tissues. In this essay, we discuss the concept of cell state dynamics and tissue fields that emerge from the collective action of genes and of cells in their morphogenic context, respectively, and how they help explain inconsistencies in the context of somatic mutation theory. Okay, so a lot of jargon here. Let me translate. This paper wants us to stop thinking about cancer in the context of genetics, that we have been over-reliant on this idea that our DNA is destiny. And so this paper wants to argue that we should be taking a more organismal approach instead of one that relies more heavily on understanding what our genomes are. So this is why I chose to cover this paper, right? This is not an insane idea. The way that it starts in terms of the introduction is insane. However, the idea itself is not that crazy. Yeah, welcome. I see Rico's in chat. Hello, welcome Rico. Hi, Nickname and Gamero and Ezin. Welcome. Good time to jump in. We are just getting started here. So again, we have this crazy introduction here. It is said that the wise only believe in what they can see, and the fools only see what they can believe in. I love that line. The latter attitude cements paradigm, and paradigms are amplified by any new looking glass that puts one way of seeing the world on steroids. Again, they're very casual language, which I appreciate. So that's basically saying we've been thinking about cancer in one particular modality for far too long, that we have been closed-minded because we are tempted by the technology that makes certain fields of research easier than others, and we have been taking the easy route out. In cancer research, such a self-fulfilling prophecy has been fueled by next-generation DNA sequencing. So what the author is trying to argue here is that because sequencing technology has gotten cheaper and more accessible, that we have been far too reliant on using the technology to guide our research instead of our research guiding our technological approaches. Not gonna lie, people seem to refer to genetics too much, even outside of a strictly biological context. Hate when all of my traits and abilities are being compared to my relatives. I mean, yeah, like, this is why, as a biologist, I'm very careful about what is called biological determinism, right? That your existence is measured not by your achievements, accomplishments, personalities, actions, but by DNA code written into your cells, which is reductive, to say the least. The ease of access to sequencing has stimulated the unfettered quest for genetic alterations in tumor cells that would explain cancer. Since the 1970s, the elevation of the concept of cancer as a genetic disease has been driven by a number of factors, including discoveries of cancer-associated mutations, narrowly gene-engineered mouse models, funding policies that follow crowd thinking, and the promise of targeted therapy. The propensity of the human mind to cling to simple, mechanistically plausible explanations further promoted narratives of oncogenic quote-unquote driver mutations. The somatic mutation theory and its tacit claims to represent the truth on the origin of cancer is concisely articulated in this quotation by M. Stratton and L. Alexandrov from a decade ago. All cancers originate from a single cell that starts to behave abnormally due to the acquired somatic mutation in its genome. So, yeah, I mean, you know, if you think about it, the way that everybody learns how cancer operates is that there is a mutation in a cell, and that mutation causes the cell to behave abnormally, and then that abnormal behavior results in unchecked replication. And this is also a theory that I myself have almost quoted word for word in my cancer stream, my 12-hour glioblastoma stream, because this is, whether or not you like it, the predominant model by which we understand cancer to operate, and that there's a mutation and that mutation causes cancer. It's almost insane to kind of challenge this theory, because this is a theory that makes a lot of, intuitive sense. And so this paper is going to have to do a bit of work to try to convince people to stop thinking about cancer as a disease of genes. So I think, okay, if we're not thinking about cancer in terms of genes, how should we be thinking of cancer then? So let's see what this author says. This quotation dates back to 2014, and we will present an updated 2024 version later. However, during the past decade, widespread acceptance of the somatic mutation theory has driven the use of next-generation sequencing. With lowering costs of genome analysis, the genetic paradigm led to precision oncology, but targeting driver mutations has invariably met the challenge of relapse of treatment-resistant cancer too fast and in far more aggressive form than can be explained by selection for new mutants. Knowing the mutational profile of a patient's tumor genome has yielded little benefit in personalized/precision cancer care. So the author here is saying that, like, if cancer really were so simple as understanding the patient's genome, we probably would have cracked it by now. And we've actually seen this idea come back in the 12-hour charity stream that I did on glioblastoma, especially in that paper where I talked with Dr. Layla Akari about how complicated and complex the tumor microenvironment is, right? So there's some precedent for this even on this channel, where it's a lot more complicated than just, do you have a mutation? The tumor microenvironment itself is an extremely complex system. And so if we take a view that genetics equals cancer, then we are handicapping ourselves, let's say. Ironically, over the past years, the same sequencing technology has exposed new cracks in the edifice of the cancer genetics paradigm. Table 1 summarized the most salient findings that challenge the paradigm. Some sequencing results are compatible with oh my god. I've been trying to ignore Purkinje in the litter box. If you hear scratching, that's what that is. Purkinje is taking her sweet time in the litter box. Sorry, where was I? Some sequencing results are compatible with the postulate of oncogenic mutation, but many inconvenient findings are overlooked for a lack of critical discourse, resulting in an unbalanced view. Thus, an open discussion of the growing sequencing data that are contradictory to the genetic paradigm is due. The apparent paradox goes both ways. Many cancers harbor no consistent driver mutations, while canonical oncogenic mutations are found in tissues that remain free of cancer. Paradoxes, according to Niels Bohr, love quoting Niels Bohr, are of course central for progress if properly recognized. So this is really, really interesting, right? And this is something that is not common knowledge. Certainly wasn't common knowledge to me, that you can have cancers without a genetic mutation, and that you can have mutations that don't necessarily result in cancer. So that last point is pretty salient, right? With the advent of 23andMe, you can, of course, order an entire copy of your own genome and discover your risk of cancer. But implied in the term risk is the fact that genetics is not destiny, right? Your DNA is not your destiny. That you might have certain mutations that can elevate your risk of developing certain cancers, but it isn't, the causative link is not fully penetrant, we'll say. And, you know, I think this is something that we've just all been trained to accept. You go, okay, so that's not that hard to accept, right? The fact that genetic mutations don't always result in cancer, because we can just kind of hand wave, well, the cell is complicated and organisms are complicated, but it's still fruitful to understand these oncogenic mutations. And so where this argument is going is that we might kind of be wasting our time focusing too much on individual genetic mutations, because, yeah, that might be a bit of a myopic view. And we'll take a look at that table in just a bit. Large-scale cancer genome sequencing endeavors, such as the Cancer Genome Atlas, launched in 2006 by the NIH, have uncovered a mesmerizing breadth of genetic mutations associated with cancer. The vast, nearly chaotic diversity of genetic alterations found with the same nominal tumor type between patients is staggering and questions a deterministic logic of causation of cancer by mutations in oncogenic pathways. However, patterns such as recurrent mutational signatures and chromosomal rearrangements are characteristic for distinct tumor types and stages, and point to a complex interplay between stochastic elements, stochastic meaning random, and some rule-governed biological causation, right? So that's all to say that we have a lot of knowledge actually right now about which genes are associated with cancer. The question is, why have we been so unable to do anything about it? And this is, of course, taking the taking for granted the work that people have done so far in the field of cancer treatment, right? So to buy this premise, you should be wary that this author has a particular point of view. One person might think we've made great progress in treating some cancers, and some people might think that it's not fast enough, right? So here's a matter of opinion. But what is true is that, with all of the sequencing data available, I do get the idea that, like, yeah, we've sequenced the crap out of cancers, and why haven't we solved it yet? Is a genuinely good argument to make here, and one that I think is pretty hard to argue against. So even though, right, like, again, when talking about Darwin being very reverent of people's belief, instead of being reverent, these authors here are more standoffish. But I think it works because so far the content of their argument is actually, I think, hard to argue against here. All right, let's move on and we'll take a look at that table. The immense diversity of the functions of the mutated molecular pathways alleged linked to cancer causation and the fact that cancer is a robust universal phenomenon amongst metazoans. This is unnecessary jargon. A metazoan is just an animal, okay? Tells a story of principles of development and evolution of multicellular life. Behind the very existence and near inevitability of neoplasia, so this is abnormal growth, transcending phylogenesis, ontogenesis, and oncogenesis. Too many fancy words, okay? Calm down. However, in the quest for predictive biomarkers and molecular targets, the cancer research community has abandoned deep thinking for deep sequencing. Ooh, burn, dude. Interpreting data through the lens of clinical translation detached from fundamental biology. Again, a very spicy statement here, right? And a lot of zingers, right? If you don't agree with me, you're an idiot. And also this zinger, abandoned deep thinking for deep sequencing is just like insane. What crazy paper are we looking at? We are looking at an essay that is arguing that we should stop thinking about cancer in the context of genetics, that we should kind of move beyond genetics as a causative agent in cancer. And for reasons that we'll mention later in the paper, it's a little more complicated than that. But really, I think the writers are on purpose trying to write in a very shocking manner, calling people out to get them to pay attention, which I think is, we'll talk at the end about how, actually, maybe we'll take a little bit of a break now and talk about how we think about the tone of this essay so far. So for those of you who have stayed with me since the beginning of talking about this paper and have heard me talk through it, what do you think of how this paper is written? Does it offend you? Does it intrigue you? Is this an effective use of language? I'm just kind of curious to get your thoughts. It seems analogous to a pyrogen, something to get you into a defensive state of emotion. I think it's really interesting because for me, because I'm personally not a cancer researcher and don't have like a horse in this race, I don't feel anything at all here. Maybe if I were studying cancer in some form or another, and if I bought into the idea that cancer is driven by genetics, maybe this would offend me. But as someone who looks into cancer research primarily for enrichment and entertainment rather than any serious research endeavor, this doesn't offend me at all. And I think it's hilarious, and actually, like, it perks my ears, because I actually think that if this paper were written any less aggressively, people would kind of think that this paper is just going to lead to an expansion of the existing theory rather than a replacement for the existing theory. So I do think that these choices in language are purposeful, believe it or not. He had better have a good argument. Yeah, I think we'll see what the argument is here, and I'm very interested in what's about to come. I'm not in any of these fields, and I don't feel attacked, but it does feel like he is trying to provoke a response, and I think that's good, for reasons that I'll get into at the end of the paper, because the context that this paper is coming out is a very different context than the context that Darwin existed in, right? Darwin existed at a time where everybody thought that life had a supernatural origin, and so Darwin was right to be soft in his language. Right now, you could say that the context is similar, but for reasons why I'll get into in just a little bit, I don't think that the context today is that similar. I like that old school flamethrower tactics are leaking into academia, and in really eager to see response papers to this one. Dialogue is what's going to be real interesting. Yeah, like, so much of science is dialogue, and the public is really not able to see what that dialogue is. So I'm really happy to be able to show you kind of the opening salvo, I guess you could say, of this quote unquote new paradigm shift, which I don't think is actually that new to begin with. Spoiler alert. That's why I'm very happy to talk about this paper. Nothing in this, I shouldn't say nothing. There's not much in this paper that is new to me personally, but to have it all laid out and to get me in a defensive mode, kind of, I think is really interesting. Well, we're going to address that in just a little bit. A bit blunt. The tone is kind of arrogant, still intriguing because it reminds me of some of my coursework. That's interesting. I wonder what coursework you're referring to. Yeah, the tone is super arrogant and I love it. It's so good. It's so good. I think it uses provocative language. Maybe it does lack striking arguments, which is tried to be covered up. Yeah, we'll get into what the arguments are. Yeah. Okay, so let's head into the next paragraph. So we start off the kind of argument. So that was the introduction, right? And now we're heading into actual argument. Before we get into the argument, let's take a look at the paper, the table that the authors were trying to, let's see if I can, it's kind of annoying. Let's see, let me do this. Yeah, because that kind of clips in an annoying way. So let's expand this for a second. See more of this interesting table that the authors want us to hear about. So this is the background as to why the genetic model of cancer deserves to be challenged. So this is a table of paradoxical findings. So let's just go over the observation column and see how we feel about whether or not the genetic model of cancer should be challenged. So observation 1, some tumor genomes display very few or no recurrent genomic mutations despite a recurrent characteristically abnormal phenotype. And then the details remarks. Examples for cancer that lack consistent oncogenic mutations include rhabdomyosarcoma and ependymoma. I'm not a medical guy. I don't know if Rico's still in chat, but I don't actually know what these cancers are. I think this is a muscle one, rhabdomyosarcoma, and then I don't know what this next one is. But apparently in these cancers, there's not a consistent genetic signature that results in the same phenotype of cancer, we'll say. Conversely, mutational signatures known to be associated with exposure to chemical carcinogens can be indistinguishable from that of sporadic cases of the same cancer. Along the same line, almost every gene in the genome has been associated with cancer. That's just funny. Right? If you look hard enough, and good thing Mylan's in chat, because Mylan, we've been talking about like big N's and how if you have enough N's in a sample size, everything starts to comes off statistically significant. This is kind of the same idea where if you have enough deep sequencing data, all of a sudden you can find associations for cancer across all sorts of different genes, right? Basically the idea of if any of your genes are suboptimally performing, that can lead to cancer, and that's not necessarily useful. So I don't know enough about cancer research to back up the validity of these claims, but I take it at face value that these are good faith observations that are framed in an appropriate way. Yeah. So let's see. What is next here? Genomic DNA of normal tissue contains numerous mutations, including those considered oncogenic. So, the normal tissues can tolerate mutations and stay normal without transforming has long been noted. This is evident in the ubiquitous accumulations of somatic mutations in non-cancerous tissues in individuals with inherited cancer syndromes that affect DNA repair. So this is basically saying that not every time, just because you have a mutation that breaks something that could lead to cancer, it doesn't mean that you're guaranteed to develop cancer. So the link between individual genes and cancer has always been kind of not straightforward. We'll put it that way, right? Complex, right? It's not deterministic on its own, not sufficient. We'll put it all that way. All right. I personally thought cancer to be polygenetic. That's not incompatible with what the authors are saying here, right? But the idea that we should be studying cancer by looking at cancer genes, the authors are arguing that this is starting to become a waste of time. Okay, and that's I think the argument here. Now they're putting it in much stronger language. I'm softening it because I want to paint the argument in the most appetizing way, but I think the authors are purposefully being aggressive to shake us out of this stupor, right? And I think there's a reason for that. Okay, we'll go to the next observation. Indolent and potentially pre-malignant lesions consistently contain driver mutations, yet rarely progress to cancer. This is kind of the same thing, so I'm not going to bother reading all of that down there. Ionizing radiation of cells does not trigger clonal expansion of mutated cells. Instead, multiclonal growth ensures after cell generations. So this is kind of interesting. So let's see what they write here. Instead of the assumed expansion of a single mutated tumorigenic clone after genotoxic irradiation, genetic alterations appear only several generations later and are multiclonal. Thus, ionizing radiation causes neoplasma via cytoplasmic or microenvironmental effects such as cell stress or tissue injury and inflammation that may result in genome instability. Oh, so this is kind of an interesting point here. So what this is saying is that, say you have a bunch of regular healthy cells and you blast them with ionizing radiation, something that we know causes cancer. In their next cell division, they're not immediately cancerous. It actually takes a couple of generations for them to become cancerous. And so that implies, and I don't completely buy it, it could still be genetic for a couple of different reasons I can think of in my head, but it is noteworthy. The fact that they are not immediately cancer means that there's something going on during cell division and during the lifetime of a cell that is contributing to cancer formation, and it's not just breaking the gene itself. Because once you hit that cell with ionizing radiation, those are broken genes already. That's not to say that, you know, things like inflammation caused by bombardment with ionizing radiation can also lead to defects in DNA repair, which can also lead to genetic mutations, which can also lead to cancer down the line. But I do take the point here that the connection is not so strong. Although I think this is a pretty weak point. Tumor cells carrying oncogenic mutations are not necessarily under positive selection, but they are usually too rare to drive progression via selection for new malignant phenotypes. So this is saying, and let's read their explanation, a central tenet of SMT is that cells carrying the tumor-driving mutations should be under positive selection and take over tumor cell populations. Such events are barely seen. However, neutral drift can extensively propagate non-advantageous mutations by chance, creating the semblance of enrichment in genetically heterogeneous cell populations. Marginal positive selections of a mutated gene does occur, albeit rarely. Yet for the logic of precision treatment that seeks to eradicate the tumor, genetic fixation is required, otherwise residual disease is guaranteed. This is saying that the idea that a single mutation in a single cell gives rise to tumors is hard to believe, because a mutated cell, and this is again something that I talked about in our cancer stream, that there is selection, not likely positive selection. So a mutated cancer cell is actually practically not that likely to have an advantage over healthy cells that surround it, right? Because by definition, a mutated cell is damaged in some kind of way. It is a very rare occurrence that is the actual explanation for how cancer works. Again, I'm just saying what the authors are saying without fact checking. I assume that there's some teeth to this, especially because this is not inconsistent with what I talked about in the cancer stream, right? This idea that there is an environment by which a tumorigenic cell has to operate in, and that environment is likely pretty hostile to a cell that has been damaged in some kind of way. Although it does happen in a non-zero frequency. Genetic clonal heterogeneity and reconstructed lineages of cancer cells reveal complex subclone kinetics with branching parallel and monotonic patterns, defying stepwise clonal selection. Oh, okay, so this is that's a lot of jargon. Let me read that one more time. Clonal heterogeneity, so this is talking about the amount of variation in a single population of cells, and reconstructed lineages of cancer cells reveal complex subclone kinetics with branching parallel and non-monotonic patterns. So this is basically saying that progression in tumors is complicated, I believe. I'm not going to get too much into that because that's a little heady for me right now. Cell lineage tracing at single cell resolution reveals rather stochastic clonal kinetics of recurrence, and relapsing tumors often have no lineage relationship to the primary tumor, defying genetic bottlenecks as a signature of otherwise of stepwise clonal selection. So this is basically saying that the idea of tracking one tumor cell into tumor doesn't always really work. That sometimes the actual cells that make up a tumor itself don't necessarily start from the initial cell that is quote unquote defective. Again, interesting, and that's also stuff that I did not know. Oh god, there's a whole second page. Okay, these are more heady arguments that I think I will not get into because this table is extremely long. But I think you get an idea for the vibe that the authors are trying to hit you with, right? So now I'm gonna have to rotate this a bunch of times. Okay, we're back. So given what you've heard in the table, do you think that the idea of the genetic model of cancer deserves to be challenged here? Ooh, it looks like we're about to actually run into an ad here. So that's actually a good time for us to farm some thoughts from chat. Looks like we're just about to hit the ad. I think if you are YouTube Premium, then you should be good here. But yeah, so we'll talk about your ideas. A particularly fortunate ad break here. So Alexandra says yes, Nickname says yeah. I think that I agree that there are enough observations that demand complex answers that I think that thinking about the relationship between genetics and cancer definitely deserves to be reevaluated for the very fact that we could be thinking about cancer in a way that is less efficient, although the authors would have you believe that it is almost flat out wrong. Again, very strong language. I'm peeling it back a little through my own filter, trying to make it more diplomatically acceptable. But I do think that there are enough ideas in there that I think warrant a fresh look on things. Yes, it sounds like there's something wrong. I agree that the relationship between an individual gene and cancer occurrence, like you could still argue, right, that there's still an argument here where, okay, if we lived in a world where we understood exactly what every gene does, are we going to be able to fix cancer from there? And I think what the authors are trying to argue here in this paper in general, and not this part because I'm inserting myself in here, is that a perfect understanding of genes is not sufficient to be able to take down cancer or to have even a basic understanding of how cancer works. I think what's really striking about the way that the authors have laid out this argument is this idea that the model itself seems broken, that the relationship and the predictions that the model makes don't necessarily occur, right? So if cancer really is a function of a single broken cell becoming a tumor, why don't we see that actually happening? It deserves an update to me. Ads are plebs, let us converse undisturbed. YouTube Premium Elite. Thank you so much for being a YouTube Premium enjoyer. For those of you who don't know, if you have YouTube Premium, it also gives the content creators that you love a bit more ad revenue, which is great. I have no YouTube Premium, but I don't have ads because you're a member, Dreamless. I believe that is the case here. I don't actually have a way to know if we're back from ads. Are we back from ads? Anybody in chat want to update me when the ads are done? I'm just yapping into the void. Yeah, if I don't hear anything from chat, then I'll just continue. Oh, my ads ended a while ago. Great. So we can continue on. Maybe there's too many variations of the defective genes to the point where it's very hard to grasp. It might be impossible without the use of AI, maybe, to have that level of complete understanding. And I think even with AI, you might never. I think even with AI, if you stick with a paradigm of genetics, you might never understand what actually happens during cancer. Like, again, even if you had perfect knowledge of what every gene does, because the argument that the authors are making is that this is not a problem of genetics. So it's pretty interesting. Part of the upper class of the YouTube Premium, so no idea there were even ads on live streams. Yeah, it's something that I started that I decided to experiment with just to see whether or not I could be a little more compensated for the YouTube work. Also, welcome Jim the Evo. I don't know, Jim, if you've read this paper because it's caused quite a splash, at least on my science social media feeds, and it's very, very interesting. So for those of you who don't know, Jim the Evo is a microbiology professor, and it's always great seeing Jim in chat. And as an actual professor, because I'm not an actual professor, I'd actually love to hear what you think about or what your understanding is as someone who's probably a lot more updated in the general biology know-how than I am. All right. So with the ads done, let's continue. We are going to review sort of the idea that we are challenging here, which is the core of the genetic paradigm, right? So this is the idea that the authors are trying to challenge. So let's define exactly what it is that we're trying to shift away from. Genetic paradigm here. To revisit the role of mutations in carcinogenesis, so the starting of cancer, we need to recall a central core. Oh, let me actually redo the size of this now that we're out of that table. Sorry about that. Okay, great. That's a lot more readable. To revisit the role of mutations in carcinogenesis, we need to recall a central corollary of the cancer genetics paradigm that links somatic mutations. So these are mutations in the body instead of the germline. So every cell that isn't sperm or egg, to the cancer phenotype and cancer progression, namely the idea of somatic Darwinian evolution of the mutated cell. Herein, random mutations may by chance confer functional advantages, promote autonomous proliferation that increase cell quote-unquote fitness. Then, under natural selection in the cell populations, those cells carrying mutations that confer a fitter phenotype would clonally expand with the fittest clone, eventually taking over the population. This neo-Darwinian scheme is applied to tumors in a loose, qualitative, if not figurative manner, often departing from rigorous concepts of evolutionary biology and eventually becoming known as the somatic mutation theory. The idea of SMT has its origins in 1914 when the embryologist Theodore Boveri proposed that cancer originates from chromatin alterations in a cell. It was then recast in terms of accumulation of a number of genetic mutations by Baer and Nordling in the 1950s, implying a need for increased cellular proliferation that is acquired as a novel trait, but still without invoking Darwinian positive selection. All right, so let's take a look at a visual model here. I did skim the paper, so I know that we have a visual that can go with this. So here, I gotta resize the thing again one more time. 34 words per minute, absolutely crushing the psychologist. I figured I would crush the psychologist. The V-theorem psychologists are they're yappers, and I'm laser focused here. So let's take a look. Not to say that yapping is bad. I actually really love hanging out in their streams, but we do have a very different style of streaming papers, we'll say. So this is what we are calling the traditional paradigm of cancer as a disease of genetics, right? So we first start off with green. Oh, this is such a pet peeve. A good proportion of the population are red-green colorblind, so please stop using red and green, but whatever. Okay, we have green cells here in the first panel. These are healthy cells that don't have any mutations, and then we have a mutation event that causes one of these cells to mutate. And then through the action of selection, right, we are proposing that mutated cancerous cells have an advantage over their neighbors, which allows them to overtake a population of cells. So this is what happens when you get cancer, right? So selection, that clonal expansion increases, and eventually we see that we have an actual tumor. So that is the idea that we are trying to challenge here. Nearby tissues can secrete oncogenic cytokines to nearby tissues. Just because the tumor itself doesn't have mutations, other tissues secrete cytokines that can increase cell proliferation. Yeah, exactly. So that's kind of what the authors are hinting at, that like cells are not organisms in a vacuum, right? That they are part of a whole, and we should stop thinking about them as sufficient to give rise to actual tumors. So it's not, these are not new ideas for biologists, right? And that's why I think the authors can afford to be so aggro, right? Because I've heard all of this before. We even talked about it in the oncogenic stream. So this is literally why the authors are so aggressive. It's literally the invincible meme. Think, biologists, think. Like, you already know the answer here. You've already known in your heart of hearts that cancer is not genetics. That that genetics is not cancer. Your DNA is not your destiny. Think, biologist, think. You already have the answer. I know I'm repeating myself, but it's like the authors are yelling at biologists, being like, you already know, you have had this information before, and you should stop thinking about cancer in this way because you already know that it's wrong. So I think that is why they are actually taking this very aggressive writing style to try to wake us up from our stupor. So it's very interesting, this writing style. All right. So let's move on here. The concept of carcinogenesis as an explicit process of somatic evolution in the neo-Darwinian sense was popularized by Noel's seminal paper in 1976. However, he introduced a pluralistic view. This correlation between observable genetic change and tumor progression does not, of course, prove causality, and some workers believe that at least some aspects of the evolutionary process may represent epigenetic rather than genetic phenomena. So this is kind of hedging this idea that your DNA is destiny. Sometimes it's what is being done to your DNA that causes destiny. So, this is in the jargon tag, jargon command, but epigenetics is what happens to the A, G, C's, and T's of your DNA, right? So your A, G, C's, and C's, and T's that your DNA code is written in can be further modified by sticking chemical groups on top of them, which can cause the actual expression of those genes, so the conversion of DNA to mRNA to protein, to be modified. So this is acknowledging, okay, maybe the actual letters of your genetic code is not the end-all be-all when it comes to cancer. Maybe there's this other level of genetic control that is represented by stuff happening to your DNA. I think that's fair to say, right? It's maybe acceptably myopic, right? Short-sighted to believe that the A, G, C's, and T's are your destiny, because we do know that there is this additional layer of regulation here. They are positing things we don't know as rebuttals against cancer being genetic, which is not an argument. Let me think about that. They are positing things we don't know as rebuttals against cancer being genetic, which is not an argument. I think that this is more about talking about the shortfalls of the existing models. This idea that there are answers that cannot be gotten from taking a myopic view on how cancer works. And we'll talk about what the authors want to do to replace this kind of thinking. But I think it's an interesting conversation to have, because by the end of the paper, I'm really curious as to whether or not you will fall into, which camp you fall into. Because, spoiler alert, I'm very open to the thesis of this paper. So I'm already finding myself kind of sympathetic here. But that doesn't mean that I'll stop being critical, right? Because there are certain things that I'm looking for in this paper, and if the authors don't address them, I'm going to be a little upsetty spaghetti, right? Okay. These cautionary words have been largely left out of the equation of cancer genetics in the following 50 years. I don't really think that that is true. I've heard plenty of arguments being made for epigenetic control of cancer as well. So I think this is maybe a matter of perspective. I'm not a cancer researcher, but as someone who consumes cancer content passively, epigenetics, in my view, is not really ignored, although that could be a factor of, how I was trained personally. Despite its rapid ascendance, somatic mutation theory remains a conjecture supported by spotty selective evidence rather than a precisely formulated theory, notwithstanding its use of mathematical formalisms. Specifically, somatic mutation theory left out broader considerations of historicity and physiology. First, autonomous proliferation appears to be a latent default state baked in organismal existence already in the last universal common ancestor, and is not an evolutionary novelty that is attained through natural selection in tissue. Second, the expansion of faster proliferating cell clones is not limited to tumors, as evident in aging normal tissue, and yet chronic diseases where hyperproliferation results in oligoclonal expansion. Yet convenient for its simplicity but misleading in its meaning, the neo-Darwinian principle has become the unquestioned truth in tumor biology, which, as Table 1 shows, is responsible for many of the cracks in the genetic paradigm of cancer. Is myopic in jargon? I know it means short-sighted, but not everyone might. That's a good point. Yeah, it's not in jargon. I was only thinking of the view of like what is considered biology jargon, but myopic is in fact like a pan-academic jargony word. I should just refer to it as short-sighted. Isn't DNA damage from alcohol/smoking part of epigenetics? I am not enough of a geneticist to know the answer to that, although I'm sure smoking and stuff like that causes epigenetic changes in some way or another, for sure. Yes, I don't completely buy the argument that we've been ignoring epigenetics. I guess it kind of depends on who you ask, though. And then there's some heady stuff about clonal expansion and Darwinism here that I kind of take for face value here because I don't have a deep enough understanding of how evolution works on a mechanistic mathematical level to say otherwise, right? I'm not going to step out of my field here. Okay. Robust manifestation of natural selection in tumors would be hard selective sweeps, the complete overtaking of the tumor cell population by a malignant clone carrying the driver mutation, along with linked but neutral mutations representing one step in Bolstein's multi-step regression model of linear evolution. Again, the evolution stuff is a little heady for me. The genetic fixation, as it is called in population genetics, of the oncogenic allele in the tumor cell population would be required for the logics of precision oncology that seek to target a mutation to eradicate a tumor. So this, I think, is worth pointing out here, right? That there's this frustration that we've been sold that with the advent of deep sequencing, that we would have precision medicine, that, you know, we're always on the cusp of having treatments that are tailored for people because we are increasingly knowledgeable about the human genome, and that hasn't happened. And so you could make the argument, well, it hasn't happened yet because we just don't understand enough yet. But I think the author's frustration here comes from maybe we will never get enough information if we keep on looking at cancer in this view. I think they're overstating how ignored some aspects are a bit at a few points. Yeah, exactly. I have the same feeling here. It's part of the general rhetoric style, I guess. Yeah, they're purposely being inflammatory here. Again, like nothing here I'm shocked by, except from the fact that there are some tumors that don't have oncogenic mutations. That's pretty interesting. Let's see. Where did I left off? Where have I... In reality, a tumor contains thousands of genetically distinct, apparently independent cell clones, giving rise to genetic intra-tumor heterogeneity that came as a shock to precision oncology world. This finding led to attempts of adjusting precision oncology by targeting trunk mutations shared by every cancer cell, and by combination therapy directed against multiple oncogenic mutations, approaches that still adhere to the somatic mutation theory. So this is something that I do kind of agree with, in the sense that, or rather, this is something that I am shook up by, because I had never bothered to challenge the trunk mutation idea, and I've definitely heard people like present at conferences this idea that we should be targeting mutations that are common amongst cancers to try to get at the root of the problem. That if we just fixed the root mutation in cancer, because of the model of having multiple mutations elevates your risk of a certain cell becoming cancerous, if we just hit the root mutation, the one that cancers share, that we should be able to stamp out cancer no problem. Question is, we've designed stuff that technically works on that principle, but it hasn't really been that successful yet. And again, I'm not a cancer person, so I don't know. I'm taking their word for it that we haven't had that much success. And of course, if we did, then we would have probably heard about it in some form or another here. So I think that is something that my mind is open to changing on, right? This idea that the trunk mutation strategy has not been super helpful. And again, you could argue that, okay, maybe we're just not doing a good enough job at shutting down those trunk mutations. But I am now being swayed into thinking, well, maybe that's the wrong approach, because that's an extension of an outdated theory to begin with. But okay. There's no good sense in disproving a theory or trying to do a paradigm shift if you don't have a paradigm to shift to, right? Otherwise, this just comes off as normal criticism instead of a paradigm shift. Like, sure, there's stuff that falls through the cracks here, but unless your way of thinking about cancer is better, this is not really anything that surprising. Yeah. But even if a clone appears to expand faster and dominate the tumor, there is little proof that a complex tumor phenotype is sculpted by natural selection for the specific oncogenic mutations that produce said adaptive traits, as neo-Darwinism postulates. It is likely correct that simple Darwinian evolution is at display in the case of evasion from targeted therapy, notably kinase inhibitor drugs, wherein a point mutation in the kinase protein interferes with multiple aspects of kinase activity energetics, as best studied in the oncogenic protein CBR-ABL or AB1, as it becomes resistant to the highly selective inhibitor imatinib. So that, I think, is a cancer drug with an actual reputation and name. I've seen that name come in multiple forms. But adaptive selection of such molecular phenotype is a far cry from the suggested generation of complex traits that embody the hallmarks of cancer by natural selection during tumor progression. Thus, SMT suffers from the same shortcomings as neo-Darwinian evolution, in that it only explains distinct localized adaptive traits obviously linked to the selection pressure, but not the emergence of complex higher-order biological function. So this is, again, I didn't read the paper beforehand, but the idea, I could predict where the author is going next, right? We have drugs that try to work on this model, but they don't appear to be great. It's worth noting that imatinib and trastuzumab are really quite successful examples of precision medicine. That's a very good point to make and not background that I have, so thank you for contributing that. They of course have issues, but they have drastically improved on the previous standards of treatment. Yeah, exactly. This is like what I had mentioned before, that I am open to being swayed, but I'm not a cancer person. And so it's a matter of perspective whether or not you buy the author's argument that we've not had enough progress, and that is something that reasonable people can disagree. But thank you for that input. And I've lost myself. Okay. One must also not forget that neutral variants can be enriched without being selected for because of random genetic drift in relatively small cell populations. Okay, so this is something I'm going to address in a future video, actually, but, mutations don't always have to give rise to selective advantage or disadvantage. You can have mutations that are, sorry, let me back up for a sec. The prevalence of a certain genotype or a certain mutation can change not because of selection, but because just random mathematical processes. Enough versus not enough is ultimately kind of arbitrary. I agree with that. But that's, I think, where, again, you can reasonably disagree. Where was I? In finite cell populations, in the mathematics of random birth-death processes alone accounts for the highly skewed allele frequency distributions that defy our numerical intuition. The rich get richer without being more capable, although neutral drift may be overestimated because of late-appearing subclonal driver mutations. Okay, this is an argument from math, and I'm not going to step into that because I don't know enough evolution math, to really say that that's true or not true, but I go, okay, I will buy that argument for now until someone else tells me otherwise, right? I'm a casual observer in that particular sense. Complicating the picture of somatic evolution are interactions at a higher level like those between subclones of cancer cells that affect fitness of the entire tumor, creating non-cell autonomous dynamics. So again, you can go for jargon for that. So this is saying that so something that is cell autonomous is a feature, behavior, result of a cell within a processes happening within their cell. Non-cell autonomous processes happen across cells. So cells talking to each other, causing them to talk to their neighbors, to affect their neighbors. I forget who said it earlier in chat, but the finding that cells can talk to each other and induce a cancer-like state is included in this idea. Beyond direct mutual stimulation, the distinct clones may functionally complement each other because of their specialist capabilities that support the cancer cell community. For instance, though metabolism that alters pH and nutrients in the intracellular space, detoxification, promotion of angiogenesis, alteration of tissue mechanics, or averting anti-tumor immunity. Oh, that's a long sentence. Beyond direct mutual stimulation, the distinct clones that may functionally complement each other because of their specialist capabilities. These biological functions are not sufficient to grow a tumor themselves. Conversely, some tumor cells may keep their more malignant neighbors at bay and must not be killed. That's interesting. So a lot of stuff there that I haven't really thought about here. like in organogenesis. Yeah, Alexandros, that's actually something that the authors will wrap up their argument with, is this idea that maybe we should stop thinking about cancer as starting from a single cell and start thinking of cancer like its own organ. Yeah, and they're about to get into that here. Thus, if we view the tumor as an evolving ecosystem, as has become fashionable, we must also embrace the evolutionary/ecological principles that have emerged in the past few decades and that question the dominance of the simple scheme of natural selection of neo-Darwinism. This included punctuated equilibrium, stasis with sudden bursts of evolutionary events, group selection for fitness of a group due to social behaviors, niche construction, evolving organism modifies its own selective environment, and structuralism. Natural selection must obey constraints of physics, chemistry, and geometry, which contribute to shaping the phenotype with no selection. Okay, a lot of things there, but the author is saying thinking about cancer in the view of individual cells, it just doesn't work anymore. We got to think about cells, or we got to think about tumors, less about cells in the language of cells, and start adopting the language of its own thing, its own organ, its own tissue, etc., etc., etc. And then applying these ideas in a responsible way, right? The idea that a single cell is gaining an evolutionary fitness advantage over its neighbors, the authors are arguing that that just doesn't pan out when we take a look at the actual data. Of particular relevance is the group of evolutionary theories that emphasize phenotypic plasticity. These theories all suggest that the direction of evolutionary adaptations of populations tracks the physiological adaptation of the individual to its environment as enabled by regulated phenotype plasticity, such as acclimatization. These theories include genetic assimilation / genomic accommodation, the Baldwin effect, and canalization and phenotype first dynamics. Okay, a lot of jargon going on here. These mutationless mechanics may lubricate and channel natural selection, thus they actually support Darwinian natural selection by making evolution of adaptive traits more likely in the view of unlikely mutations that need to happen. Thus, despite being largely absent in somatic mutation theory literature, all these theories indirectly support Darwinism by considering non-genetic plasticity, thereby relaxing the rigid one genotype, one phenotype constraint. Okay. Oh, okay. We have reached the end of that very meaty section. Let me kind of attempt to summarize what's going on here. Yeah. So again, this is all saying sort of the same thing of that. Let's see. I don't know what the Baldwin effect is, etc. But again, just driving home the point that we should stop thinking about cancer as a disease of cells and start thinking about them as a disease of organs or complex environment and tissues. Okay. Yeah, and thinking about the rigid one genotype, one phenotype constraint. This is, I think, a jab at scientists who are primarily working under the assumption that they can crack cancer by what do I want to say? That they can crack cancer by studying individual cancer genes, right? So we kind of already know that maybe this is kind of an irresponsibly short-sighted kind of idea. So they're trying to snap us out of this. Astral says, words per minute holding strong. Yeah, I think that if I were better at yapping, my like Cider and Myelin are, I would have a much lower words per minute. But because I think that like once I get on a train of thought, I find it very hard to get off that train of thought. And if a paper sends me on a train of thought, I find it actually kind of hard to leave that kind of train. So yeah, there's a lot in this particular section of the paper that I think is a bit repetitive for me personally, because I'm not like an evolutionary guy. So I think this is mainly talking to other cancer researchers that like, again, as Chad has pointed out, these are not really new ideas, but I think the language is really aggro because they're not new ideas. And I think what the authors want to happen in the field itself is that we stop doing experiments based on this idea in particular, relaxing the rigid one genotype, one phenotype constraint. Like if I were to summarize what has come before in this paper, it would pretty much just be this line here. We need to relax the thinking that the link between genetics and cancer is all that strict, and we need to reprioritize as a field how we should attack cancer experimentally. And for those of you who are saying that this isn't necessarily incompatible with the idea that genes drive cancer, take a look at the next title here: Overcoming the Limitations of the Current Paradigm, Expand It or Replace It. So you could say that you could just take what is known here and do more of it, right? Like, this is not incompatible with somatic mutation theory. You just add an addendum that the genetics are really complex. And so I think here is where I think I am most likely to be sympathetic to a change in paradigm shift, that if there is a convincing argument that we should stop thinking this way, then that is worth noting. And again, I've already said I'm pretty sympathetic to this mode of thinking, right? That, like, I buy it, right? I buy it. I'm good with thinking of cancer as not primarily driven by genetics, just because that is my personal acceptance, and reasonable people can disagree, I think. All right, so before I head into the next section, oh my gosh, dude, the timing. So we're about to head into another ad break, so I want to get a temperature read from chat about what you feel about the argument here so far, right? This idea that we should stop thinking of cancer as one gene, one phenotype, or even a collection of genes to a cancerous phenotype. So what is the actual temperature in the room about how likely you are to accept a new theory to replace this old theory here? And so let me actually also kind of define what I mean by a paradigm shift. So, you can, if you're, so if you're thinking about cancer and you load in cancer, and the first thought you have is this model of one cell, mutation, advantage, clonal expansion, tumor, instead replacing that with something else entirely. For me, I think that I'm very comfortable with replacing that idea here. Including the scope of RNA-omics and proteomics. So Marlon is basically saying maybe this model of cancer is incomplete, and we should not be reliant on things that modify DNA to be the only way that we understand how cancer works. That we also have to include how the mRNA works, how other RNA works, and what proteins are involved. And I think that given my knowledge about this theory, this not theory, I shouldn't call it that, this model of how cells work together as not being primarily driven by DNA is one that I'm familiar with, because a lot of research in the universities that I've been a part of have been making this argument for years now. The idea that we should be looking at cells, we should be looking at a lot of different things all at once and using larger sets of data to understand the complexity behind an actual tumor. So not only looking at DNA, we are hamstrung by our fixation on DNA. We might be losing out on a lot of stuff. So certainly incorporating ideas like RNA and protein are going to be, I think, important in this discussion here. I particularly find the lens of the Waddington landscape to be an interesting view to apply here, and they will apply that here in the upcoming parts of this paper. I'm not really familiar with the old theory and was kind of under the impression that it sort of worked like this anyway, assuming that I'm understanding it, which is yeah, it's possible, depending on what background you've had, that people are already starting to think of cancer as a disease of tissue and not of individual cells. I think it's okay if we don't throw out links we already know works from practice, like causality, like causally. Yeah, like the paper is coming off a lot stronger, but I think where I land is that nothing that is being said right now necessarily conflicts with the old stuff, right? It's we already kind of know that it's a little flawed, but we should be moving away. Again, this is what they're trying to make the argument. We should be moving away from experimental questions that rely too heavily on understanding genes themselves, and we should be focusing more heavily on what cancer does. I'm in the altering existing theory camp. Yes, epigenetics of strange genome instability are a welcome addition, but mutations are important for cancer and can even cause the epigenetic changes. I'm quite underqualified here to get an overview. Fair enough, fair enough. Cell niche is not independent of gene presence. Yeah. Tissue, microenvironment, tissue matrix, signaling, of course, are all involved, but I don't think you can get all the way to cancer without also having some mutations occur. So, Kotaro, there was a table earlier in this paper that states that there are some cancers actually with cells in the population that do not have cancer-causing mutations, which is challenging this idea that getting to cancer requires genetic mutations in cells. So you are right in the sense that mutation is often involved, but I think we're about to see what the authors want to argue is a more appropriate, limited view of the role that genetics plays in cancer. And so I don't know how the authors are going to do that here. I'm kind of making what I think the arguments are going to be for the authors, because again, I'm already very sympathetic to the idea of replacing our existing paradigm. Again, because that influences the way that scientists think. It's not just technically. Okay, so maybe this is important context. It's not necessarily about whether or not genetics causing cancer is right or wrong, but is this idea useful anymore? And I think that I am leaning towards being sympathetic that it's not useful to think about genes driving cancer in a very serious manner anymore, that we have evolved as a field past the point where we need more information on what individual genes do. And it's time to take a different approach. But what approach that is, I think, is, at this point of the paper, not exactly obvious here. All right. So we're probably back from that ad break now. So overcoming the limitations of the current paradigm, expand it or replace it, calls for overcoming the blinders of the genetic paradigm have a long history. What is new is that first, the same omics technologies that employ to identify new oncogenic mutations have been used to better understand non-genetic processes in tumor genesis that involve cell plasticity and tissue organization. And second, the quest for drug targets or biomarkers have meanwhile reduced much of cancer research from a scholarly biological discipline to a transactional operation concerned with cataloging molecular alterations and finding associations than with understanding fundamental principles and using formal theory. The zest for intellectual discourse on the nature of cancer, the type of scholarly discourse that advances all scientific disciplines, is long gone or viewed upon with suspicion. So yeah, this is, I think, again, where I am very sympathetic in terms of why I think replacing this definition is actually pretty helpful. That there's this attitude that, like, oh, okay, so what we'll do is we'll chuck a bunch of cancer cells in a single cell sorter, and we'll just see what, like, the genes are, and then that's going to allow us to get a drug out of it. And we'll publish, and we'll try, we'll sink millions of dollars and try to develop a drug out of it. And sometimes it'll work, but most of the times it won't. And let's just keep on doing that ad nauseum. And yeah, I don't have a deep enough knowledge of cancer research to understand, like, how slow or how unoptimized this particular paradigm really is. But again, as a casual observer from the outside, I'm like, okay, I buy that, like, if we're a little intellectually lazy about being like, okay, we'll study this oncogenic gene, this single oncogenic gene, and try to get a drug target, I get how that can be viewed as not taking the whole of cancer all that seriously. Although, again, I have to emphasize, it's not that that is a bad approach. It's just that, like, cancer might be too complicated for that approach to work very well anymore, right? And as previous commenters have said, it's not like that it never works. It's just that cancer is really complicated. And if this was the way to go about it, we might have expected more progress by now. No model is true, but good models are the ones that work, paraphrasing from other model discourse in other fields. Yeah, exactly. My research advisor used to say all models are wrong, but some models are more useful than others. And I think that is fair. And replacing the model of genetics is, I think, it's not as drastic as I think the author's inflammatory writing makes it out to be. PhD biomedical sciences here. We already knew this in 2016, but you're right. Some researchers don't seem to have moved in the direction with their grants and approach to experimental design. Exactly. Thank you so much for confirming that, Kotaro. As someone who is also in this kind of field, that like this, I've been saying this the entire paper, that this is not new information, but this is more like a wake-up call to scientists who are still going, we have a gene target. Let's make one drug to target this gene, and then that'll be what fixes cancer. Again, not to say that that never works, but maybe because it's failed so often, that might not be the right approach to take anymore. That is from George Box. All models are wrong, some are useful. Oh, thank you so much for the source of that quote. I'd also love to hear, Jim, what your thoughts are currently, and how not only about what the paper is saying, but how I've covered it so far. A friend of mine made the Einstein box plot. Y is model wrongness and X is simplistic. That's pretty fun. That's awesome. This also kind of bleeds into other sciences too, right? Where it's often said that, it's often used as a bludgeon against climate science, where, climate models are always predicting that we're 1 or 2 years from being underwater. But our understanding of those models continues to improve, and so as those models continue to get more complex, they'll also be more in line with observation and research. Yeah. All right. So where were we? Okay, since one should not postulate the end of a paradigm without specifying the alternatives. Yeah, that's what I was saying, right? Like, you can't just say your idea is crap. You got to give us something to replace it. So exactly. Since one should not postulate the end of a paradigm without specifying the alternatives that may correct or replace it, we will discuss here 2 sets of ideas that have been sidelined by the dominance of the genetic paradigm of cancer. First, that cancer is not a disease. So let me back up here. When I say that we knew this answer already, what I'm referring to is, like, people have talked about this idea that cancer is a disease of tissues and not cells for quite some time. And I think the reason why the authors are so aggressive is that, again, we already know in our hearts that this is true. So let's stop designing experiments that are informed by the simple one gene, one cancer hypothesis, right? We shouldn't be trying that anymore. Or if we do, we should have really, really, really good reasons to do that, because the opportunity cost is that we're ignoring some more interesting, fruitful lines of inquiry instead. I think I'm starting to get the idea. The current approach is more like a case-by-case basis. It's more like... So I was playing with this analogy in my head that, like, the Bohr model of the atom, right? You got your nucleus, which is made of protons and neutrons, and then you have electrons that orbit the neutrons in these circles. And the Bohr model is useful, right? It's cool, it's all right. Like, it gets the point across, and you should still learn it because it helps you understand what the atom actually is. But then when you get to undergraduate, you start learning that, neutrons are surrounded by these clouds of electron probability, that they're not really circles, that they have all these weird shapes and all this other nonsense about them, which is why I never got into chemistry. But that model is more accurate. So this is, I think, saying, okay, we have the genetic theory of cancer, which is okay, but we should stop treating it like it's reality. It's kind of like the cancer equivalent of thinking that an atom is a collection of protons and neutrons surrounded by circles of electrons. It's much more complicated than that, and we are losing ground against the fight against cancer if we don't get our butts in gear and start designing more complex experiments here. I'm enjoying the paper. I think, as an evolutionary biologist who's done some cancer work, I agree with authors, but it isn't aimed at me. I feel it is aimed at medics and pharma folks who design and give drugs. I think that's a really good point, that we should consider who the author is trying to wake up. Again, because, like, I feel no resistance to this, right? Like, I'm very, like, on board with this idea of, yeah, I mean, it seems kind of silly that, if the model is so simple, we haven't solved cancer yet, so what gives? So it makes sense that this is more aimed at the medical field and big pharma to be like, hey, you are sinking millions of dollars, billions of dollars into ideas that are outdated. Maybe they're not wrong, but they're slowing down progress. Like if we're talking about how to increase, this is such a whatever. I'm not gonna go down there. I'm not gonna go down there. It's very, very off. Mathematically, modeling Bohr's atom gives very screwy results. Exactly. And I think that's the same analogy that the authors are trying to make here, that Bohr is fine for learning when you're not capable of learning things at a more complex stage. It's a nice introduction for how the atom works, just like how SMT is a nice introduction for how cancer could work. But it's a lot more complicated than the Bohr model. It's a lot more complicated than somatic mutation. All right. Okay. Let's continue here. First, that cancer is not a disease of the genes, but of gene regulation and thus of the cell. We must consider the dynamics of collective actions of genes and in a network that governs cell behavior. So again, this is what I've been saying earlier. It's not your DNA is not destiny, but it's the stuff that your DNA does that's actually important, right? Creation of mRNA, creation of protein, those proteins do stuff, etc., etc. Second, that cancer is not a disease of the cell, but of tissue. Again, I already said that. We must first consider principles of tissue organization. Both of these alternatives are discussed in detail below. To interpret observations afforded by powerful new technologies, we more than ever need a solid framework grounded in biological principles that must be rigorously formulated but may not yet be mathematically formalized. Cancer research needs to return from current transactional activities to the scholarly investigations enjoyed by generations of biologists who have placed new findings in the context of organisms, their development, physiology, and evolution. So this is a call out to me specifically. I've actually mentioned this to Octopus Lady in DMs, where we were chatting about biology, and I called her the biologist that touches grass because I am very far removed from the biology of an organism. In the work that I did as a graduate student, as a researcher, is mostly focused on cells and how cells talk to each other, and I very rarely thought about their impact on the organism as a whole. I mean, I wrote about it in grant proposals, right? Like, you still have to say that you're learning about organisms, but in reality, my research did not translate very well into organismal biology at all. So this part is a call out post to me. But I'll say that my attachment to thinking about biology in the context of cells is one of personal preference, because I find cells to be interesting, and I find that the models by which I learn are more in line with thinking of cells as individual units rather than contributions to a tissue. So it's like a weakness of my own biological training, but I think it's a weakness that I accept because I'm no longer a researcher, so it doesn't really matter. If I had to make a living writing grants and getting things published, then maybe I would be like, oh, I gotta get my button gear. But now I call myself a biology bard, right? I sing the tales and songs of biologists more capable than me, so I don't actually take any offense to this call out post. As Philip Ball points out, so this is a editor for Nature and the writer of a very influential book, I think called How the Cell Works or something, influential biology writer. On the apparent lack of progress in the biological sciences may be related to the adoption of a mistaken paradigm. Probably thinking too short-sightedly about understanding organisms from the viewpoint of cells. Herein we will illuminate the path out of the genetic paradigm, touching on scientific, epistemological, and sociological aspects of cancer research. Ooh, look at that. Acknowledging that scientists are humans, right? Like, again, this essay isn't a research paper. It's not presenting new data, really. This is an argument, and an argument is being made to humans who also do research, right? So acknowledging that this is argument, that we are talking to each other, we're having a dialogue, and it's not like, I'm bludgeoning you with results. It's like, here's how you can accept a new way of thinking, which I think is smart here. A biology sea shanty? Maybe one day. How Life Works, the user's guide to the new biology. Yeah, that's the one. Haven't read it myself. I wonder if I should review it on stream or something as content. It'd be kind of interesting to review kind kind of more casual literature. Not saying casual like in a bad way, but more accessible review type literature than the stuff that I'm used to reading. Okay, so authors of the paper, how will I make my way out of the genetic paradigm? Please show me the light here. So, history of anomalies before the era of molecular genetics, the normalization of cancer. So talking about sort of maybe why we feel so defensive over our existing paradigm here. If so many sequencing results are inconsistent with this somatic mutation theory, why do they not trigger a rethink amongst cancer researchers? Revisiting an overused idea of the philosopher Thomas Kuhn on how science advances anomalies, findings inconsistent with respect to established knowledge, should have begun a shift long ago in the paradigm of what he calls normal science. So, you know, referencing Thomas Kuhn is a real pro gamer move. He's a very well known rhetoric of science kind of philosopher, and if you ever venture outside of just studying your books and want to think about science more in a philosophical light, you will almost always come across Kuhn's writing, as I did as an undergraduate. So it's really fun to see that, like, the rhetoric of science class I took as an undergrad, I'm like, hey, Thomas Kuhn, I know that guy. If you are like someone who wants to get into the field of science, I highly recommend taking at least one philosophy of science class, just because, you know, not only does it enhance your understanding and appreciation of science as a philosophy and as a mode of thinking and a way of life, it also gives you an appreciation for when truly staggering and amazing things happen in the field of science. Is that the one, science advances one death at a time, or something where dogmatic figures are removed in order to allow new fields to sprout? I could not tell you with exact detail exactly what Thomas Kuhn's like framework was, but Thomas Kuhn, I believe, is the paradigm shift guy. Or at least I think one of his students was the paradigm shift guy, like talking about how we, as, you know, a species, as humankind, make large advances and chaotic shifts in the way that we understand things. So citing Kuhn is a real power move, because essentially the authors here are positioning themselves as important as Darwin was to evolution. You know, again, real power play here evoking Kuhn, I think. So this is basically saying, like, we've been like racking our brains over this cancer problem for decades, and our predictions have not exactly been working because people still get cancer. In fact, a lot of people still get cancer. It's a very common disease, and even with, you know, 75 years of molecular biology, with the ability to pinpoint precision, manipulate genes, you know, CRISPR allows you to surgically change whatever gene you could think of. With all these freaking tools, we still have this stupid, annoying, deadly, tragic disease around. And don't you think it's time for a different way to think about things? Certainly I am sympathetic about that idea. Puzzling findings at the organismal level predate the recent wave of hard-to-explain results produced by deep genome sequencing by more than half a century. They question the notion of cancer as a disease of the cell that is irreversibly transformed to a malignant phenotype by genetic alterations. On center stage is the normalization of the cancerous cell accomplished by physical contact with a pertinent embryonic field, which has prompted some researchers to view cancer as a problem of cellular differentiation. So cancer does not exist in a vacuum, right? One cancer cell gets bonked, they have their genetic code changed, but it's still in contact with the regular tissue around it. So cancer has to be, you know, whatever model of cancer that you have has to have some element where a cancerous cell talks to other cells. Normalization is perhaps the most prosaic anomaly to the mutation paradigm. When Peter Noel popularized clonal evolution of cancer cells, Beatrice Mintz's group presented evidence that you know, I'm talking so much and so fast, I'm starting to get lightheaded. Give me a second to drink a bit of water here. That's something that's been in my recent coursework. Yeah, I think, again, if we're thinking about who this paper is for, I think most of us studying this already get it. It may be trying to convince the last few scientists who are so stubborn in this kind of big way, and to give permission to people to start talking about cancer as a disease of tissue rather than cells. Because I think you need something as spicy as this to be like, okay, so we're done talking about SMT. And I think that's interesting. Speaking of normalization, right? Okay, let me catch my breath here. My goodness. So, as I catch my breath, how's everyone doing out there? How are you hanging in with this paper? Are you enjoying it so far, or what's the temperature in the room like here? I really need to drink water. It's chill. I'm glad. Okay, let's continue. When Peter Knoll popularized clonal evolution of cancer cells, right? So you get pinged, you break a cell, and then that cell expands, and that becomes cancer. Beatrice Mintz's group presented evidence that teratocarcinoma cells injected into the mouse embryo gave rise to cancer-free chimeric mice in which the mutated cancer cells were present in most organs. Okay, so this is interesting, right? So Beatrice's Mintz showed that if you inject the literal cancer cells of one mouse and inject it into a healthy mouse, an embryo, you get a result in which those cancer cells still exist, but they no longer give rise to cancer, that they spread, and the mouse might be actually fine. So what is happening here is a phenomenon called normalization, that these surrounding cellular environment is beating down the urge of these cancer cells to become cancer. So that's really important. It's a really important idea and a really important dimension of cancer that I think I acknowledge is kind of ignored in a lot of the ways that people talk about cancer these days, right? So it's, again, we already had the answer in our hearts, but people still talk about cancer in the context of somatic mutation theory, and this is something that I think is interesting to note, right? This idea that healthy cells can suppress cancer. Very, very cool. Normalization. So again, like talking about if this new model is more helpful, we can already start to see that if we incorporate normalization into whatever model of cancer that we're thinking about, we already see the potential for different classes of drugs to be created, right? Because a lot of drugs are made from the perspective of, okay, so we know these genes are broken in cancer, so if we introduce a drug that fixes those genes, we should beat cancer. And if we incorporate the idea of normalization, what we're actually saying is maybe we could design drugs that help healthy cells beat cancer cells. And it's that kind of like paradigm shift that's really important, because now your third eye opens and you're like, oh my gosh, maybe we've been too narrow-minded this entire time, that trying to fix cancer cells by targeting their broken genes might have diminishing returns after a certain point. Coon woke me up like a sleeper agent, though. Exactly. When you hear the word coon as a science person, you're like, oh my gosh, coon. Very, very cool. Very, very cool. Alrighty. It could be a new way of, exactly, Ethan. It could be a new way of treating cancer. Normalize it into our body. Maybe cancer is friend and not foe. Yeah, like this is a very Undertale kind of like mercy. Like, oh, if we just show the cancer enough molecular love, it won't grow to be a delinquent tumor, which is a very interesting drug design strategy. And I think, you heard it here first, folks, that if you see drugs that start to become this class in the future, then and maybe this paradigm shift is partially responsible for that kind of innovation. Normalization is also observed during the maturation of human tumors, whereby proliferating tumor cells sometimes develop into mature non-proliferating cells, as most clearly seen in neuroblastoma. Hey, look! Neuroblastoma showing up here, where cancer cells become ganglion cells. Oh, I didn't know that. That's wild. So this is saying in sources 150 and 151. Again, look at how much work went into this paper. It's absolutely insane. We are so shallow into this paper, and we've already hit 100 plus references. So apparently, I guess, in neuroblastoma, sometimes cancer cells differentiate into actual functional cells. I had no idea that happened. I'd probably have to read those sources to check how real that is. But that's very surprising to me. I'm being reminded of hormone-dependent cancers, but there are other ways cells communicate, and normalization may take advantage of those communication pathways. Absolutely, right? So hormones are long-distance communications, but cells also talk to their neighbors, right, through their membranes. So thinking about ways to manipulate that, like I hope that is giving the biology students in the chat right now a lot of ideas about what they can do with this information in the future when you all become very successful scientists, right? Like we are so potentially past the point of this gene is broken, let's design a drug, and now thinking about how do we repair, so how do we get these cells into therapy and get them to start communicating properly again. In the hundreds, giving 30 words per minute. Yeah. That sounds it sounds like I have a pretty consistent word per minute here, which means again, once you get me on the train track, I'm very hard to tear off. Like I could not deviate from, you know, I'm very fixed on topics, I should say, which may or may not be a good thing. Certainly, hopefully enough of you are still in chat who like that kind of like one track, like I just do my thing and I go. Yeah, I actually feel yeah. Sorry, my brain zoned out a second. Okay, so we're talking about normalization. Hopefully that gives the biology students ideas in the future when you all become successful biologists, that there are dimensions out there, and you should not be limited in your understanding of cancer. All right. In addition, some therapies, including target selective inhibition of oncogenes aimed at killing cells, often trigger a wave of differentiation of the immature cancer cells into specialized post-mitotic cells, despite not being designed as differentiation therapy. So, okay, so an offshoot of some treatments cause some cells to flip into a different cell fate. This is most dramatically apparent in differentiation syndrome, the dreaded surge of differentiated neutrophils. It's me. It's me. Oh, but it's preceded by the dreaded surge. Okay, so I'm learning something here. The dreaded surge of differentiated neutrophils in the treatment of acute myeloid leukemia. Oh, so cancers that involve the stem cells that become neutrophils, if you treat that blood cancer, you get a surge of extra neutrophils. And the fact that this is called differentiated syndrome means that that surge of neutrophils is probably not too good for you. Wow. Holy cow. Whoa. Okay, I'm learning something. I said I knew most of this already, but I didn't know the specifics here. So I actually had no idea that certain cancer treatments cause a surge in differentiated neutrophils. That's actually kind of crazy. You got an ad? I didn't even see the ad timer come up. Okay, I'll just yap a bit, and then you can all tell me when the ad is over. What do neutrophils do again? So neutrophils are white blood cells that go around and are the, I don't know what you would call them, the backbone of the innate immune system. So they go around, they throw DNA around and trap pathogens using their DNA sticky nets. And they also have some phagocytotic functions where they can eat pathogens and destroy them. Although that's primarily the domain of the macrophage, right? But, you know, there are overlapping functions here. Also immune signaling and stuff. But the main unique power of the neutrophil is the ability to eject our DNA and use it as a net to trap and kill pathogens. Ad is over. Cool. Yeah, I don't really know what the heck is happening with how prevalent these ads are. I don't really think it's like Twitch, where I think Twitch it's like pretty consistent here. And hopefully the ads are not very invasive, and like if I see how much I earn from ads and it's like pretty negligible, I might not do ads anymore, but you know, we'll see. Yeah, well, we're experimenting. Okay. Okay, so this is really cool and maybe a topic for a video, because I could see, you know, a good thumbnail. Cancer treatment causes immune cell to proliferate. I don't know. That's such a bad thumbnail, but something like that. Would this be an interesting topic to tie into your VTuber lore? I already have lore. My lore is that I am a neutrophil that is chemotaxis deficient and is bad at killing things. So instead of becoming an immune soldier, I became a communicator, which is not, you know, it's pretty reasonable for what I do here. But yeah, that's really cool. I will definitely cover that at some point in the future. As I said, like when I started this channel, I was like, am I gonna run out of biology? I'm never gonna run out of biology. Every paper I read generates like 8 different ideas. Okay. Thus, a latent developmental potential of cancer cells is occasionally realized, allowing the cells to non-genetically enter a mature state. Provided a biological context that opens access to trajectories of physiological development. The old notion of cancer as a differentiation arrest is consistent with the potential for normalization of the malignant phenotype when the appropriate signals relieve the arrest. Conversely, stressors, notably cytotoxic treatments itself, often cause de-differentiation in the cells that survive treatment. As noted in point 10 in table 1, cell stress can, independent of mutations, induce a stem-like nature that confers xenobiotic resistance. So much jargon. I don't even know what xenobiotic means, to be honest. Reorganize the tumor microenvironment and promote a pathological healing by exploiting developmental fields, all in the absence of Darwinian selection of genetic mutants. The logical inverse of Mintz's experiments is that work that produced the teratocarcinoma cells used in those very experiments, the cells were generated by implanting normal embryos under the testicular capsule. Later, it was observed that grafting normal, presumably mutation-free embryonic stem cells into the mouse can produce teratocarcinoma. These historical examples show that both differentiation, or normalization if it happens in cancer cells, and de-differentiation, which creates the cancer stem-like phenotype, are biological processes intimately linked to the existence of cancer. They represent drastic phenotypic conversion that lacks the characteristics of selective clonal expansion of mutated cells as the agent of pathological change. Instead, phenotypic transitions are controlled by a complex tissue context, which implies the involvement of an array of often poorly understood coordinated signals. So here is like, you know, the slap, the think biologist think moment here. Like, this is stuff that you already knew, biologists, right? You already knew. It's you know, you already had this in the back of your mind, in your heart of hearts, that cancer is more complex than genotype, that when you inject cancer cells into a normal environment, that they don't always become cancer in that new organism. They can become induced to be less cancerous, and then you can also do things that causes cells to become more cancerous, right? This idea that there are treatments that do not affect the genes at all that can force myeloid cells to become even neutrophils, right? Or revert more mature cells into a younger, quote unquote, stem-like phenotype that can then become cancer. So we're not talking about genes anymore. The author is saying, listen, you are not stupid. You've heard of these models before, and these are models that are pretty well accepted. It's about time that you prioritize thinking about cancer in this lens versus the lens that connects one gene to one phenotype, right? Like, you already know this. So shut up with the one gene, one phenotype thing. Stop trying to kill cancer by looking at single genes, right? You already know what you should be doing. Which I think is interesting. Listening to it on a drive, it sounds interesting, and I think they're on the right track. I do still think the old model isn't so much that's wrong. The old model can still produce tumors, and we have those types of tumors, but it doesn't explain everything. I think what the authors here are saying is that it's been fine. They're being less generous than me, and I'm more in line with what you're saying, Kotaro, that like we've learned a lot about cancer by studying it in a cellular genetic context, but like I'm leaning towards like we could be doing much more if we just stopped thinking of SMT and having the first thing that pops into our mind when we think cancer is tissue instead of genes. I think is a lot more appropriate. It's like these single mutational hits might produce a cell that disrupts the tissue environment, and it's those disruptions to the tissue, that environment, that gives rise to the cancers. I think what the arguments that the authors are making here is that that's not how the typical cancer happens. That is actually the exception and not the rule. Like, in theory, it could happen, but what the authors are arguing is that in a real organismal context, that is often not what happens when we measure real tumors in real people, as opposed to models that we have in extremely inbred mice with specific genetic mutations. Okay. Yes. Okay. is this an appropriate time to go to the schematic? Let's see, because we've been talking about this idea of differentiation and de-differentiation, so it might be time to look at their updated model. Okay. So this, they might go into this a bit more, but just to give you a visual, oh gosh, this one's from Marlin, Waddington landscape. Okay. So when we're talking about cell fate, this is always the little figure that pops up, this drawing here. It's an excellent drawing, and it's one that like you can obsess over because it's such a long-standing picture of how cell fates work applied to cancer, right? So if we were to step back, this is how, right, we used to think about cancer: one mutation in a cell, it clonally expands and becomes cancer. Okay, let's update that and view cancer more as a cell that falls into a cancer state. And so this is the Waddington landscape. This is not necessarily oh, you know, I can thanks to Bobo, who is who posted in Side Array's Discord, we actually have the ability to draw on my slides, which is so fun. Or draw on the OBS capture, which is great. So the Waddington landscape envisions stem cells as balls that are on top of a curved hill. And if you roll this ball down, there's some non-zero chance that this ball kind of falls in a certain pattern and gets stuck in a local population or a local area here. And this entrapment is a cell fate, right? So let's imagine that this is a stem cell. If you fall down this curved hill, you get passed across these groves and you might end up here, and this might mean you become a neutrophil. Now, this is ultimately a molecular sort of, this is mechanistically determined, right? So it's not really by chance, right? A certain cell is going to experience a lot of molecular events that push it into one of these local landscapes. But the idea here is that from just like a basic understanding, that ball could roll down here and become a neutrophil. Maybe that ball, that's not how to draw on the thing. Maybe that ball can roll down here and become a macrophage, or maybe that ball can roll down here, become a monocyte, right? So this is what is meant by, you know, if you want to envision the cell as trying to determine what it becomes, like a progenitor into terminally differentiated cell. And this is how it would normally work in the context of normal development, right? This model allows you to visualize this idea that the cell could become any number of things, but because of the way that these groves are formed, there's going to be a preferred state because it's going to roll down here more often than not. Kind of like a weighted, what is it? Pachinko? Like a weighted pachinko machine where like 60% of the time you're going to go down here, and then like 30% of the time you'll go back down here. Now, if you had perfect knowledge of everything about that cell, you would know with 100% certainty which fate it goes down, but you don't. And so this landscape allows us to think about differentiation as through the lens of probability and the factors that sculpt the cell into its final state. The actual surface exists in a much higher dimension, but that's a bit too abstract to depict visually. Exactly, this is just like a helpful visual. And so the way that this gets converted into an understanding of cancer is then this complex model here. So this is the idea that if you have cancer, you end up in like a weird place, like a non-0% of the time you end up in a weird landscape, which causes you to become cancer. But there are all sorts of things that can knock you out of cancer or into cancer. The Waddington landscape can be a very chaotic pachinko board. And so we should be thinking not as individual genes driving cancer formation, but rather different pachinko states or different pachinko boards or something that are a bit more complex to think about. It's not as easy to think about biology in this context, but it is kind of where the authors are heading towards. And so I think the authors are going to bring this up. They're about to bring this up. And I wanted to give you all the visual before we talk about it, because if you're a biologist, you probably have seen the Waddington landscape before. But for those of you who haven't, hopefully you have an idea now about like cell destiny, or what I mean by cell fate. 200D pachinko. Yeah, yeah, exactly. I really love the Waddington landscape. I think it's such a intuitive way to understand biology. But I want to pause before we continue because I think in order to get the most out of the rest of the paper, you're going to have a pretty okay understanding of what that Waddington landscape actually means. So I'm going to take a real quick poll here while I take a drink of water. So, do you kind of get, chat, what I mean when I say that, or rather, what the Waddington landscape is? So, we're about to talk about how stem cells become cancer cells, or how cancer cells become stem cells, and being pushed and pulled out of this chaotic pachinko board. It's a much nicer, more nuanced version of the narrative of cells simply becoming more or less stemmy. Exactly. Like, it's not wrong, right? Like, you can revert to more stem-like phenotypes, but it's not like a satisfying model. And this, like, Waddington landscape, this pachinko board, allows you to kind of visualize, like, if you get popped out of a certain state, you can settle in a different state, theoretically. We've also been long aware of many slower growing tumors being heterogeneous genetically, having different regions with different dominant mutation patterns. It is plausible that they might be a co-op, like they might be cooperating. Yeah. And Kotoro's pointing out that, like, again, it's nothing that we don't know already, which is why I think they're allowed to be this inflammatory. Like, it's hard to argue against all of this. What was this vote for again? If you kind of get the idea of what it means when a cell becomes more stem-like versus less stem-like, using the Waddington landscape, which I'll just pop on the screen one more time here. So if you want to think about how we get to a cancerous state, it's like this grand pachinko board where you're rolling a stem cell down a hilly road, and most of the time you're in this gray area where you become the cell fate you were always supposed to be. But then sometimes in the right environment, your pachinko ball gets popped off into one of these purple areas that you're not supposed to be in. You like clip through a wall or something, and you're in the back rooms, and that's bad, and that can become cancer. But you can also pop out of that place too. Yeah, biology is just one complicated pachinko board, is kind of what the Waddington landscape idea is meant to be. That was observed in the glioma paper. Exactly. Like, we're already familiar with this idea. We're ready to accept this idea, which I think is, fantastic here. Okay. So let us continue. So cell phenotype as collective actions of genes. Okay. Here we go. A new malignant cell phenotype in tumor progression is still often explained by a genetic mutation that follows the conventional one gene, one trait scheme. Again, something that we want to peel ourselves away from. But as widely noted, there is no simple correspondence, i.e. no one-to-one mapping between genotype and phenotype. Genes directly code for proteins, not phenotypes. I mean, this is something that we have to be very cognizant of, right? Just because you have a mutation in a gene doesn't necessarily translate into a protein. That's why anytime you do a genetic manipulation, not only should you do RNA-seq or some level of RNA measurement, not RNA-seq, qPCR to measure the RNA, you should also do a Western blot to make sure that protein is gone. Because, there's information that travels between these biological molecules. Gene loci interact with one another via regulatory proteins they encode to collectively produce a cell phenotype. So a cell as a whole, it's a collective action of genes. It's not just one or two genes, but you have to view them as networks instead. The fundamental principle behind the fact that cells within an isogenic, aka clonal cell population, can have distinct phenotypes as manifested in the existence of cell types of the animal body is that cells differ not in their genomes but in their genome-wide configuration of the activities of all gene loci. Such gene activity configurations produce the distinct gene expression patterns or transcriptomes that we can measure and interpret as the molecular basis of cell phenotypes. So many words to just say that one-to-one gene to phenotype is not a thing, which, I think nobody can argue against. I do wonder sometimes if cell differentiations are more of a combination of Pokemon moves rather than Pokemon evolution. I'm not mentally agile enough to figure out what you mean by that. One-track minded here. The multiplicity of cell phenotypes given one same genome obviously defies the tacit assumption of bijective mapping between genotype and phenotype. Such a relationship is required for neo-Darwinian logic to work. Natural selection enriches a population for a given genotype via a corresponding phenotype to which a fixed fitness value is assigned that determines the selection advantage. The phenotype diversification that produces the adult cell types from an unchanging genome follows the rules of development that are imposed by the regulatory interactions between gene loci. Oh, okay. So, yeah. So the fact that you can have one cell with the same DNA have multiple different expressions of cellness, yeah, that's fine. Again, no one's arguing against that. That's very obvious. Because, you know, if you've listened to anything that I've done with terms of development, right, you know that every single cell besides your sex cell, besides your eggs and your sperm, and I guess your T cells and B cells have more or less the same genetic code, but you have all sorts of different tissues, right? So this relationship was never that clear to begin with, right? Your muscle knows to become a muscle because of input it gets from the surrounding cells, right? That's, again, an argument that you can't even have. I guess the question is, how are we leveraging that argument here? Talking about fitness is always so hard because, yeah, it's just like I'm not mentally agile enough to view that critically, so I just take that for face value. The phenotype diversification that produces the adult cell types from an unchanging genome follows the rules of development that are imposed by the regulatory interaction between genes. Okay, right, we already read that. The interactions between the genes are hardwired in the genomic sequence and collectively form a complex network, the gene regulatory network, that orchestrates the activity of genes to produce those gene expression patterns that underlie the biologically meaningful phenotypes. So genes work together. Nice and easy. Such a genetic network was first proposed by Jacob and Monod. That's another neuron activation name. Jacob and Monod, from graduate school. I don't remember what they did, but like I will never forget those names. I'm sure they were uber important in the beginning of molecular biology because it's a paper in the 60s. Damn, I wish I remembered what Jacob and Monod did. They founded the lac operon. Oh, thank you, Jim the Evo. Yes, the lac operon. Okay, so thank you so much, Jim. So the lac operon. Okay, so the reason why bacteria are so important in the understanding of all biology is that like the discovery of the lac operon informed us how genes are regulated as a whole. So bacteria and eukaryotes do it in slightly different ways when it comes down to the details. But Jacob and Monod, oh my god, those experiments were so cool. I should definitely cover it one day. I don't remember enough of them to do it confidently right now, but with very clever mutational work, I believe they managed to lock down the structure of your DNA, meaning, finding the operon, the promoter, the actual gene, and mapping them out, finding what order they belong in, and figuring out that there are parts of the DNA that don't code for protein, right? We know that a lot of the DNA doesn't code for protein, and actually discovering that there is non-coding DNA that have genetic value because they recruit polymerases to transform your DNA into mRNA into proteins. So, oh my god, thank you so much. That's why I can never forget that, because dude, the lac operon is like one of the most important things in all of molecular biology. So thank you so much for continuing to hang out and chat and filling in the blanks for me. So cool. The destroyer of undergrad. the lac operon, I think, is really, really elegant. And I think if you have a good enough teacher, it's very intuitive. But then when you get it should be the destroyer of graduate students, because going through the Jacob and Monod experiment is pretty tricky at first. But once you understand with a butter knife, basically, right? Because molecular biology in the 60s was not that sophisticated. So Jacob and Monod, with very prehistoric tools, managed to actually figure out how genetic how genes are regulated. Just absolutely bonkers. Such an incredible set of research. Yeah, Joe, that's why I had such a strong emotional reaction to that name. Monod also spent his evenings during the Second World War fighting Nazis as the leader of the French resistance. He was kind of awesome. That's incredible. That's awesome. What's the stream about? We are talking about a shift in the current understanding of cancers. So for a long time, the popular understanding was that you have a gene that is mutated in a cell, and that gives rise to a bunch of cancer cells because cancer cells have a competitive advantage over their healthy neighbors. And with enough time, those cancer cells can become tumors. This paper is saying that we should stop, we should bash it out of our heads that tumors come from genes. Tumors are actually a result of bad communication between cells in their environment. And we are working through how convincing this argument is here. Thank you. Oh my God. Yeah, oh my God, I should totally cover. The problem is that old molecular biology papers are often not copyright free, which is super annoying. But if we can get our hands on the lac operon paper and share that and go through it, I would love to relive that part of graduate school, because again, they took a butter knife and almost invented 80% of modern molecular biology, which is just crazy. Okay, very cool, very cool. The central idea, as can be formulated mathematically, is that in coordinating the activities of genes, the gene regulatory network tends to interlock them in distinct self-stabilizing gene expression configurations called attractor states. A key postulate formulated in 1969 by Stuart Kaufman is that attractors represent the gene expression patterns that correspond to cell types. Being attractor states such as gene expression patterns are robust to perturbations. They reestablish spontaneously after disturbances and thus can be inherited across cell generations. The existence of multiple attractor states is a feature of a particular class of complex dynamical states to which the gene regulatory networks belong. Like this is like really tough to parse. So like, remember when we took a look at the peaks and valleys of this pachinko model? So those of you who are just jumping in, right? You can think of a stem cell as a ball that is being rolled down these hills. And so where it gets stuck is where that stem cell finds its stem fate. So if it gets stuck in a particular valley, it might be a neutrophil when it grows up. If it gets stuck in a particularly different valley, it might be a macrophage. And then we have attractor states that I believe they're making the argument that these are locally stable areas. Right here, you can see in the back here, I can use my pencil. You can see in the back here that there's this like flat area where you find the ability to just kind of hang out, because you can where cells tend to get pushed. And if you find yourself outside of the normal valleys, you might end up in a state where you are becoming cancer. Attractor states of the valleys in these metaphor images. Thank you for making that a bit more clear to me, because I wasn't sure if they were making the argument that they are particularly stable valleys, but I guess, yes, sorry. It's been a while since I thought about Waddington, but thank you, Marlon, for clarifying that for me, that these valleys, these local areas of stability, are where stem cells are rolled down and remain stable, and therefore it can become any number of different cell types. Okay. In the metaphor, the balls typically do not have momentum. Oh, that's fair. Ah, I see. Then maybe that's why they drew it this way. Because in the Waddington landscape, we can actually see, if we compare the original model, that Waddington landscape actually does have momentum because you're rolling it down a hill. So this is saying maybe you get popped off every once in a while by perturbations. At least I think that's what we're going for here, although I'm open to be shown wrong later. So, okay. Where were we here? Okay, the multiplicity of cell phenotypes given one genome defines a blah, 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 blah. Okay, great. Gene regulatory network. Okay, cool. Gene regulatory interactions of a given genome are implemented by transcription factors and also by regulatory RNAs and chromosomal configurations. Transcription factor proteins and regulatory, if you don't know what those are, exclamation point jargon, 1, 2, or 3, or 4. I don't know which one it is, but if you want to catch up, you can do that. Thus, their regulatory action is highly gene locus specific and is written in the genome. They collectively define the wiring diagram of the gene regulatory network. The gene regulatory network was wired by evolution via the tuning of transcription factor DNA motifs. Okay, so basically saying, and we could take a whole look at the model in just a bit, but let's continue reading their explanation here. Sequences, transregulation, and their cognate target elements dysregulation. So basically saying that transcription factors, the actual proteins that control the expression, the conversion of DNA into mRNA into protein, there is selective evolutionary pressure on those to form in certain ways that give rise to stable, healthy attractor states. This resulted in binding specificities between regulators. Such gene expression across all loci was forced into a set of genome-wide patterns, allowing the network to produce attractor stable patterns that encoded meaningful cell phenotypes. The locus-specific TFs and regulatory RNAs are supported by non-locus-specific apparatus epigenetic modifications. So here we're talking about your methylation, chemical modifications you can make to DNA that turn genes on or off, that sharpen signals and modulate temporal patterns, right? On, off, epigenetic modification. Thus, the gene regulatory network is a distributed information processing system of mutually regulating genes that can form webs of feedback loops, contrary to early views of genetic networks as hierarchical cascades of causation that rigidly link genotype to phenotype. So, okay, so how I've been talking about the central dogma the entire time. This is actually saying that the central dogma could use a little updating too, right? Hierarchical DNA, RNA to protein, that it's a lot messier than that, which I think is fair to say. That's not to say that the central dogma is wrong, it's just that it's incomplete. Yeah. This probably explains why certain tumors have made a lot more headway, so to speak, using the old model. Things like colorectal cancer, where the tissue is already undergoing clonal expansion. And a huge number of the genes that we are studying under the old model are related to differentiation processes, which is a pretty big hint. Yeah, so thank you for offering that information, because I'm not a cancer guy myself, is that, yeah, maybe the gene model was successful because it successfully predicted the action of certain cancers, but not all cancers are created the same, and maybe more of them fall into this more complicated model that isn't clonal expansion. That maybe for certain cancers it's true, but maybe it's messy like this is. So let's have a cartoon view of how this model operates. So this is what we are trying to replace our model of clonal expansion with, right? So here is, we'll point to this. So here, I'm going to get a stylus at some point, so I don't have to draw with my mouse. So we'll start here. This is a top-down view of a gene regulatory network, right? These are your different chromosomes. There are different genes. The genes have the mRNA that become proteins. These proteins form these transcription factors. It's a whole feedback loop. Transcription factors turn genes on and off, or sorry, not technically turning them on and off, but rather producing what they code for or not producing what they code for. So this is the chromosome. And then here we can see kind of zoomed in what this actually looks like. This is a very dynamic regulatory network. You have gene 1, gene 2, gene 3, etc. And these are all interacting with one another, right? You don't really have to see how these are linked up. That's not the point here. But the point is that they're all talking to each other. Biology is a huge, messy mess of probability. And that probability does things that you don't have the brain to really fully comprehend, right? Plus stochasticity, right? There's some element of randomness in this. Again, if you have perfect knowledge of every single molecule at every single time point, maybe you don't have stochasticity, but from a practical point of view, there's some randomness included in this process. Gotta run, enjoy the rest of the stream. Thank you so much for hanging out, Jim. Okay, so if you take a look at the expression of these genes, so actually mapping the DNA and their function, this is what you might see. And you might see that for regular cells that you have, let me scroll down. For regular cells, you have a certain profile of the contribution of your genes that allows you to land safely in one of these locally stable attractor states. But if something in your gene regulatory network is wacky, it might pop you off into an area you're not supposed to be in. Again, kind of clipping out of existence into a potentially dangerous spot where you get stuck and then you can become cancer. But again, as this arrow points out here, there's plasticity, right? You can move in between healthy and not healthy. And we've already known that because if you inject tumor cells into a healthy mouse, you don't always get cancer. And that's because you can be popped off, you can be pinballed off of these local valleys and get back into a better state or in a worse state, who knows? But this kind of allows you some mental flexibility. And when you think about cancer, this paper is arguing you should think about this. This should be your mental representation of how cancer operates. That cancer is not a disease driven by individual genes, but of a cell finding itself popped into an unstable not an unstable, that's a bad term for it. Cancer is a disease of finding a cell or a collection of cells popped in a certain state where they are going to crash out and become cancer, right? They're going to tumble into a place where they're going to be bad. And that's a subtle difference that I think is really important, because this understanding, this pinball pachinko understanding, it enables for more creativity when it comes to drug design. Beautiful, beautiful, I think. I think it's a very compelling argument to make here. Damn, your channel is pretty cool. Do you have a Discord? You can do !Discord for that link. Thank you, Marlon. Okay, so I love this, right? I really love this visual representation of what cancer is, because it has greater explanatory power, but it's also not that much more complicated, right? Like, one of the main advantages of this model, one of the main advantages of thinking one mutation into cancer, is that it's easy. It's pretty simple to understand. And what they want to replace it with is not on paper, right? I mean, if we're going to talk about designing experiments, this is complicated. But if we're talking about just our mental representation of what cancer is, I'm in complete favor of replacing that old model with this pinball model. It's actually quite interesting here. I just had some ads, but I think most people should be done with them by now. They're actually more frequent than I had imagined, even though I set them to the least frequent they possibly can be on YouTube. Yeah, I'll have to think about that in the future. I hope you all don't mind the ads, is the other thing, is that it does really enable me to determine whether or not I can do this full time in the future. Question, how would an experiment be designed? We're going to talk a lot about that. It's not really appropriate to do right now, but if you have to go, maybe I'll touch upon it. So, okay, so here's the advantage here, right? With the clonal model of cancer, the idea is if you just prevent this thing from happening, you could potentially stop the entire thing from becoming cancerous. It's been a very interesting strategy, but has had mixed success, we'll say. And so, I'm not a cancer researcher here, right? But maybe instead of delivering a drug to a cancer cell designed to sort of repair broken genes, maybe we design some drugs that instead help the healthy cells around the cancer ask that cancer cell to stop being cancer, right? Like, maybe we've been thinking about this entirely wrong. That maybe we don't have that much control over the individual cancer cell, and we might have more success targeting its environment. And that might be easier, paradoxically, even though the experiments themselves might be a lot harder. But it's also like not something that I necessarily want to not spoil. I mean, whatever. It's not like I'm going to be a cancer researcher in the future, right? I'm pretty much out of the game myself. And so it'll be up to you. Like, if I have a cancer solving idea, you're all free to take it and make $1 million bajillion off of it. I have no stake in the game anymore. And I was very broad with, you wouldn't even be able to get a cancer drug over what I was saying, because it was so broad. Okay. Yeah. So that's their model. This is what we are going to replace our current understanding with, and I am totally on board. This doesn't strike me as that surprising, that detrimental, that incorrect. It's just saying stuff that we've known for years already. The gene regulatory network is not a clockwork. Haha, I don't know clockwork if you're still in chat, but that's pretty funny. Stochasticity, so this is randomness, in cell type diversification has already been proposed by Kupiec in 1983. Thermal fluctuations of molecular concentrations, due to small number of molecules in cells adds a layer of non-determinism to the rules of the gene regulatory network. Okay, so basically thermodynamics screws with precision. That's fair, right? Just sometimes cells have more of a thing and sometimes it has less of a thing, and that's fair and good and true. allowing attractor states to be represented as probabilities of being occupied. The coexistence of deterministic logic and its probabilistic realization permits the definition of a potential like quantity, which is aptly represented by Waddington's epigenetic landscape. All right, we already talked about what that means, so we're going to move on. Cancer is the worst gotcha to have ever existed, right? And this is more of that, in which values correspond to attractor states and whose depth represents the relative stability of the cell phenotypes they encode, right? So deeper valley means you are harder to kick out. For simplicity, but sufficient for this discussion, we deal here only with fixed point attractors and omit more complex dynamical structures such as limit cycles, in which cells are trapped into a closed loop trajectory such as the cell division cycle. That's also a pretty interesting idea. I'm getting some quantum mechanics math vibes from this paragraph, not in a way that I think is irresponsible, right? Like, this isn't like woo crystal healing stuff. This is a more agnostic, less presumptuous model of how cells differentiate and how they operate. And again, I think it's not controversial to say that if you had perfect knowledge of every single molecule in a cell and all of their modifications in exact quantities, their exact temperatures, their exact isoforms, that you could predict with greater than 90% certainty what that cell eventually becomes if you had perfect knowledge. But we can't. It's too much. So instead, we have to deal with this quantum mechanics-esque supposition, which I think is fine for now, although with the advent of AI, maybe we'll be a little more confident in the coming future. Okay. Now here is a crucial corollary. On the epigenetic landscape of a given gene regulatory network, there exist, for mathematical and network evolution reasons, many more attractor states than are occupied by cells in the healthy adult organism. The phenotype of these unused attractors has been proposed to represent malignant cells. In this view, cancer is a possibility, imminent to animals, and hence is less suited to be seeing as being caused by something, but rather primarily the unleashing of something latent. Carcinogenesis would then be the accidental entry into these unused, latently present attractors, which thus have been referred to as cancer attractors. And these we must be avoided by normal cells in development and under physiological conditions. This is the reason why the old adage of the molecular biologist, again calling me out, that development is tightly regulated is more profound than one may think. So not actually in violation of molecular biology, but rather that it is cell fate that determines cancer, and not cancer determining cell fate, or rather not genetic mutation determining cell fate. I think, again, very consistent stuff here. Thank you all for hanging out. Thank you so much. It's really fun to get to talk to you all about such an interesting essay here. Okay, do we want to read this next part? Because I believe this is more of the same stuff. Let me just skim here. Sorry, Astral, if you're still in chat, you might have to just cut these words out because I might not read them, because I think... Okay, so this is talking about epigenetics here, so maybe we should mention this, because this is another layer by which we have something that isn't related to the A, G, C's, and T's of your actual genomic code. Epigenetics being modifications, chemical modifications that are stacked on top of your genetic code. So two people, twins, can have the exact same A, G, C's, and T's and be exactly the same in that regard, but if one twin has different chemical modifications on their DNA, they might be a little different than their twin. That's kind of the idea of epigenetics here. In the modern interpretation of the epigenetic landscape, cancer is thus built in, but normally not realized feature as epitomized by unused attractors. Since in physiological conditions, these attractors are unoccupied, they are not exposed as phenotypes, and thus they are not seen by natural selection. Most of these states would establish gene expression patterns incompatible with life in the modern animal tissue. Okay, so I think what this is saying is that we all kind of get that epigenetics is a thing, that chemical modification of A, G, C's, and T's is there and it matters, but we kind of like hand wave that away in terms of caring to measure them or incorporating them in our language at the very least. Those few unused attractors can sustain viable cells in the tissue and are not fine-tuned by selection. When occupied by cells that are thus by definition become neoplastic, cancer attractors would command phenotypes with primordial behaviors not shaped by animals' evolution to support cell societies that maintain the tissue homeostasis. Crap, that's complicated. Let's see. When occupied, that cells by definition become cancer. Cancer attractors command phenotypes with primordial behaviors not shaped by animal evolution. See, every time they mention evolution, my head starts to hurt. To support cell societies, in this perspective, cancer would represent atavism, reversion to its ancestral form, and cancer cells would revive selfish protozoan functionalities. This is a bit of woo language here, but whatever. However imperfect. Cancer represents reversion. Yeah, I mean that's what we've seen before. Cancer cells revive selfish protozoan attractors. Okay, so we're now trying to describe these cancer valleys that cells might be popped into. More generally speaking, unused attractors represent cell programs of phylogenetic past. This I brush up against in a... I don't, this is the first time in this paper where I'm like, I don't like that. So I think, and I could be wrong, I think what this paper is saying is that cancer cells are reverting to a genetic state that is more reminiscent of our single-celled past than our multicellular past. It's a very colorful description, but I think that goes a little far. Yeah. I think that goes a little far. I would phrase epigenetics more as modifications of chromatin rather than modifications of the basis, since covalent histone modification and more general chromatin organizations are very important. That's fair enough. My comprehension of epigenetics is somewhat basic, just put it that way. I know enough about it to get what it's about, but not enough specifics. So I accept that I might have simplified it a bit too much because of my own short-sightedness with regards to epigenetics. So thank you for that, Gil. Yeah, I don't love the comparison to protozoa because that is inviting hypotheses that I think are not necessarily supported even in the text in this text at least. So that's the first time I've bristled up against the authors here. The accidental yet highly organized nature of the vestigial phenotypes and their consistent reoccurrences naturally explained by the reoccupation of unused attractors that are an integral part of the same epigenetic landscape that is encoded by the genome and governs the unfolding of the normal extant phenotype. The cancer reversion idea should, in its argumentation logic, not be conflated with the notion of proliferation and motility being the default state of cells, discussed in detail later. Okay, don't really get that, but we can move on here. I think I've heard of this reversion of cells to a more unicellular state of nature before. It's kind of old. I've heard it a few years ago. I've actually never really come across this idea of a protozoan-like, again, because it invites too much imagination, in my opinion, unless it's supported by some actual convincing argumentation. But I guess if it helps people formulate hypotheses that are testable, who am I to judge? Okay, so we have here a more illustrative representation of the cancer landscape. Let's see if this gives us additional information, because I think this is a lot of the same stuff that we've seen before. So, okay, so what information do we get with this updated graph here? Let's see. So we now have it be more like Waddington landscape, where we have a ball rolling down a hill. So now we do have some momentum here. So developmental trajectories. So we have a stem cell here, and it's being rolled down these valleys, and they become, look at these guys, look at these little crayon. Oh, no, you can't see that on the screen, unfortunate. Let me shrink this for a bit. That's kind of annoying. Let me just scroll down. Yeah, so we have a ball, and it's being rolled down the developmental landscape, and we get them to be terminally normally differentiated cells. And you can get popped off of your normal route. Again, a gene regulatory state perturbation or genetic mutation, right? You can get popped get popped off of the hill and drop into an area not expected by your normal genetics and become cancer. And you might find it really hard then to pop back into the regulatory framework that you should be in. And then you have another side here where, what do they describe these as? Byproducts of complex gene regulatory dynamics and are unseen by evolution. So these are mostly maybe theoretical states. You know, the gene regulatory network. So these guys, they exist and are technically possible, but are not often seen. Yeah, because like when you have genes that regulate each other, you get an extremely complex map that is sure to produce cell types that are technically possible but don't practically show up because they are so rare. Yeah. Does the Discord have a channel you watch for comments during live, or is it mostly YouTube channel thing? I only look at the YouTube chat, actually. Yeah. Protozoan is too specific in a jump, I agree, but more generalization of the idea could be useful. Yeah, I brush up against the idea that we're like reverting into our ancient past. That kind of evokes an idea that I don't think we need to evoke that idea. It's not additionally useful in my opinion. Yeah. I mean, pretty cool model. I think hard to argue against. I like it. Clear the arrows. Let's get this back on screen. Alrighty. Cancer cells are also stuck in an ontogenic past. Because unused attractors do not partake in development of latter evolved mature tissue, they are more likely to produce developmentally immature dysfunctional traits that lack fine-tuned efficient homeostasis, mathematically represented by deep, smooth basins of attraction. For example, being unevolved, they are likely to lack sophisticated animal capacities to contribute to tissue morphogenesis, or to replicate genomes with absolute fidelity. So, these unused states, yeah, like, they might be technically possible, but they might be so useless that you don't practically see them because those cells just don't really do anything noteworthy, bad or good. They exist, but maybe not in any kind of way that matters. More importantly, they are not connected by developmental paths to the mature cell's attractor states, right? So that's more like clipping out of the back room or clipping into the back rooms. Such paths were carved by evolution via GRN wiring into the epigenetic landscape to ensure that normal immature cells respond to appropriate tissue signals during development and efficiently differentiate while being shielded from undue entry into the uninvolved cancer attractors. Once trapped in such attractors of imperfect homeostasis, cells are easily perturbed by hypoxic, inflamed, or otherwise stressed stroma, all of which are known to cause genome and epigenetic instability resulting in genetic alterations, further promoted by the absence of optimized DNA repair systems. Okay, so basically saying that, you know, cancer is not just genes, it's not just attractor states, but you can get bumped into bad states by your local tissue environment, which can be like a lifestyle thing. So it's good to not have a short-sighted view of just genes into cancer, but like the local environment of the tissue, exposure to certain chemicals, lifestyle even, or even genetic things that happen that can affect a tumor microenvironment. You know, these all play in a very complex way to give rise to cancer. And if we ignore those difficulties, you might find yourself wasting millions and millions of dollars. Stroma, that's a great word that I should have put in the jargon thing. So stroma are like the cellular support systems for tissue, so like connective tissue. You can kind of get away with thinking of stroma as the other cells that support cells in question. So when we talk about stroma, it's like the environment by which the cells that we're talking about are embedded in. Yeah. Okay. This model is growing more and more on me. Yeah, it's again, it's if you watch the 24 hour, Jesus, can you imagine next year 24 hours women's health charity stream? No. If you watch the 12-hour charity stream, the idea of cellular environment, tumor microenvironment is already one that biologists readily accept. but for some reason we still stubbornly hang on to this one gene equals cancer, or one gene arrow development of cancer. Okay, rethinking the genetic causality for cancer. Okay, so we kind of already, is this part going to be reductive? Hold on, let me give a quick skim, because I have been yapping for quite some time. My voice is starting to get worn here. Genetic alterations. So I think, despite the absence, so I am just giving this a skim. Okay, all right. We'll talk about it. Okay. So this section is trying to reconcile people who are still holding on to the SMT, the somatic mutation theory, the idea that in one cell you get a genetic mutation that clonally expands into cancer, is trying to be like, okay, actually, like, I might have called you an idiot at the beginning of the paper, but let's meet down the middle and see if you can accept what I'm putting down here. So we will read this because this is an important part of the upcoming argumentation. The concept of accidental entry into cells into a latently existing but undesired attractor and then being trapped in it supports the notion of cancer as a process of unspecified unleashing of potential rather than of specific de novo causation, right? So all of your cells are inborn with the capability, these landscapes that exist outside of the bounding box of normal cells. Like, you can clip into cancer, essentially. I really like that analogy, actually. Like, you can clip into being a cancer cell, for those of you who have who are familiar with the idea of speed runs, right? Like, clipping through the wall, you can clip through your regular development and end up a cancer cell, as opposed to cancer being caused. It's more like behind the wall, you can clip behind the wall and find yourself in a state that is technically permissible. Yeah, it's an interesting way to think about it. Cancer is more like backrooms. Exactly. Cancer attractors explain the unfathomable diversity of molecular disruptions and perturbations that all can accidentally trigger the entry into them and produce, as if orchestrated by an invisible hand. Callback, I think, to a lot of different academic fields. A specific gene activation configuration that governs the cancerous phenotype. These disruptions can result from an immense diversity of random genetic alterations that affect proteins of a variety of functional classes, or form a vast array of defects in regulatory interactions. From the rewiring of gene-gene interactions by genomic translocations, including creating novel fusion proteins or swapping promote. Okay, so this is talking about how much do I explain this? These disruptions can result from immense diversity of random genetic alterations that affect protein of a variety of, or from a vast array of defects in regulatory interactions, from rewiring of gene-gene interactions by genomic translocations, including creating novel fusion proteins. Yeah, so if you've hung out in my cancer stream before, you know that like a lot of your genes are classified as jumping genes. And the pieces of your DNA can just leave the chromosome and insert into other places in the DNA. And if it does that in a funny way, it can also contribute, well, we won't say cause cancer because this paper really doesn't want us to say cause cancer. So instead we will say contribute to the development of cancer. And the dysregulation of non-coding RNA, hey, shout out to our Nobel Prize stream, and RNA splicing to altered function of broadly acting epigenetic and chromatin modifying regulators. The immense diversity of mechanisms, all accidental, can converge to producing the cancerous phenotype with minimal help by Darwinian selection. Again, really hammering the cancer does not inherently grow better, and, you know, it is more like the whole tissue is freaking out and allowing this to happen, as opposed to one cell getting an advantage over others. Is perhaps the most prosaic yet profound manifestation of cancer attractors as latent structures in dark regions of the vast data space of gene activation patterns. Very, very cool. Cool. Gotta go. 32.39 words per minute. Been a very interesting stream. Thank you for keeping track, Astral. Very fun to see data come out. And I can't wait to see your combined graph of where I stack with the V-theorem psychologists. So if genetic alterations are involved, what is our criticism of the claim of a genetic cause of cancer in the current paradigm? Cancer cannot be explained by explicit genetic causation in the traditional sense of a gene for X that phenotype Y being defective. And we've known this, right? Mendelian stuff is very rare. There's a handful of stuff that works in this way, right? Like we know there's a Mendelian inheritance, for example, of what is it? I'm struggling to even name one. Sickle cell, right? That's one example. But most inherited diseases are not Mendelian. They're very, very complicated. I can post it somewhere later in the Discord. Which channel would which channel would be best? Maybe the Ethereum channel. That'd be kind of fun. Or stream discussion. That's fine too. The landscape provides a formalism that places the role of genetic anomaly of tumorigenesis in a new light. Genetic mutations essentially alter the GRN wiring. In mathematical models that map the wiring diagram to the landscape topography, a localized gene regulatory change, a point mutation, or a larger scale rearrangement is a localized rewiring and will most often gently distort the topography of the landscape. There are only so many ways a landscape can gently change before it's shaped while still ensuring viability, and one typical way is by altering the barrier height and separate the attractors. Such a distortion in turn affects from cell trajectories. For instance, it could facilitate the accidental entry into a cancer attractor from a given place in the normal trajectory, driven by molecular noise or non-mutagenic and environmental stress. It is in this convoluted sense that one could admit that gene mutations can cause cancer. So here the authors are like, fine. Technically, if you still want to say that genes cause cancer, you technically can, but I think that you are stretching. That, like, fine, you can say that genes cause cancer, that I haven't disproven your paradigm, but look at how much better our paradigm is compared to yours, where you don't have to do backflips to say that genes cause cancer. So what that paragraph is saying is if we take a look back at the clipping of the backrooms, right? So again, and we've predicted this since the beginning of the paper, this paper is not saying that gene regulatory networks are wrong. It was never about it being wrong. It's just about it being not useful. Although their inflammatory language gets you to pay attention because they're shaking you by the shoulders and being like, wake up, stop it. Don't even try to update your current understanding, right? Don't get away from the idea that genes equals cancer because it is holding you back. Because in order to make that model work, you have to jump through a lot of hoops. Instead, take a look at this landscape. And this is also pretty elegant, right? So when you have a gene mutation, such as like the BRCA mutation, BRCA for breast cancer, that's not BRCA causes cancer. What it's doing is that when you have a mutation in BRCA, maybe you change the height of this slope here that causes the pinball gotcha to punt a cell into the cancer backrooms. So you could say BRCA causes cancer, but in reality, it's a messy, hilly valley landscape, and it is one of the things that can shape the environment, but it doesn't cause it in a direct sense. The link is there. It's technically causative, but the explanatory power is very low. So rhetorically, you should excise the language of genes cause cancer from your brain entirely because it will fix you into a certain headspace. And the headspace that you probably want to be in instead is this landscape-based headspace. I think that's a very good argument to make. And I agree. I see something here. Their view of cancer is high level and abstract, while the mutation theory is more causal and mechanistic. Difficult to compare theories at different level of abstraction. I would say that their model of the Waddington landscape is not incompatible with mechanistic, as long as you are able to abstract mechanism into regulatory networks. So this is not my preferred scope, right? I prefer to think of biology in the language of cells because I just find it easier and more fun to do it that way. Not to say that it isn't powerful on its own, right? Like, we are here in our current level of understanding of cancer because we have a good understanding of what happens on a cell level. But I think you could draw mechanisms on a network level if you open yourselves into thinking about biology as big data. And so hopefully the paper will address how we take this paradigm and apply it, because that's ultimately what is going to convince a lot of people to abandon this idea of genes cause cancer. Okay, where were we? Oh god, I see Darwin's name again. Darwinian selection. Did we already read these? mature state normalization, genes being defective. Mendelian, yeah, we talked about this. As such, okay, so we finished that paragraph as well. One slight limitation I see with this specific landscape model is that it seems to consider the regulatory state of the cell as a variable, while the genes are seen as fixed parameters that determine probably that state. I think, while something like defective DNA repair genes, the genes themselves can change. Let's see. What do I think about that? Seems to consider the regulatory state of the cell as variable, while the genes are seen as fixed parameters that determine the landscape. Are they fixed, right? Because I think they kind of admit here that mutations in genes are underlying the gene regulatory network. Is just that they're not the only thing that can change the network. I might be either misunderstanding the paper or misunderstanding you. Please feel free to let me know if I've misrepresented your idea. Defective DNA repair genes, the genes themselves can change. Yeah, if you have DNA repair defective genes, then you can imagine the landscape as being more wibbly and wobbly across time. And I think the more wibbly wobbly that entire landscape is, the more likely you get bunted into the back rooms, essentially. Yeah. Okay, so despite the absence of developmental paths to normal mature states, the epigenetic landscape still grants cells and cancer attractors an intrinsic potential for normalization, as manifested in the rare reversion of the cancer's phenotype under particular experimental conditions. The reversion potential is rarely realized because very distinct tissue constellations are required to overcome the energy barrier to exit the cancer attractor. What do I think about that? Rare reversion under particular conditions. Okay, so we don't really see in these models, experimental models, the reversion of cancer because it's hard. Okay, it's a little bit of a cop out, but is it a cop out? Hold on, am I reading this wrong? Reversion potential is rarely realized because very distinct tissue constellations are required to overcome the energy barrier. Okay, so this is saying this generally doesn't happen, but it does happen sometimes. Because in neuroblastoma, they can spontaneously normalize. Okay, I mean, that is kind of fair, but if we don't really see it experimentally, then there's some weakness there. But I don't view it as a weakness. I'm just putting my critical thinking cap on. The difficulty of normalization is consistent with the limited success of differentiation therapy, and with exceeding rarity of spontaneous remission of cancer despite theoretical reversibility of cell states. So this is kind of showing like showing your weakness out loud, right? So this is saying previously that, okay, this model of like gene into cancer sucks because we've tried to pursue this with drugs and it just hasn't worked all that well for many cancers. But adopting this idea of redifferentiation has also been quite limited, because the way that we're thinking about it still means that cancer treatment is hard. So that is at least honest, I think. Do I like that? I like it for being honest, but do I like that in my ability to buy the argument? I mean, I'm on board with the idea anyway. I just don't know. I'm trying to determine if this observed experimental weakness kills this idea for me in particular. All right, good night, Alexandros. Thank you for dropping in. I guess what I'm trying to say is that the genes could also be considered as variables in an even higher dimensional state space. I don't think the authors disagree here. Although you can correct me if I'm wrong, if they're actually wrong on that. More on equal footing. Ah, I see. So I think that's probably where you brush up against the authors, is the relative contribution of genes you might be in disagreement about. And I think that's a dimension where reasonable people can disagree. Um, okay. Yeah, so they're admitting that like even therapies that try to take advantage of normalizing cancer don't seem to be working very well. I mean that could just be because not a lot of people are trying it, but okay. So we have an acknowledgement of sort of some experimental shortcoming. I don't know if I would have even mentioned that, or it could be misunderstanding what they're trying to say here. It's tough to have all the synapses firing despite the coffee I've had. I think we basically agree. It's just a slight nitpick of how the model is presented. That's fair. That's fair. We can now more precisely articulate an indirect convoluted role of genetic mutations in cancer to reconcile with this SMT view. So, okay. I love how like begrudgingly these authors have to be like, okay, we'll give up a little bit of ground. They're so resistant to throwing SMT people a bone here. If cancer isn't genetic, why do cancers have a very specific mutational signatures? So, for those of you who may have just dropped in, the authors of the paper want us to believe that cancer works like this, that we have stem cells that are rolling down a hill of probability to become normal, healthy cells, and through, chance, you can get booted out of the normal landscape and into a cancer-like state. And so, cancer is not incorrect to think about cancer as being genetic. It's more like it is becoming increasingly clear that thinking about cancer as genetic is not yielding a lot of results here. And so the mutational signatures are what is reshaping this complicated hill into being more favorable into for the ball to be rolling down the cancer side, right? And so it's not like genes don't contribute, it's that there are other things that contribute, including the cell environment. So if the surrounding tissue is compromised in some kind of way, for whatever reason, it's hypoxic in certain areas, that can also reshape the landscape. And so we should not be thinking of cancer as being primarily driven by genetic mutation, but genetic mutation being one of a billion gazillion different things that can subtly sculpt these probability valleys. Why is it heavily related to age then? Age gives you a lot more time to reshape these valleys into valleys that can pop off stem cells into more cancerous states. Yeah, yeah. Good questions, good questions. Where were we here? Okay, yeah, this paragraph. Both the genetic paradigm of cancer and neo-Darwinism rest on the tacit assumption of one-to-one mapping between genotype and phenotype. Yeah, it's a bit of an assumption. They're really pushing, or this is a bit of a straw man, one-to-one genotype and phenotype. I don't think that people who subscribe to SMT believe that there's one-to-one, even if they're a working assumption, even if it's, again, that's why they call it tacit assumption. They're not saying that SMT people believe it actively, but they are designing experiments that assume that this is true. And I think that's kind of more fair. Are the authors comfortable enough with testing this hypothesis with CRISPR-induced CRES or these mutations? So the whole point that the authors are making is that you can do those experiments by messing with genes, but you are essentially playing in a very safe sandbox, right? So yeah, you can get cancer to happen in a dish, but that's kind of not the point, because when you analyze tumors in real people, they often do not resemble cancers that you can force in animals. So meep. I think you are likely the audience that this paper really wants to talk to, actually. So if you open the paper and go to table 1, you can see that the authors have collated observations where there are observations that are inconsistent with a primarily genetic model of cancer. Again, not to say that genes don't contribute to cancer, but they are one of many different factors, as we are now trying to reconcile in this part of the paper. In this view, one must, by logical necessity, explain any phenotypic change by a genetic mutation. But as computer simulations have suggested, most mutations in the core genetic regulatory element, which in principle alter the wiring of the GRN, do not qualitatively affect the topography of the epigenetic landscape. Oh man, I am getting winded. but as said above, cause a gentle distortion. Thus, the majority of mutations, even those that alter a gene activity, are readily buffered away. However, mild distortion due to rewiring can alter the relative stability of attractors and thus may affect transition rates between attractors, stem cell and differentiated states, and senescent states, including rates for epithelial mesenchymal transitions. This is a really important tissue type transition that happens during cancer. An entry into unused attractors. Altered transition rates in turn lead to imbalances in tissue homeostasis. One manifestation is the widely observed transdifferentiation event seen in malignancy. Okay, so I want to go back to Meep's idea of testing this model. And I do agree that right now, I think a major weakness that I am... Okay, before I get into that, chat, I want you to come up with... I'm trying to ask you to predict why I think right now this paper is lacking something. And Meep's comment actually evokes this kind of potential frustration so far, because I buy the model, but there's something that I'm looking for, and it's something that I've been looking for since the very beginning of this paper that hasn't come up in the writing yet. And I'm wondering, chat, if you can hypothesize and allow me a little bit of break time to catch my breath, because I get so wrapped up in talking that I'm feeling a little hypoxic myself and a little lightheaded from my nonstop yapping. So I'm gonna go ahead and take a short break. And so my explicit question here is, Meep's question here is touching upon a weakness that I find in the paper so far. This high-minded abstract idea is something that I agree with, but I do think that it needs something else for me to, I don't know, be in love with it, I'll say. So I'll go ahead and have a sip of water. Oh, I need oxygen to my brain. I mean, nucleus. Experimental evidence against model X is provided, but none for proposed model Y. And Marlon says, an example. Yeah, so it's great to point out flaws in the gene to cancer pipeline. And, you know, it's certainly stuff that I find pretty compelling. But this idea so far has not been pressure tested. And I'm going to be keeping my eye on whether or not we actually get any pressure testing of this idea too, because what's another famous all-encompassing theory that is good on paper mathematically but has yet to have experimental evidence attempt to falsify it? Wondering if anybody can pick up on what I'm hinting at here. Robert says string theory. Yeah, it's kind of feeling like string theory to me right now at the current stage of me reading this paper, right? So string theory is this idea that, you know, I'm gonna get this so wrong, but like things are just vibrating strings that vibrate across 10 plus dimensions. And, you know, from the talk that I hear physicists say, you know, in public communication, that this is a nice idea that works mathematically in paper, but it is unfalsifiable. So in that sense, I don't know whether or not this model is really a model at all. As a paradigm, it works fine in the sense that, like, we should stop focusing on genes and start thinking about other things that contribute to cancer states. But like, is it really testable? And I'm talking out loud. It might not have to be testable to be useful, believe it or not, because as long as it shakes us out of our deep love for simple explanations like genes into cancer, it might do its job relatively well, right? You know, if the whole goal is to get biologists to think biologists think, you know, if that's their goal, then maybe this is a paradigm shift that doesn't require falsification, because this is so, it's feeling very like, how could you argue against this? I can't come up with anything better, actually. Or maybe I'm just not clever enough, right? I could be my own limited creativity in my current state. How much more of this paper do we have? I am so tired. Oh god, there's so many more pages. I can do it. I did a 12-hour stream before. We can power through. I am loving every minute, but again, I'm getting a little lightheaded from all the yapping. Most of those pages are actually just references, so I think I have like 6 or 7 more pages to go. As to the much needed higher level organismal biology view of cancer as complex disease, a first step towards organicism is the interpretation of large scale genome alterations as a source of cell physiological and not genetic informational departure from the norm. Aneuploidy and polyploidy in cancer cells often results in the formation of polyploid giant cancer cells that are found in nearly half of all high-grade cancers. Oh, I didn't know that. I didn't know that there are that high-grade cancers have cells that have multiple copies of genomes. That's surprising. Again, not a cancer guy, so I'm learning a lot about that. That's nuts. Very cool. Polyploidy and associated expression of meiotic genes in malignant cells offer a link between cancer biology and organismal biology at a larger timescale and could represent a rather generic alternative attractor that is often assessed by tumorigenic dysregulation of the GRN. In fact, since tumors often reactivate diapause, have no idea what that is, and gametogenesis programs that mark the entry into new sexual reproduction cycle and epitomize the immortality of the germline, it has been hypothesized that cancer may recapitulate, albeit in an aberrant and abortive manner, to the life cycle of organisms. Okay, that's a little weird. That's a little weird. That's a bit strange. So, making the argument that, like, I'm very comfortable with thinking of stem cells as reverting to a more stem-like state, or popping out of the back rooms or into the back rooms of cell determination. But to say that cells become more like new organisms, like a fetus, is kind of wild. It could be I'm not a cancer guy. Maybe cancer people like have no issue with this, but for me, that's weird. That's bizarre. I just joined, so my view is limited, but have they tried this in live mice models over multiple generations yet, or is this a theoretical applications paper? The whole point, I think, that they're trying to get at is there is a hubris in assuming you can take genetic tools and target individual genes in a mouse, give them cancer, and then have that be applicable to how it actually happens. Because a mouse does not generate... a mouse that you genetically modify does not generate a tumor in the same way that a human living a life normally develops a tumor. And so we should stop our reliance on that as an experimental model. That is to say, we've been talking about this entire time. We don't have an experimental way to verify this yet. But I have a couple of ideas in my head that could be tested, we'll say, and I'd be really curious to see if there's any research like that out there. So, and I know for a lot of people that if they think, oh, this experiment is not verifiable or unfalsifiable like string theory is, they immediately tune out. What I'm saying is that, like, for me, this is a weakness right now, but that's why it's interesting and difficult, because how do we test this? I think this is ultimately more testable than string theory, though. So I implore you to stay till the end. I'm thinking of doing like a proper video on this idea in the upcoming future, just because, like, I don't know, it's quite cool. If you just joined, I will refer to this model again. Sorry for people who have been around for a while, but it's always worth seeing. Cancer, we should stop thinking of cancer as being driven by genes, but rather cancer cells being the result of stem cells popping out of these pachinko holes where they pop out of normal differentiation fates into quote unquote the back rooms, as I've been calling it, and developing into cancer cells. So instead of thinking of genes driving this whole thing, we should think of genes as being a way to subtly shape these hills and valleys, but genes are only one way that these hills and valleys can be manipulated. Chemical modification, chemical exposure, hypoxia, oxygen levels, inflammatory signaling, things that have nothing to do with the genome can also subtly craft these hills and valleys and pop cells into the cancer phenotype. Oh. I run a cancer informatics group. We sequence DNA and RNA from cancer patients. This paper almost feels like flat Earth to me. Yeah, so it could be that this paper is yelling at scientists like you. So we disagree in that regard, where I don't actually view this as flat Earth at all, in the sense that cells are much more than their genes. DNA is not your destiny. It is mRNA, it is protein, etc., etc., etc. Then show me evidence, provide metrics. I can provide metrics all day of dangerous somatic mutations. We can provide genetic metrics that indicate whether a patient is likely to relapse. Yeah, so if you go to this actual paper, there is a table in which there are observations that are inconsistent with the somatic mutation theory. And the epigenetic landscape model is one that tries to ideologically shift our attention and focus away from this particular idea. So there's this acknowledgement that it's not that there is no contribution of genetics to the development of cancer, but rather it is overvalued right now. And so where you might brush up against the current authors is your assessment of whether or not the particular weight of genetics is appropriately managed in the way that current scientists think about this kind of stuff. Yeah. Yeah. So I'm also very curious if so at the end of the paper, I will give the authors the potential for first blood here in suggesting an experiment to actually test what seems like string theory here. So I wouldn't say it's flat Earth. It's more like string theory because it has this air of unfalsifiability, but I actually have some ideas on my own, and we can actually brainstorm as a chat ways that we could test this. But hopefully, I'm like really hoping that they throw in their own ideas here too, because it shouldn't be up to us to make their argument for them. Kind of a pet peeve, but yeah. Okay, where were we here? I'm losing it. Barriers. I'm so tired. I think we are here. Finally, beyond its putative role in generating mutations and neoantigens, there are also organismal consequences of genome instability, the mutator phenotype, which itself is a more likely consequence of the abnormal phenotype produced by unevolved attractors than the needed source of cancer-causing mutations. Indeed, the idea of what is actually, you know, before I say this, meep, the idea that I have is actually in line with the work that you would do. Like, yeah, the authors are very like anti-deep sequencing as a model for this landscape model, but I actually think that, you know, that we can handshake meme on this. Like, MGT and landscape can handshake in the middle, and I think that we can work, that these two camps can work together to actually test this hypothesis, which I think is great. Okay. Mutator phenotype, which itself, blah, blah, blah. Okay. Indeed, the idea of what actually came first was articulated by Richmond. It may be more correct to say that cancers beget mutations than it is to say mutations beget cancers. Then we should also remember the physical. Okay, that to me is a little funky. So this is a chicken and egg argument where the determinism behind the t=0 initial state of cancer might affect a local tissue before mutations even happen, which is feels a little circular to me, but whatever. Then we should also remember the physical, not informational consequences of genome instability. The DNA repair response triggered by genomic instability produces unphysiological DNA configurations. Different ways that we can break the DNA that leak into the cytoplasm. Okay. And which in turn, mistaken for viral invasion, activates stress and inflammatory responses via NF kappa B and sting interferon, thus affecting the cell's microenvironment. This brings us to the tissue level process. Do I what do I think? Okay. Okay, so this is an example where the authors might be going too far for my personal understanding of how cancer works. So so this is implying that DNA repair mutation is not the thing that is the strongest determinant of cancer, the strongest, most direct determinant of cancer, but maybe DNA, you know, the mutations in DNA are giving rise to an abnormal inflammatory immune effect that is then driving the actual cancer behavior. So implicit in this model is that if you treat the inflammation, then you can kind of ignore the effect of the mutated DNA. So it's like it's more important what the cell state is than what the actual code is. So the code can create that state, but if you solve the state, then you can solve the cancer, is kind of the argument that I am filling in the blank for here. So they're using an ABC framework without considering that the AB route could end in insert any number of variables here rather than C. I'm not exactly familiar with that kind of wording, Remrem. As a disease ecologist, these theoretical studies without proof has always made my head spin. In my field, certainty is required, fieldwork and proof. This paper seems to be lacking both. That's why this is an essay, right? This is a way to be like, stop focusing on this other model that doesn't seem fruitful. Let's try to shift our thinking in this particular way. So I hear all of your, it seems dismissive to call them cries, but I'm lacking a better word for it right now. Your requests, that's almost, it's in between cries and requests for experimental evidence. And again, I'm hoping that they have it here. At the very least, what they should do, so the thing is that we already have evidence that genes are not the whole story, right? It's called the tumor microenvironment, that there are tumors that lack genetic mutation, and there are genetic mutation in cells that lack the phenotype of tumors. So we already know that the link between genetics and tumors is not very strong. It's there. The authors are not making an argument that it's not there, but if you fixate, the whole thing is if you fixate on that connection, you are wasting your time. That's what they are saying here. It's a nice thing, much obliged. Yes. That might soften sort of the attitude here, because it's a persuasive essay and not a paper. Yeah. Hopefully that addresses, who is it? Meep Meep's concern as well. Like, this is a persuasive essay. It is more akin to Origin of Species than it is to the book that came after Origin of Species. Oh, okay. I'm turning blue in the face. Okay, we can do this. We can do this. Okay, so I think, we're about to hit an ad actually, so that's actually a really good time for me to take a drink of water. Okay, so we're running into an ad for at least some population of the viewers, and oh gosh. So I think this is also a good time to marinate on the idea that, like, we are now reimagining the pipeline of DNA repair, gene being broken, leading to genetic mutation, leading to cancer. And now we are thinking of DNA repair causing a freaked out cell, causing bad inflammation, influencing, and it's the inflammation that informs the genome to become a cancer cell. This is saying that all of our cells have the potential to become cancerous given the right ingredients. And when you have DNA repair problems, that is causing the cell to have the right ingredients to become cancer. And I hope that difference in idea is making a lot of sense. The usual impulse is to fix as many variables as possible and reduce a complex and dynamic system to simplify the examination/experimental design. It makes sense to do that, but we often get stuck. Exactly, Kotaro. That's exactly the vibe that the paper is trying to get at. That, like, The genetic model of cancer, it's excellent. It's so good for designing experiments, writing grants, and progressing science. However, the attitude that the paper is trying to get across is that you're tiptoeing, right? You are advancing cancer biology by nanometers and not inches. And if you get out of the view that it's all the genes and ignoring the role of inflammation and tumor microenvironment, then you are missing the forest for the trees. And again, I have ideas on how you could test this. I really want to get to the point of the paper where they do the same thing. Oh, okay. Another drink of water. How are we doing, chat? Does the idea of, okay, so let me just write in chat so that I have a physical record. Model A is saying DNA repair broken leads to cell cycle problems into cancer. This is what the SMT is trying to convey, somatic mutation theory, right? This idea that you have a single cell who gets their DNA repair stuff broken, cell cycle stuff gets broken, and then you have cancer stemming from that. Idea B is that you have DNA repair broken, DNA repair broken, a whole bunch of messed up stuff happens, and that causes cancer. Or C, anything messed up happening can be cancerous. I think if I were to summarize the model, it's C. You screw up a cell enough in any way, genetic, chemical, whatever, you get cancer, if it's done in a certain way. I think that's not hard to accept. Okay. And, if you are a biologist, if this is rubbing against you, I would, again, stick around in the end. We'll brainstorm how to falsify this. So if you reject this outright because it lacks evidence, we can work together and come up with some ideas that you might want to test, just so that you can stake your current understanding of the paradigm on something tangible, which is always important when we're talking about paradigm shift. Okay, cancer is a tissue-based disease. The tissue organization field theory of carcinogenesis, so I'm guessing this is going to be talking about tumor microenvironment. The interpretation of genome sequence-based lineage reconstruction defaults to the view that the tumor originates from a single cell, the initiation or founder cancer cell. This is correct in a fundamental, almost trivial way because a group of cells always has a common ancestor. It just depends on how far back one traces the pedigree up to the zygote, right? Okay, so it's like okay, the authors are like so rude. Just being like, duh, like, of course, dude. If you go back to cell lineages, eventually you get a cell. It's called the zygote. So that's not useful. Again, I think that's a little too rude, but whatever. Unfortunately, I have to go. So glad to have people discussing ideas I don't agree with in a thoughtful way, as science should be. And it's fun, I think, to go back and forth with the chat on these ideas, because I'm not a deliverer of biological truth, right? I'm just some guy. I'm just some cell. But yeah, thank you for popping in. Okay. The idea of one initiating cell in which the truncal tumor initiating mutation of the clonal tree happened, making it a founder cell, ignores that its outgrowth has to depend on its neighboring cells, right? That's true. That's hard to argue with as well, because cells exist in a context, and cell-cell communications happens very frequently, and the decision to replicate is often regulated by its partners, right? So ignoring the environment comes at a cost for complexity here. I would argue that this is more about the thought process, the philosophy of science, and a reminder to put what we are learning about cancer in the context of the organism. Exactly. That's exactly what this paper is trying to get us to do. Conversely, not all cells that contain that same early mutation that would allow for later oncogenic, otherwise lethal mutations to accumulate will become cancerous. Hi, you just tuned in. Did I miss anything interesting? Yeah, I mean, we're having a really thoughtful conversation about whether or not we're having a thoughtful conversations of how much genes actually matter in a cancer environment. This paper is saying that genes are not that big of a deal. That yes, genes do contribute to cancer, but so does a whole bunch of other things, and we should stop thinking about genes as causing cancer, but rather genes as being one of a billion different other factors that subtly craft this idea of cancer formation. So you could argue that it's not incompatible with genetic theory, but I would argue that it is more useful to think about cancer development as the result of an abnormal cellular environment than it would be genes. Because while genes contribute, as we've seen, just living in our day-to-day lives, just because you have a mutation in a breast cancer gene doesn't mean you're 100% guaranteed to get breast cancer, you know, to the hope for anybody who has these mutations. And that's because, fairly enough, genetics almost can't fully explain whether or not you get a disease. And it's much more complicated than that. And I think that's fair. Yeah. Okay, so empirical evidence contradicting the view of cancer as a disease of the cell was already available over 60 years ago when Smithers published an essay entitled An Attack on Cytologism. That's a fun term. Smithers refuted the idea that cancer originates from an abnormal cell. His and others' observations suggested that cancer is not a disease that starts from a single tumor-initiating cell, but rather an anomaly of tissue organization, right? So again, coming back to this idea that if we lock ourselves into thinking about genetics, we are missing the forest for the trees. This argument only proposes that it is not as simple as postulating a cancer cell, but does not deny the existence of individuals with enhanced potential to drive tumor growth. A compelling illustration of the need for a tissue-based rather than a cell-centric view is the finding that while a bulk injection of hepatic carcinoma cells into the liver generates a liver tumor, injection of the same amounts of the same cell type into the spleen, which distributes transplanted cells individually throughout the liver, fails to generate a tumor. Ooh. Oh. That's cool. So here's where my like anatomy is like bad. So apparently the spleen and the liver talk to each other in the way where the spleen can contribute cells to the liver, which I didn't know. Yeah, I don't really know that happens. So if you inject cancerous cells into the liver, you get cancer. If you inject it into the spleen, that feeds the tumor, that feeds the liver, you don't necessarily get a tumor in that regard. So this is showing that environment matters, that the entry point of the tumor cells and the dispersal and particular context that the cancer cells find themselves in is suppressive, normalized by the liver by direct contact with normal hepatic cells, right? So there's something about the healthy cells around them that's capable of suppressing their growth into or differentiation into cancer cells. So this is not something that this is not new information. We talked about normalization very early into the stream. Yeah. So I mean that's kind of cool that the spleen contributes cells to the liver. I didn't know that. These observations led to the concept that there is no cancer cell because cells declared as such behave as normal cells. Yeah. So we should move away using language that focuses on cancer as a disease of cells, but rather thinking about them as tissue. I think that's fair. I'm curious to see how their framework functions when introducing nutrition, infection status, and immunological variability. Does this paper assume a normal cellular environment outside of cancer? Again, we're not even close to talking about testing this experimentally, although keep that in mind when we do, at the end of the paper, discuss how we could test this in a reasonable way. Instead, cancer can be broadly understood as development gone awry. That's also not a controversial point, at least from my understanding. That's this is actually how a lot of cancer people I know personally talk about cancer, that it's a defect in development instead of a defect of cells. Although when I grew, quote unquote, grew up, that was the dominant way that people used to talk about it. Yeah. Within this perspective, the tissue organization field theory is based on two principles that unite phylogenesis and ontogenesis. First, the default state of all cells is constitutive proliferation within variation and motility. Therefore, proliferation and motility do not require an explanation. Instead, what ought to be explained is why cells do not proliferate and move. Secondly, cancer is a tissue-based disease whereby the tissue organization constrains the default state of its cells are weakened. Consequently, cells that become free to express their default state, thus proliferating, generating variation, and moving. This explains tumor growth by the accrual of new cells as well as invasion and metastasis. Sure. Like, I don't think that's controversial. Cancer is a disease of tissue, and proliferation and motility do not require an explanation. What ought to be explained is why cells do not proliferate and move. Yeah, so this is another shift in perspective thing that I don't think is that crazy here. The other major facet of carcinogenesis at a tissue level phenomenon pertains to the role of non-cancerous cells in the tumor microenvironment, or the stroma, aka the supporting cells. The idea that the TME, consisting of extracellular matrix, ECM, mesenchymal cells, and resident and invading immune cells, play a role in carcinogenic was accepted by the cancer community only in the 1990s, despite pointed reference by the pathologist Rudolf Virchow more than 150 years ago. The all too obvious role of the tumor microenvironment in carcinogenesis, which involves angiogenesis, inflammation, immune suppression, and cancer-associated fibroblasts, soon led to an exploding research funding in this area. Yeah, so we already knew the answer in our heart. It's not a disease of cells, but a disease of tissue, of the microenvironments and the cells around the microenvironment. However, the stromal component, so the supporting cells around the tumor component, has mostly been seen as a necessary enabler and rarely as a driver of carcinogenesis. So, yeah, I mean, to me, even in the language in this paper, that's kind of an assumption, right? The supporting cells have been suppressing carcinogenesis, but maybe we should say that carcinogenesis might be because the surrounding supporting cells are not doing their job, bunting the cancer cells back into a normal developmental state. That's also interesting. So here we can see that, like, the stroma, which could be genetically normal and not have any genetic perturbation at all, like, if they're compromised, maybe there's too much inflammation in the stroma, and that's actually going to prevent them from stopping a cancer from growing. That's a non-genetic mechanism by which a cancer can expand, and one that is testable. One I think that is obviously testable. Yeah, there we go, a testable hypothesis, right? So here in the chat, for those of you who want an idea of a way to get at a non-genetic origin of cancer, get a cancer line that is more representative of how cancers actually develop, one that has mutations that improve its odds of becoming cancer, and put it in two different environments with a permissive stroma and a non-permissive stroma. And you can try a couple of things to see what aspects of the stroma, which are genetically identical in both cases, right? And see what the contribution of, say, for example, shutting off one of the flasks from oxygen and seeing whether or not you actually get a tumor, right? So that's pretty interesting. That's a test that you could actually do. If there are cancers that are not driven by individual tumor cells genetically, what you should see is that you have these cancer cells. My drawing is so bad though, but I hope you can see that already by outlining these particular points. We have real experimental models that we can test, that this string theory test is becoming less string theory and more something that we can actually apply, right? So that's interesting here. Inducing tumors from otherwise healthy and intact cells without the need of genetic modification. It's a little more complicated than that, because you could just dump a carcinogen on top of it and they'll eventually form cancer. That's because you've wrecked every cell and caused them to go into chaos. Not that you necessarily did an exact mutation that turns on a cancer program, right? And this is specifically talking about the fact that cancer cells live in a context, right? So they live in a stroma, and then the modification. What if I did this? I'll put this in chat. Okay, so here's my idea, right? Flask 1 can't cancer, quote unquote, tumor cells with one cancerous gene mutation plus healthy stroma. Expectation, flask 1 is not going to develop a tumor because the stroma are keeping it in check. Stroma are the supporting cells around the cancer. And then flask 2, pre-cancer tumor cells with cancerous gene mutation plus hypoxic stroma. Expectation, flask 2 is going to develop a tumor because the stroma encouraged the development of a tumor. In both of these cases, the initial cell population that is going to become a cancer is genetically identical, but in one cellular context, they are suppressed or prevented from shifting, and in the other, they might be kicked out of it. So very interesting cellular experiment that you can do to get away from this idea that genes cause cancer. And again, it plays a part, right? You still need a tumorigenic line, right? A random healthy cell would be very hard to do this with because the chance that it becomes a cancer cell is very, very low. Oh. Some of the 8 hallmarks of cancer are related to cells being insensitive to their environment not doing its job. Apoptosis and insensitivity to growth/stop signals. I think what this paper is trying to get us to think about is suppression, enabling, and encouraging. They should all be valid routes of investigation. I don't know enough about cancer biology specifically if there are known mechanisms by which stroma cells not only permit but encourage cancer, but I don't know enough about whether or not the environment can pull at cancer there. But very cool, right? And you know, that's why I'm very sympathetic to this idea. Even though they haven't really talked about how to experimentally verify this, they're leaving us with enough information for us to do that ourselves, which is nice. And also bringing up again those observations that are not completely consistent with a genetic model for cancer. Ooh, okay, so cancer is complicated is what this says. Stromal, blah, blah, blah. It provided the biological embodiment of the convenient notion of context that is often invoked to mitigate anomalies plaguing the SMT. Okay, so even pointing out that, like, you know, again, trying to really move away from SMT. Throughout the 20th century, carcinogenesis was considered a genetic disease of the cancer cell despite accumulating experimental evidence pointing out to the causative role of the stroma. For example, the injection of a chemical carcinogen administered intraperitoneally generates mammary cancers in susceptible rat strains has been attributed to mutations caused by carcinogens in the glandular epithelial cells. However, all cells of the organism are exposed to carcinogen. From a developmental biology perspective, the question is, what is the target of the carcinogen, the epithelium, the stroma, or both? Oh, that's a little complicated. Eesh. Okay, so what's that saying? Yeah, even I refer to it as context, but it seems like the authors don't even like me doing that, which is kind of interesting. Injection of a chemical carcinogen administered into the hoo-ha generates breast cancer in rats, and that people are attributing to because the cancer cells, the actual cells that are the cancer cells become cancerous. Yeah, and that ignores the fact that the entire organism is exposed to the carcinogen, unless it's trafficked to certain cell types. So I wouldn't really know the biology of the rat in that particular context. Okay, but leaving it as... But, okay, so I think what that statement is saying is that don't take it for granted that when you have genetic mutations, that the causative ones are necessarily the cells that become the cancer. I think that's fine. My expectation, the cancer cells may have a hard time to get anywhere if not entirely die out. Yeah, if they're being surrounded and regulated by their partners, they're unlikely to get out. I'm a bit out of my comfort zone doing biology because I'm a nuclear engineer. Very cool to have people from different disciplines around. If it makes you feel better, Blake, I'm a disease specialist and my knowledge on cancer is rather limited too. I'm not a cancer person either, right? I'm a neurobiologist, but we can all enjoy this paper together because it's not written for a casual audience, but it can be translated to a casual audience, I think. Yeah. Okay. Yeah, so old studies that have genetic manipulations that are organism wide, you should rethink those in the context of tumors and their environment and not directly as an effect of mutating those cells in particular. I think that's fair. Wait, are you not an immunobiologist? Yes, I am a neural immunologist, or rather I was trained in the school of neural immunology, meaning that I have some immune background, I have some neurobiology background, but I'm not a full like immunologist. I'm just a hobbyist. I think the organism, I think the immune system is so fascinating, and I really, because I had a foot in both worlds during graduate school, right? One foot in the world of the brain and one foot in the world of the immune system. And I loved both parts, but if I were to say I loved the immune part a little more, but because I never had formal training in immunology, I don't call myself an immunologist. Maybe the cells in that part of the rat have a higher reactive oxygen level baseline and thereby are more prone to the carcinogen. No? Maybe, but who's causing the ROS baseline, right? The tumor exists in a microenvironment, and that microenvironment exists in the tissue context. So maybe the carcinogen is wrecking the stroma. The stroma has a hypoxic environment, and that's causing all sorts of stress to happen in the tumor itself, or the would-be tumor cells. I'm trying to be very careful to adopt the language that the paper wants me to adapt. Your translation of the paper is excellent, Fi. Thank you so much. Thank you so much. Yeah, I love doing these. Research is cool, and not a lot of people go through the trouble of showing you how scientists communicate. You are immunologist in my heart. Thank you. I dare not adapt that moniker, though, because immunologists have to have memories. They have to have hippocampuses the size of skyscrapers. There's so many things to memorize there. Okay, so I think this is one of the last arguments that we're talking about here, and it's something that I think Marlon may have pointed out at the very beginning of stream, talking about cancer in the perspective of organogenesis. So taking a developmental biology view and thinking of cancer as a budding organ instead of a disease of cells. I don't think that's a bad idea. To understand the relationship between the stroma, the support scaffold of an organ, and the parenchyma, the functional part of the organ, in cancer, we shall review the process of organogenesis. The genesis of tissues and organs entails complex reciprocal interactions between components of the morphogenic field defined by a distinct 3D array of cells that gives rise to an autonomous tissue structure of organ. Okay, a lot of jargon, but it's basically saying the organ doesn't grow in a vacuum, right? The organ, again, all of your DNA is almost exactly the same in every single one of your cells, and so cells need to talk to each other to figure out, okay, I'm going to become a liver cell, I'm going to become a liver support cell, right? That's not new. That's how organs develop. They talk to each other and they figure out who's going to become a real liver thing, who's going to become capillary, who is going to become vein or artery. They all talk to each other. They don't inherently have the understanding of what their fate is. They all kind of cooperate to figure it out. And so we should think about the development of an organ the same way we think about the development of cancer. Not to say that cancer is an organ, but the framework is there, right? The mental framework that underlies the hypotheses that we want to test as biologists, to get out of our asses and to not think about cells anymore and to start thinking about organs and tissue. Yeah. Where were we? Numerous epithelium mesenchyme recombination experiments have shown that it is the mesenchyme that determines the type of skin adnexa. I don't know what that is. Again, when we talk about tissue, I'm not going to know what these jargon means. Wing mesenchyme, I know this is a type of tissue. Wing mesenchyme dictates the presence of chicken feathers, regardless of whether it is recombined with epidermis from foot or wing, and scales appear regardless of the origin of the epidermis if recombined with the mesenchyme of the foot. So this is saying that when you have these Frankensteining experiments, that as long as the cell is getting the right signal, it'll differentiate the way you want it to. That you can take it out of the context, and as long as it still gets the right ingredients, it'll differentiate into something that makes sense. Yeah, so there's something inherent about epidermis that borders the wing mesenchyme that allows the wing mesenchyme to become feathers, even if it's from the foot of the wing. Okay. As in organogenesis, the stroma is equally as important in tumor genesis, where mammary epithelial cells were exposed in vitro either to mutagenic carcinogen N-nitrosomethylurea, great, or to vehicle before being transplanted into the mammary stroma of rats surgically cleared of epithelium. Carcinogenesis only occurred when the stroma was exposed to in vivo to NMU, regardless of whether or not the epithelial cells were exposed to carcinogen. Ooh. For everybody who wants to see experimental evidence of genes of cancer tissue not mattering, we have a manipulation of the stroma that is causing cancer of otherwise healthy cells. You could argue that this is still cancer, right? But genes into cancer, but it's not gene into clonal expansion into tumor. This is definitely not that. This is a genetic modification of the surrounding tissue causing cancer in a genetically normal cell. Again, this is fascinating, right? And if this matches up with human data, like if you biopsy a human tumor cell and you can find cells inside tumors that are genetically normal, this lines up. And in fact, the authors have cited papers in which tumors themselves might be genetically normal. Maybe it's a disease of the whole organism or the surrounding of the organism that is the issue. Wow. And that is very, very, very cool. Right? So I'll say it one more time for chat. The cells that become the tumor might be genetically normal because when you implant cells that have been genetically modified, the cells that are going to become cancer maybe are cancerous because they are either not stopped or they are forced to become cancer cells by their environment. So I don't remember who it was in chat earlier who had said that the tumor microenvironment is potentially permissive. This is an example of the environment not being permissive, but activating. So a disease of the tissue, it is not allowing the cancer cells to become cancerous. It is forcing otherwise normal cells to become cancerous. Again, this is a very artificial system, but this is very, very compelling. Now the paper seems to be applying a bit of ecological theory. I like it. Yeah, it's making the argument that like being too molecularly, cellularly focused is maybe hurting us in the long run. And I think that's an excellent point here. And it's these are very compelling arguments. This must have been a lot of work to put together. Would my work be more likely to cause cancer? I mean, you have OSHA to tell you how much radiation you are allowed to be exposed to before you have an appreciable risk of cancer. Yes, my favorite YouTuber is live. Welcome, welcome. We are in the middle of talking about how we can rethink the way that genetics affects cancer, but we are about to head into an ad, so I will pause for those of you who are about to watch an ad to kind of get a temperature read. So chat, what do you think about what we've talked about so far? So this is the argument that showing experimental evidence, and of course we're not reading the paper, we're just taking the author's word for it, that cancer can occur in genetically normal cells if their environment is not normal. That healthy cells can be forced to become cancerous if their environment is angry enough, we'll say. Because the stroma is not becoming cancer. The stroma is just there to support, but the stroma after mutation is giving the normal cells something that isn't changing their DNA and is asking them to become cancer through a mechanism unknown. And I want to get a read right now for especially those of you who have been resistant to this idea that we should totally junk the genetic theory, because knowing this set of experiments I'm like, yeah, like, okay, obviously genetics matters, but we don't need genetics to explain cancer in, to completely explain cancer, right? Like, it's a part of the story, but I'm beginning to believe more and more that it is a relatively overvalued aspect of cancer. And I want to get a read on chat if you are as convinced as I am right now, because I'm here thinking, okay, surely the authors are just saying that, like, okay, genes work together and there are all sorts of funky things going on, but ultimately genes still play a big major role. And now I'm thinking, okay, sure, genes might be playing a bigger role, or might play a big role, shaping the gene regulatory network. Obviously, genes shape the gene regulatory network, but now I'm beginning to believe that, like, oh, maybe the reason why we have not solved the cancer yet is because, dude, we have not been thinking hard enough about how to treat the surrounding tissue of the cancer, how to treat circulation and that kind of stuff. Like, millions and millions of dollars going into fixing broken genes. Maybe we should be funding more projects that talk about the surrounding tissue. I'm buying this argument a lot. Yeah. Okay, so I'm going to assume that that's enough time to yap for us to come back from the ads, and let us continue here. Okay. Very cool experimental results here. Mutation in the HA-RAS1 gene, which was assumed to be the cause of cancerization, did not correlate with the initiation of neoplasia. So RAS is a very, very critical gene in the cell cycle. So it's an oncogenic gene because anything that messes with cell cycle is associated with cancer. And it appears that mutation of this gene does not correlate with the initiation of abnormal growth. That's what neoplasia is. Not only was it often found in both cleared mammary fat pads of vehicle-treated animals and intact mammary glands of untreated animals, but it was also absent in various carcinomas. Okay, that's more of what we're hearing about, like the relationship between gene and cancer is not that clear. Such tissue recombination experiments show... Oh, sorry, skip this sentence. Yep, such tissue recombination experiments show that the stroma is a crucial target of the carcinogen, while epithelium is not. And in this case, the mutation in the HA-RAS1 gene is neither sufficient nor necessary for neoplasmic development. Again, the linkage between one gene and one cancer is not entirely clear, not that powerful. A comparable experiment showed that radiation-induced changes in mammary gland stroma contributed to the neoplasmic... Oh, gosh, so a lot of words here. A comparable experiment showed that radiation-induced changes in mammary gland stroma contributed to the neoplasmic progression of non-irradiated, quasi-normal, TP53 mutated but non-tumorigenic mammary epithelial cells. Okay, so more of the same, I think. Or it's not a new argument from the mouse thing. Finally, non-tumorigenic human prostatic epithelial cells from benign prostate hyperplasia develops into tumors when recombined with prostate cancer stroma and not with regular stroma. Oh, okay, so that's actually interesting as well. So if you take like a benign tumor, a benign prostate tumor, and then you recombine it with the stroma from a real prostate cancer, you get prostate cancer. But if you take a benign prostate cancer and you pair it up with regular healthy cells, you don't get cancer. Yeah. Yeah, okay, so this is, yeah, it's basically all pointing to the same conclusion here, that the surrounding tissue of a cancer is a major driver of cancer, at least experimentally here. Recent stromal epithelial tissue recombinant culture systems and single-cell transcriptomics have begun to unravel the molecular signals that mediate such stromal tumorigenic effects. Very, very cool. It sounds really interesting, and I'm so curious for future experiments related to finding out the mechanism. We will do some brainstorming because I think this is also very cool. And now that I've actually read the paper, my biology brain is like buzzing. I can't wait to mine ideas from chat because and you should all be mining ideas yourselves, right? Don't just wait for me to give you the answers, but we're having a discussion here, so come up with your own ideas. And again, you don't have to have biology background. I'm not going to yell at you if you don't know all the words, but even with non-biology background, you can still hypothesize. That's the fun of science, right? It's the fun of science is taking a guess and trying to test it. So can a benign tumor turn malignant? Of course it can. So benign tumors, the paper is arguing, we have shown experimentally that you can turn a benign tumor bad by just giving it the bad cancer environment. So there's not necessarily an intrinsicity of the benign tumor that causes it to become cancer. Oh my gosh, Purkinje! Purkinje, chill. It's not time for you to eat food yet. Although I might have to take a break to feed Purkinje in just a second. But I'm going to try to get to the end of this section before we do so. Purkinje is rioting. Kitty cat. Kitty cat, kitty cat. I wonder if this will inform tumor excision surgeries. That's a really interesting point, Marlon. Like, if you excise a tumor, if you cut it out, you are almost by definition affecting the local inflammatory context because you're cutting that tumor out. And I'm sure that cutting the tumor out of the context of the tissue context that exists in is going to make the surrounding tissue quite angry inflammatorily. So maybe cutting out tissue without maybe applying an anti-inflammatory or targeted anti-inflammatories might be why some people relapse. That's a hypothesis. I don't actually know. I'm not a cancer guy, but that's something that we can now think about in terms of cancer dimensions. That maybe just lopping out a cancer is not enough. Maybe we have to control the surrounding tissue and make them a happy environment so that the tumor does not reoccur. An excellent question. Yeah, excellent idle thought, Marlon. That's awesome. Cat, kitty cat, I'm so close. Please just let me finish this section. I will feed you, I promise. Oh, please. Can you be in my lap and not touch my keyboard? Yeah, there we go. Come on. Come on. I wonder what happens if the stroma type doesn't match with the would-be cancer, like put a lung cancer tumor and a prostate cancer tumor stroma. Yeah, I think that's an excellent experiment that I would like to see done. I don't really have a driving hypothesis behind what that would test. It would just be cool to me, unless you can think of a pointed research question. Okay, I promise I will feed you. Oh, okay. Not only can carcinogenesis be initiated by injuring the stroma. Oh, there we go, Marlon. That's your point, by injuring the stroma. But I think this means mutationally, by giving them a carcinogen. But a normal stroma can also act as a gatekeeper of neoplastic development that suggests abnormal growth by constraining cells belonging to a neoplasm to behave as the normal cells of an organ to form normal tissue structures. So having a healthy tissue environment can actually convince your would-be cancer cells to just chill and be normal. The experimental normalization of mutated cells that were once part of a tumor has been reported for a variety of cancer types among them melanoma by the neural crest, hepatocarcinoma, or mammary cancer. So these are a collection of cancers you do not want. So this dimension of thinking about how these cancers form, if we've been missing this idea, maybe this is a fruitful path to go down. A compelling demonstration of tumor-restraining role of the stroma is the case of localized pancreatic cancer, where targeting of originally considered tumor promoting CAFs, I don't really know what that means, resulted in tumor progression and invasion. Compelling demonstration, localized pancreatic cancer, we're targeting. So it looks like certain drugs that we designed on the old model did not help, but rather hurt. And maybe that's because it was hurting the stroma. And yeah, maybe we're doing more harm than good in certain cases because of our flawed understanding. Okay. Intriguingly, the same chemically inert material can cause tumors in animals when implanted as rods, but not when injected as a powder of the same material and same mass. Oh, these poor animals. This suggests that chronic wounding and or mechanical stress of the tissue causes carcinogenesis through a breakdown of physical tissue architecture in the absence of genotoxicity, something that has long been observed in multiple cancer models, underscoring the importance of tissue fields. Same chemically inert material can cause tumors. I'm wondering what material they're referring to here, but this is maybe a stretch. Without reading this paper, I have no idea what this is referring to, but I'm guessing this is like you perturb the environment, you can get cancer. Yeah. Yeah, I don't really know what that's going on. I think it's an argument for tissue disruption, but I'm not entirely clear. In the light of the central role of the mesenchyme on determination of the parenchyma phenotype, it is unsurprising to see the surge of data showing a tumor-promoting effect of altered stroma, notably extracellular matrix composition, so the stuff that's in between cells, fibroblast phenotype, so supporting cell phenotype, and tumor-promoting chronic inflammation, right? So saying that, like, it's the environment, stupid. While these findings have triggered a massive quest for underlying chemical mediators, one should not forget that tissue organization plays not just a supporting but a determinant role. Okay, so whatever this thing about rods is making the argument of tissue organization. The organist's perspective is based on the interdependency of the organism and its organs. It recognizes a circular causal regimen by closure of constraints that makes parts interdependent, wherein these constraints are not only molecules but also biophysical force. Since these views encompasses tissues not simply as collectives of cells but as emergent anatomical and functional organized entities, it affords very little opportunity for distinct molecular targeting, limiting its appeal to reductionist research program. Yeesh. This is a call-out post. This line is a call-out post. Since such a view encompasses tissue not as simply as collective of cells, but as emergent anatomical as functional organized entities, it affords little opportunity for discrete molecular targeting, limiting its appeal to reductionist research programs in search of a molecular silver bullet. This is a direct call-out in every grant that is trying to find a silver bullet for cancer, saying that you will never find just one thing that makes everything better. Again, reductionist research programs, a very aggressive call to stop funding. This is like, I'm getting the vibe that this paper also wants to talk to research grant proposal facilities, research grant programs, to be like, stop giving money to people who have this short-sighted view that genes into cancer. Because you're never going to find the silver bullet, this one thing that destroys cancer at the foundational root. Because while that is a very attractive model by which cancer operates, if cancer is this complex disease of tissue microenvironment, the chance that you get a silver bullet by fixing these genes is not super likely. Yeah, so that is a direct call out post. That is a very aggro position there. And it's trying to do like a wake-up call to biology as a field. Okay. I am going to go feed Berkenji. In the meantime, you can enjoy my wonderful Be Right Back screen, which I haven't actually used since the other cancer stream that I did, funnily enough. So enjoy the Be Right Back stream while I feed Berkenji, feed the beast. Don't go anywhere. I will be right back. See you in just a little bit. All right, we're back. My microphone is now unmuted and we're ready to get back into it. So, let's see. Let me catch up on chat here. The writer seems to have umbrage versus gene-focused cancer researchers. Yeah, it's like this is like too. That to me is like pretty aggro, right? Like, I think there's still good work being done, and we'll actually see. So I forget who said it before, but someone who knows more about cancer than me in chat said that like the model of clonal expansion still works for stuff like colon cancer, where those are actually the result of expanding clones of tumor cells. So I think maybe the authors themselves are being a little myopic, a little short-sighted themselves by bunting that view of cancer, because that could still work for certain types of cancer, but the cancers that are resistant to treatment maybe should be lumped in this model instead. Again, all models are wrong, but some models are more useful than others in certain contexts. So yeah, they're coming down hard on people who want to do this silver bullet approach, which I don't really know how I feel about that, because I'm an outsider, so I don't know what beef will be uncovered from here. But again, I am I think it's a more fun model, I think, to think about. Fun in a kind of, it gives your mind more to think about in terms of thinking about cancer as a disease of organs, or an organ-like disease, as opposed to single cells that have replication defects. Purkinje as in Purkinje fibers? Yep, exactly. I forget who it was in chat that suggested that as a name, but I was like, that's an awesome name for my cat, especially because I have a neuroscience background. Is the BRB video going to change when B Theorem gets new members? Yeah, for sure. It's pretty easy to edit this video. Oh, okay. Looking at references of the authors and the name of the author, it's not their first paper on this. In the 2000s, they were promoting their tissue organization field theory. Oh, that's a hard name to pronounce. Have even wrote a few books about it, and one is even translated in French. They cite themselves a lot also. Yeah, I don't really know anything about the background of these researchers, but the idea of like transcriptional states or even cell states is not new to me, and I've heard it quite a few times before, but never in like such an aggressive way to move past the more conventional view of cancer here. Okay. Okay, new section here. Explaining anomalies in cancer biology using under the SMT paradigm. So, okay, so talking about how the new model can patch up different observations that don't quite fit into the single cell forms a tumor model. A common way of explaining the lack of fit between the somatic mutation theory and data has been to invoke involvement of biological processes not obviously related to oncogenic mutations, such as metabolism alterations, bacterial and viral carcinogenesis. To me, that's so scary. Immune surveillance, etc., that we have characterized as compromises. In such hybridization of explanations, old ideas are kept intact, such as the claim that cancer is a disease of the cell, and that a normal cell is rendered cancerous by genetic alterations. Justifications often include epimutations due to altered epigenetic marks, or references to old dualism between initiator and promoter mutations. Yeah, this is more like trying to tear down what came before. The concept of dividing the cause of cancer into the initiator, which is the mutagenic event, and promoter, a chronic chemical stress by typically non-genotoxic agents, has been proposed based on classical experiments of skin carcinogenesis, first performed a century ago. Mice subjected to chemicals known to be mutagenic will develop skin cancers only after ensuring repeated treatment with a non-genotoxic irritant such as croton oil or phorbol ester 12O tetra decanophlorbol 13 acetate TPA, whatever that is. Recent genomic sequence analysis reveal that the initial carcinogen indeed caused genetic alterations, which remained silent until application of TPA at a later point. After TPA treatment, development of papillomas, so these are cancerous things, and carcinomas was observed. The elegance of the concept in its modern reincarnation is that the promoter is now understood not only to be exert its effect on the mutated cells via non-genetic phenotype switching, but also on the tumor microenvironment via tumorigenic inflammation. So I don't know enough about cancer to really get a sense for how much inflammation matters in the tumor context. I think it's not arguable that inflammation doesn't play a role in cancer, right? This is something that is generally accepted, but I think we're working really hard to ignore cell cycle stuff here. And it's making me like this argument a lot less. Because we've kind of ignored the model of cell cycle defects almost entirely, because that is, in all of the cancer talks that I've attended, that's still a part of the story, but we're just kind of like ignoring cell cycle stuff. I mean, it's not really ignored, it's just I would like to see cell cycle stuff more addressed, other than like RAS not being sufficient to cause cancer. Because I feel like in place of the old model where it's like cell cycle stuff gets broken and that's why you have a tumor, we're now replacing the cell cycle stuff with inflammation. And to me, the cell cycle model is more deeply entrenched in my brain, and I have a harder time giving up than this new model of inflammation being the driver. I'm not against inflammation being the driver, but again, the way that I was trained, I'm finding it hard to emotionally / mentally accept that we're just ignoring mutations and cell cycle stuff as being important. Yeah, inflammation was one of the classic hallmarks of cancer for sure, but like primarily driving like more than cell cycle mutations, that I'm having a harder time believing. Is SMT really better in explaining colon cancer, as it doesn't account for the microbiome, which also influences tumor initiation of aggression? I think the authors are making the argument here that, not the previous commentator who said that there is clonal expansion. It's like, it's clunky to go by SMT. Sorry, so let me think carefully about this. Again, I'm not a cancer guy, so I can't confirm for you that colon cancer is clonal. But if colon cancer is clonal, then it still fits within SMT, because you could have an initial insult, right? Maybe carcinogens that are made by the microbiome force a mutation that can expand clonally. So it's not... So for SMT, for colon cancer, if that's how colon cancer works, again, I don't know, SMT still might be pretty legit there. The ones that come from a lineage that undergoes epithelial mesenchymal transition during its normal differentiation has a higher chance to inappropriately activate cell mobility and metastasize. Yeah, pretty common ground between how we understand metastasis, I think. Can you elaborate on the cell cycle stuff? Like, what exactly do you mean? The G1S, G2 stuff? Yeah. So what I mean by cell cycle stuff is that a common model of cancer is that the reason why you get proliferation of cancer is because the usual checkpoints within a cell that tell it to stop dividing are broken. So instead of dividing at a rate equal to replacement, you get a cell that goes, okay, I'm just going to keep on dividing forever and not really care about what's going on here. And so it was long thought that abnormal control of the cell cycle is what gave to neoplasia or abnormal new growth. So that's what I mean by cell cycle stuff, although I don't know how modern my understanding of cancer really is there. Alex, colon has those crypt structures that reproduce extremely fast as a normal part of function. It makes them extremely susceptible to somatic evolution processes, especially if DNA repair breaks. Gotcha, yeah, so that is explaining why SMT is maybe okay in colon cancer. Although again, at organism level, I'm not like an expert. So current SMT methods, it's not methods, it's the model. It's the way that we think of things. And the methods that come out of SMT are more like, it's not like sledgehammers. I don't think that that's accurate. I think it's bringing a knife to a cannon fight. Like if we need to blast cancer like from different angles, the SMT is leaning us into a direction where we are trying to surgically deal with cancer when maybe we should be trying to blast it. It's a bit of a crude metaphor, but that's what I'll go with for now. The authors of this paper did research on endocrine disruptors as a sign. Their model may be very good for hormone-dependent tumors, but I think they extrapolate a lot outside of that. I mean, that could be true. Again, I'm not very familiar with tumor literature. I don't find a lot of resistance to these ideas up until this point. And maybe, Guy, that's what you're trying to point out here, is that, like, I was pretty on board with everything that they were saying. That, like, okay, it's not that hard to buy that, like, one cancer cell equals tumor later down the line is maybe an overly reductive model. But I'm finding myself unable to accept that inflammation is more important than regulation of cell cycle. So that's where I'm. Yeah, I was on board until right now. So if their school of thought is hormone based, it kind of makes sense that they're leaning hard away from genetic codes and more into cell signaling. I think they extrapolate a lot of that. Yeah. I think that I am not convinced. My personal understanding of the relative contribution of genetics and signaling is maybe more on the genetic side than the authors would like for me to be. It's certainly not, I'm certainly not a subscriber to TME by any means. Like, I like their theory, but in terms of the nitty gritty, I have yet to be convinced. Again, I'm not a cancer guy, but I'm not seeing anything that disproves that cell cycle regulation with genetic background is like not a huge driver of cancer. Yeah, I don't quite buy the inflammation argument right now. I'm open to changing my mind in the future though. But right now, not really. Yeah. So SMT model is like bringing a gun to a nuke fight. Why be precise when just blast it? I think that, to put it more accurately, current attempts at cancer treatment are trying to repair one or a couple of broken genes that are viewed as driving cancer, whereas this paper is trying to make the argument that we need to combine a cocktail of different things that can teach the tumor to not be a tumor. And that's more than just fixing a couple of genes. That is way too complicated to even surmise right now at our current understanding of how cancer works, I think. Yeah. Okay, so yeah, that's the first time I'm like, I don't really buy that, but whatever. The initiator-promoter model is often used to explain the well-known fact that a large fraction of listed carcinogens is non-mutagenic, and they may apparently cause cancer by triggering outgrowth of occult mutated cancerous cells. However, this picture is still too simplistic, exemplifying the aforementioned hybridization of old ideals with new findings. At its core, it still maintains a primary causative role of genetic mutations in the single cancer-initiating cells. followed by a sequence of causal steps. This qualitative dichotomy between a mutagenic initiator that creates cancer cells and the non-genetic tissue-perturbing promoter that expands them may not be as clear-cut. Instead, the reverse experiment, first treatment with the promoter followed by initiator, equally produces tumors. The result refutes this classical model that requires that the mutagenic alleged initiator must act first. Instead, the reverse experiment suggests merely a synergism between initiator and promoter, albeit a complex one that must involve tissue memory of non-mutagenic perturbation. The promoter-initiator framework by itself is an interesting model, but further investigation should be conducted through the lens of non-genetic dynamics, tissue organization, and organismal biology. Okay, that is walking things back by quite a bit. Just saying that, like, the order in which genes lead to cancer, it's not exactly straightforward. Which comes first, chicken or egg? Sure. Okay, I'm really bothered by, like, the insistence that cell cycle is, like, not that important here. Because I would love to buy this, but I just see no answer to the cell cycle problem. Yeah, I mean, if we had a finding that said mutate, like, because they brought up RAS, right? But RAS is one of the cell cycle proteins. There are a whole bunch of cell cycle proteins that, you know, I'm still of the belief that they drive cancer in those specific cases where you have a very strong promotion of. I guess they would argue that if you have a mutation in RAS, this might not be sufficient. It might not automatically give you cancer without permission from the surrounding cells. Okay. I guess I can convince myself of that being true. Okay. Another aspect that has been invoked to alleviate the discrepancies between genome sequencing results and the somatic mutation theory is epigenetic reprogramming. Okay, so now talking about chemical modifications to the DNA that involve remodeling chromatin that can turn genes on or off. A solid formal explanation for enduring changes in the landscape of cell phenotype would have to consider the dynamics of these regulatory networks because epigenetic marks themselves are not explanatory, they are reversible, and the epigenetic modifiers that place these marks are not locus-specific and must be guided by the gene regulatory element network to coordinate gene loci activities to promote coherent expression patterns that can be locked in attractors. Okay, I don't think that adds anything new right now. Moreover, the attractor stability of a cell phenotype can be disrupted by rapid change in its microenvironment that affect a large number of regulatory genes to overcome attractor dynamics. This property is evident in the recombination studies with fetal mesenchyme, underscoring the strong leverage that tissue-level interactions and tissue architecture possess in perturbing the gene regulatory network at multiple points to trigger attractor destabilization and transitions. Again, I don't know that this is adding anything super new. It's just saying epigenetics is changing those peaks and valleys. I don't think that's striking here. Interestingly, the hybridization of concepts to accommodate anomalies is also present in the extended list of hallmarks of cancer. Indeed, hallmarks only serve as attachment points for tenuous molecular causes, dictated by Dennett's greedy reductionism, namely the recourse by scientists to underestimate the complexities, trying to skip whole layers or levels of theory in their rush to fasten everything securely and neatly to the foundation. This attitude became evident in the latest installment of Hallmarks of Cancer listing that, instead of biological thinking, uses the existence of mutated genetic pathways to motivate new hallmarks that by necessity are explained by them. So this is saying that people have the circular tendency to think because our methods are good, our results are good, because our results are good, our methods are good, and not having their eyes open to an explanation they might not like. Notwithstanding, two new hallmarks, unlocking phenotypic plasticity and non-mutational epigenetic programming, are non-genetic with ironic redundancy between them. These authors write in a very spicy way. Chronic inflammation activates NF-kappa B and survival signals. This pathway can override cell cycle checkpoints, leading to uncontrolled proliferation or survival of damaged cells, which is implicated in both cancer and perplexity. The authors should name more mechanisms like these. Yeah, that would have helped, because the thing is, everything affects the cell cycle, and inflammation affects everything. And the link wasn't exactly clear what was going on there. Yeah, so we're heading into the conclusions here. So, dealing with a complex world, philosophers of science talk about auxiliary hypotheses, which are readily erected using known facts. These abound because complex systems contain a web of causal interactions as epitomized by the living organism. They offer an uncountable set of auxiliary explanations that can rescue any existing paradigm, and not only renders the falsification of a hypothesis meaningless, but also its defense by excuse arguments. Put differently, the genetic paradigm of cancer is a vague theory, and as the physicist Richard Feynman, again, name dropping all these famous communicators, vague theories cannot be disproven, which is even more true for a complex process as carcinogenesis, kind of flipping this idea on its head, because the genetic theory is actually very simple, it's just not very powerful. Therefore, interpretations of empirical results guided by formal theory based on some set of first principles and undaunted by dogmas is ever more important in an era of easy data acquisition. Again, so saying that, just because it's easy to sequence cancer data doesn't necessarily mean that it's useful. Over the years, many proposals outside the paradigm of cancer as a genetic disease have been offered in view of the accumulating finding pointing to shortcomings of the SMT. Some scientists in the SMT camp, still a minority, now begin to embrace a pluralistic view. For instance, we must recall Mike Stratton's quote in the opening section of this essay. After his team failed to find mutational signatures specific to endemic squamous cell carcinoma of the esophagus, he concluded, "We have found evidence that chemicals might be able to work in different ways other than directly causing mutations to increase a person's chance of developing cancer. We will have to rethink our ideas about the way in which some cancers develop. It is a crucial lesson. We hope that these crucial lessons will not be ignored. Pursuing a rigorous explanation of carcinogenesis entails identification of fundamental biological principles placed within a consistent formal theoretical framework, instead of a more terminological acrobatics." Very aggro. Centered around observed altered molecular pathways. As Darwin pointed out to a friend in 1821, if one were to only observe, not theorize, one might as well go into a gravel pit and count the pebbles and describe their colors. How odd it is that everyone should not see that all observation must be for or against some view, if it is to be of any service. Theories do not need to be right. They are practical tools to conduct research. If Darwin's pebbles are today's molecular alterations in tumors, then we must not simply be satisfied with their characterization in a system of ingredients that cause tumors, as illustrated by the proposed hallmarks. We must rather regard the hallmarks merely as manifestations of fundamental principles of living organisms. An epistemic shift towards a biological theory of cancer may still be an uphill battle in the current climate of thought created by the ease of data collection and a culture of research that discourages disruptive science. Here we have made an argument for dropping the SMT and its epicycles. We present new and old but sidelined theoretical alternatives to SMT theory that embrace theory and organismal biology and can guide experiments and data interpretation. We expect that diminishing returns from the ceaselessly growing database of somatic mutations, the equivalent to Darwin's gravel pit, may soon reach a pivot point. In addition, we hope then that the adherents of the genetic paradigm of cancer will begin to expand their vista on cancer in a profound way. I think that this is very well written. That this is a very interesting and well thought out essay, although well thought out and well written in the sense of like it is pleasing to read. Like I find that it is very easy to like walk through the arguments when I have the background necessary to interpret it, and also a very jabby, very spicy, interesting way of writing, right? Comparing the mountains of genetic data that we can collect from tumor cells to Darwin's gravel pit is an analogy, and it's a powerful analogy to be like, sure, you can go back to the drawing board and sequence the crap out of every tumor in the whole entire world across every person, but if what you're doing is only sequencing the tumor, we are missing crucial information about its environment, and we might be missing an entire axis by which tumors form and transform. So what do you think, everybody? So we're finally at the end of this paper. Oh boy, it has been tough. It's a very long paper, a very long essay, but I want to get a read for how we are receiving this angry paper, because it is a pretty angry paper, and I want to hear what you have to say in terms of how convincing this argument is. And are you convinced? Or do you think that the authors here are undervaluing certain common understandings that we have about cancer here? A call for nuance and a more comprehensive, albeit complex model. Yeah. They're saying that the condition resulted from that is what we should study, rather than focusing on the terminal and wiring, ignoring the reactor itself. Oh, okay, I see. That's the Blake's comment. Honestly, I think the argument was lost on me over the author's ego. I mean, as unfortunately a side effect of sociology is that like it's ultimately scientists talking to scientists, and like I think the paper started in a very fun way, but it got a little grating at the end. For me personally, maybe that's because my favorite pet theories of cancer were being challenged, right? Like cell cycle driven cancer is so ingrained even in updated models of how we think about cancer that I think that the haughty attitude without sufficient addressing of people's preconceived understanding may have hurt this paper a little bit, at least in my eyes here. The stroma experiments the authors cited changed my mind about this. I thought the aggro was a bit much, but I'm not the target. Yeah. I think the target, in retrospect, is... I started the paper by saying this is a strategy they can get away with because a lot of the arguments are not arguable, right? Like, I think you would be laughed out of a room if you thought that the only thing contributing to the formation of a tumor was the cells of the tumor itself. But by the end, I'm like, okay, I'm a little weirded out by how hard they're coming against certain very reasonable ideas coming from SMT, specifically the cell cycle stuff. The idea that it is that the stroma has such a strong influence on cancer. I'm not arguing that the stroma and the surrounding environment doesn't have an effect, but kind of feels like we're ignoring a lot of cases where it does have an effect, right? Like minimizing the effect of driver mutations to subtle changes in a tumor microenvironment. I would have to see the data that go along with that, right? Because experimental models that this paper is kind of dunking on, right? Defined genetic mutations of mice and rats. Like, and I'm not saying that they're wrong. I'm just saying, like, currently with my current knowledge of cancer, I'm not convinced of how they are weighting genes versus not gene stuff. Again, and maybe if I learn more about cancer and if I read all of the references here, maybe I would be convinced otherwise. But I'm just saying, like, thinking out loud, my cellular reaction as a neutrophil, right? Just as a thinking entity, I don't agree right now. And notice how I'm keeping my language flexible because I'm acknowledging that I have not done all of the research myself, right? There's a very good chance that if I read all 200 citations here, that I'll walk away from this paper being like, yeah, these dudes got it completely right. But I didn't, because no man can read everything, right? 200 papers, that would, oh my gosh, it would take me like a year or more to do all of that reading to see the data for myself. But as just like a theoretical framework, right? Here's what I'll concede. I'll concede that not all cancers are driven by genes. I think that's a very easy point to give up. Handily, I might add, right? Those experiments where you can force cancer-ish cells to behave normally if you give them a normal environment, right? The analogy being like, you know, if you put kids from a troubled home into a supportive environment, that they can grow up to be, you know, normally adjusted adults, something like that, right? That kind of analogy. I don't know if that's an appropriate analogy. That could be highly offensive, and I apologize if it is, but it's the way that I'm kind of drawing the connection in my mind. So I concede that, right? Like I give that up. I no longer think that every tumor is driven by clonal expansion. But I'd be really curious to see what a more level-headed analysis of this idea would be. Like, how many real-world human tumors are easily explained by SMT versus having to conform to this stroma-induced tumorigenesis? And I just don't have the reading in my background to know how true that is. Because I was very willing to accept that, you know, one gene breaking one cell becomes tumor is not really that accurate. So that's my read on things, and I'll read kind of what Chad is saying here, because I've been looking into the far distance, kind of talking my thoughts out through here. I think the ideas are good, have good merit. The writing is spicy, but they need to be more careful about undervaluing the genetic causes of cancer. Yeah, I think it's okay to be a little spicy, to shake people out of their preconceived notions, but I think there were points where you could gain more ground with honey rather than vinegar, or whatever the saying is. I think their ideas are good, but the author's aggro was too much. Yeah, they're putting in too much effort into attacking geneticists as overvalued. They should be highlighting the part that are undervaluing instead, and people will become dismissive due to their focus. Yeah, I think the idea was noble, right? The idea that we should be shaken out of a genetic paradigm is a noble quest, but I think for me they come out too hard on the opposite side. I mean, because it's written in a very inflammatory way. There's going to get a lot of people angry, and I think that if you want a true paradigm shift, you gotta serve your spicy with a side of milk, right? Like you can't just insult people until they believe in what you can't just neg people into following you, right? Like, I think that there were moments where you could do a gene by environment kind of argument where it would have been easier to swallow instead of, because again, like, I reacted pretty negatively to the idea that these tumors are primarily driven by inflammation signaling from the stroma, because that it seems like the mechanistic link doesn't seem that obvious to me. Have you heard about hybrid experiments? I watched a video where they said the transfer of mitochondria but not on the nucleus of a cancer cell causes cancer in a healthy cell. I have not seen those experiments, so seem very interested. This seems very interesting. I'm watching the reference of the paper. Most of the stroma experiments are done either by the authors or from people from their labs or in the acknowledgement sections. Kind of fishy. I'm willing to give grace on that. It's up to journalists to keep the scientists honest, and people in the field to keep them honest. If they've been manipulating data, then of course a lot of this goes out of the window, right? But my positive lean towards their overall idea is the fact that, like, cell state is like not a new idea. It's more like battling over the particulars, right? So I think their argument is quite convincing as long as everything that they've cited is real. Which is what Guy is cautioning us into believing, right? Like, if what they're saying is true, then I think they have a pretty good argument. But of course, if anything fishy pops out of anything that they've cited, then of course we might be in Alzheimer's 2.0 here, where we shift and then we realize that the shift is bad. Yeah. A lot of labs cite their prior work, especially if they're trying to highlight something that most labs are avoiding or ignoring. Yeah, it's not on its own fishy, but it's worth noting for sure. I think there's a middle ground there where they are doing a lot of the work, and I haven't like looked through the references myself, but like, it's not a reason to. It's a thing to keep in the back of your mind, but I don't think it's something that you have to elevate to suspicion. I think that's fair. The paper was a fun read. I don't know if I'm on board with the author's bitter hate of genetics, but I would love to see more non-genetics research. Excited to see a response to the paper. Yeah, this dropped on March 18th, so it's been shared hundreds of times on social media already, so it's certainly getting a lot of buzz. Is that spiciness normal in biology? In physics, it's very frowned upon. I mean, I typically don't read essays. I typically only read papers, so I don't actually know if this spiciness is normal. My guess is that this is pretty abnormal. The truth is, most cancer biologists know that. Yeah, exactly. That's what I've been saying for the entire stream. Thank you for bringing it up again, Kotaro. Like, this argument isn't hard to accept because in our hearts we know that this is true, but studying the tissue level involves an extremely complex system with a lot of variables that cannot be fixed. It makes it extremely hard to study. Yeah, and that is where we will end on thinking about ways we can apply this paradigm in a useful way, because if a paradigm shift does not result in experiments, then what the heck is the point, right? The advantage of the somatic mutation theory is that it's so easy. It is so easy to design an experiment. I'm not going to say it's easy to do the experiment, but it's easy to design an experiment in order to test a certain gene's contribution to cancer, right? Like, you could very easily be like, oh, you found a new gene, break it, see if it causes cancer. It's simple in theory and easy to test. Not easy, again, straightforward to test, maybe not easy, but straightforward. The problem with such a complex theory that I've compared to string theory is that it's really, it's kind of tough imagining with our current tools how we can design an experiment that can actually test this complicated stochastic model of cancer, right? So this this guy up here, let's get that more in frame. Let's get that more in frame. So I want to end stream by taking a look here, see if we can, as a chat, think of ways we can actually test this complicated model. So for those of you who have dropped in a little later, this paper is making the argument that cancer isn't caused by genes per se, but by the gene, by the cellular environment, the cellular context, the cellular content being this way. So you have a stem cell here and it's rolling down a field, right? It's rolling down a field, and if it's healthy, it'll pop into a terminally differentiated state. Maybe it becomes a neuron, maybe it becomes a neutrophil, whatever is in store for that cell. And cancer happens because sometimes that rolling ball gets popped into a different landscape where it can no longer escape. It can only progress as a cancer cell because genes are so complicated. So this way of thinking de-emphasizes the individual contributions of genes and basically says, well, genes are freaking complicated, so of course they're going to do all sorts of messed up things to each other and create this impossible to understand genetic landscape wherein like any tugging of this thing is going to cause a heightened or a depressed chance that one of these rolling balls of stem cells pops into cancer land. And it's a very hard model to disprove because it is so theoretical, but I don't think it's impossible. I don't think it's impossible. Yeah. Might be a little lost, but it seems like they want people to look into what molecules from stroma encourage and inhibit cancer. Wouldn't that be similar to geneticist ways? So I think it's a matter of perspective. You're absolutely right that I think what this theory is demanding is that we ignore the actual cells that become cancer for just a little bit and think about what elements from the stroma are affecting the cancer cells. It's similar to a geneticist in the way where you can view them as the same, but the things that happen within the stroma don't necessarily have to be genetic, right? Like, there can be faulty inflammatory signaling because you were exposed to a carcinogen, right? Say you work in, this is such a stupid example, but for some reason it's the only thing that I can think of, right? Bananas are like slightly radioactive. I don't think banana workers have an increased rate of cancer, but let's pretend like they do. So if you have elevated risk of cancer because you work with bananas all day and night forever and ever and ever, then you might be losing out on cancer research by only studying the tumor and ignoring the cells around it. So by sequencing the neighboring tissue that doesn't belong to the cancer, you can then get an idea of what the stroma is doing to the cell itself. And there's a chance that the stroma itself isn't mutated at all. There's a chance that the stroma itself is just faulty for some other reason, like, again, like maybe it's not that, again, I'm making the author's argument. I don't personally believe this, but I think what the authors are trying to say here is exposure to radiation, yeah, it can cause genetic stuff to happen, but it can also cause stressed cells. Because when you have faulty genetic replication events, that can cause inflammation, and that inflammation might be causing cancer to form. Could be upregulating cell cycle stuff and make it look like genetics is contributing, when in reality it's inflammation that's doing the thing. So cancer, in that case, that very specific hypothetical case, would go in this order. Like radiation damages cells. That damage to the cells causes inflammation. The inflammation causes the stroma to freak out, dump stuff into the cells that would eventually become tumors. And that might not have to do with any one particular change in the genetic code. So that's tough. uh, it's much easier to sell a grant studying something that has clear variables and means to design experiments with controls that drives the current focus. I don't think anyone dismissed tissue. Yeah, like that. I think is why they're so angry. Is that like there's an easy out, and that is the path of the geneticists, because those experiments are straightforward to do. But what I'm kind of annoyed by here is they are providing evidence, again, continued evidence that the environment is important, but they have not offered a true experiment to further. Maybe they're saving it because they don't want to give away their best ideas, but they haven't offered a path forward for researchers, young researchers, to get in the game themselves. Okay, say we adapt this rolling ball theory, and this rolling ball theory. But what do you do with it, right? Because for geneticists, it's pretty easy. You break a gene, see what it does, and then you try to fix it and see if that cures cancer. If you adapt this theory, all of a sudden it becomes a lot more complicated, and that's what I want to end on, is thinking about how you can test this particular thing. Let's see. I'm still catching up on chat, sorry. The conversations are too good. I think the paper is trying to say not all cancer starters are created equal. Yeah, that's I would agree with that idea. I wonder what this means for therapy, because fixing the surrounding inflammation of the tissue with antioxidants would also help the cancer in a lot of cases. Yeah, so maybe we have to take a look at how to effectively deliver antioxidants, and I say that in a very specific way, because we've known that antioxidants contribute to restricting the growth of cancer, but the mechanism, as this paper is arguing, might not be that clear. And so I caution anybody who makes the conclusion that, oh, we just eat blueberries, that's the thing, right? How to effectively deliver those antioxidants to the surrounding tumor microenvironment might be one path forward. So that still involves traditional scientific progress, put it that way. Oof. Let's see. How does this square with metastasis, where cancer spreads to other tissue, whereas the stroma is completely different? Yeah, I don't think they talk about metastasis all that much here, which you could argue is maybe another flaw in how to roll this out as a complete explanation of cancer. If I were to put on my hat, trying to come at it from their angle, they might be arguing that metastasis is one of these guys here, that eventually you end up in a place where you are doing things to escape the tumor. But you bring up a good point where I don't really see a whole lot in this paper talking about metastasis. Yeah, if we take this scope of this paper very literally, they are primarily talking about tumorigenesis and not like advanced stages of cancer. Maybe they don't really have a great way to explain that sans cell autonomous behavior, because maybe that might weaken their perspective, but I don't think they need it, because because they focus mostly on tumorigenesis, the formation of the cancer itself versus its spread. Because I don't want to make it seem like I'm defending them, but I am trying to be very conscious of the scope that they are trying very hard to fit in, because I think there is very convincing evidence that metastasis is cell autonomous, but I'm not a cancer guy, so I can't say that for sure, because there's stuff about like individual cancer cells like degrading their ECM and like crawling out of their local environment. So maybe this doesn't really fit that, and maybe that's why they ignored it, but I'm not entirely sure. You'd have to know precisely what pathway is being either activated or suppressed. Yeah, so testing this it's not convoluted, it's just not straightforward, put it that way. About the cell cycle stuff, is it not possible that stroma is dumping stuff when it's stressed out that messes with the cell cycle, and that sort of relates to the cancer cell cycle theory? That's what they're trying to say, that the stroma is dumping stuff into the cancer cells that are forcing them out of these local areas of stability and into the cancerous states, right? So That is what they are trying to say. So that's not in. I don't believe that that is in contention with the conclusions here. So there's like testing this model, but there's also testing predictions that this model makes in terms of developing treatments for this kind of thing. So as some of you have pointed out, a very good way to sort of test the predictions that this model makes is by designing treatments that work on the stroma versus the actual tumor itself. So like a lot of cancer drug design right now is CAR T cell therapies, right? Training T cells to recognize tumors and blow them up, essentially. And what the authors of this paper might be concerned about is that that is short-sighted, and that while that might have some success and is very exciting right now, we are ignoring the contribution of the surrounding cells, which, I mean, I can't really argue against, but yeah, we've had already some suggestions in chat, but how else might you adapt this paradigm? Like, how could you design a drug that works on this principle versus the broken gene into clones into cancer theory? I wonder why benign tumors can't metastasize while malignant tumors can. Yeah, this is a complicated question. I believe that has to do with multi-hit hypotheses that still stem in the family of somatic mutation theory, that you get multiple perturbations in the genetic code that enables a cell to escape the matrix. I don't really know how this landscape model really contends with that idea. Also you'd want to know if there is genetic damage to the receptors related to said pathway. It might be the case for a specific person's tumor that the receptor is jammed on or off. Yeah, exactly. So not only would you have to have, an absence of a genetic perturbation, but also some kind of mechanism or some kind of knowledge about the overall state. Yeah, it is complicated. Cancer is complicated, believe it or not. Right? Plenty of scientists have been fed into the anti-cancer machine because it is fundamentally such a complicated problem, right? Like, I'm not saying fighting bacterial infection is easier, but like, I think more progress has been made in terms of fighting bacterial infection because bacteria don't belong to us. We can design treatments that are bacteria specific, but finding how we can get a part of us that is growing abnormally to cut it out, is surprisingly difficult. So, any ideas of how you would design treatments based on this theory? I like the idea of thinking of antioxidants as a potential inroad, right? Because that begins the talk. I worry about how the grifter part of society will turn that into a profit, right? Buy my blueberry juice, and that's the thing that cures cancer. I wish, right? That blueberry juice still has to enter your stomach. And whether or not those antioxidants ever make it to the stroma of a cancer is questionable. Maybe a patch, an interesting patch strategy could work. I don't know. That's my hypothesis, at least. Pretty interesting stuff. I think this theory doesn't account for the possibility that the cancer itself can change the stroma over signaling. It's like the chicken and egg problem. Yeah, it could be bidirectional, right? Maybe, I don't remember them addressing that. Maybe they talked about interplay, but they seem to be very anti-cells as tumors, cells as driving tumors here. What about the stroma in healthy? The stroma is healthy, but a tumor still appears. Yeah, I mean, I don't know enough about individual cancers to know which of those are, what the prevalence of cancers with healthy stroma actually is. So that does change the interpretation by quite a bit, right? Because if you have cancers that have relatively healthy stroma, then this idea is not. I mean, it still holds up in the sense where, again, a cell is in a landscape, and if you have a healthy stroma, it prevents the landscape from changing by that much. But if you have crazy mutations, that could do it too. So it's permissive of alternate explanations, but it's kind of what makes this kind of a theory of everything, right? Which is kind of annoying. And it's very hard again to disprove, but again, that doesn't mean it's not useful. Um, I think the main utility here is trying to think of treatments that target the stroma versus the cell, the cells that are the tumorigenic cells, or not tumorigenic cells. That's the wrong word. The tumor. So I think what this paper might be trying to say is stop trying to design drugs against the tumor itself, but maybe try the stroma. And I think if it were worded in that way, I think it's a lot more reasonable. We don't have to have all this geneticist hate. More tools for this fight is always better, I think. I would try to identify differences in the environment between cancer's tumor, microenvironments, and normal environments. Yeah, I think you can easily fold in this model with your existing understanding of genes as driving cancer. Although I think the authors of this paper don't want you to do that. But I think I can do it a little bit. Like, again, not saying that they're wrong. There's no way I can know that they're wrong. I'm not a cancer guy, but in terms of just looking at the argument itself, I'm not super convinced that we should be like viewing genetics as being a very minor perturbation of this landscape. But here's where I do agree with the authors. I think entertaining the idea that the stroma is really important in drug design is a valid therapeutic strategy. It's just like, what do you do about that? How do you even set that up is the other issue, is that you would have to do these transplantation strategies of taking a cell line that is benign and doing a lot of these mismatch experiments and then designing treatments that rule out. I mean, that honestly would be really hard, right? Like, if I'm thinking about whether or not that would work, you would have to have a system where you would have to rule out genetics as being the driver. And they did that in a couple of these papers, right? That's the mix-ups, right? Healthy stroma, benign tumor equals benign cells. Unhealthy cells, benign tumor equals tumor. But the reason why that is not completely free of genes as contributing factors is that in those experiments, you still used a mutation, right? It's just in the stroma. It's just not in the tumor itself. Which, I mean, technically, again, it goes against this clonal explanation, but it doesn't dismiss the work of geneticists entirely, right? Because the stroma still are doing things. And I think they are really hand-waving away the genetics of the stroma as being drivers of carcinogenesis with, the stroma do inflammatory things. And I think combining this line that chat has thankfully allowed me to see, right? This is all your very mature, very thoughtful questions during chat that I was maybe not like super focused on because I was focused on painting their argument in the best light because that's my natural inclination. But the point that it broke for me is the insistence that it is inflammatory. And so this is why I love talking about science so much, is because we're talking it through. Like, you and me, chat, we're going back and forth in our understanding of cancer. And yeah, I think where I come down is that, yes, okay, I buy that the tumor itself doesn't necessarily have to have genetic perturbation, but genetic perturbation is still a major part of the story. And to insist that it is because of inflammation during the cell repair process, I don't see the evidence for that. Maybe I missed it, but I don't see evidence for that. So how would you test that? Well, you would take healthy stroma plus benign tumor, add inflammation. Do they do that? Did they already do that? Maybe they already did that. Let's see. I think that's the NF kappa B. It's NF kappa B part, isn't it? NF. Okay, so here's the part where they mention the inflammatory signaling. So what do they say here? We shall also remember the physical, not informational consequences of genomic instability, which in turn, mistaken for viral infection, activates stress and inflammatory pathways via... okay, so references 203 and 204. DNA damage responses meet innate and adaptive immunity. And then this ancient paper from 1962. Okay, so I'm not going to read the papers on stream because the stream has been long enough, but I still think they need to do that experiment, right? So what I'm looking for here is an experiment that is absent of gene mutations completely, or as complete as it can get. Benign tumor, healthy stroma, add NF-kappa B inflammatory signaler, and see if an inflamed but genetically normal stroma can do the same thing. And if I had that experiment, I think I would be pretty on board. But yeah, this is like the exact point where I was like, yeah. Yeah. Let's head back to that model. Yeah. What else we got from chat? That's why they check some tumors with things like being hormone sensitive, especially for things like breast, ovary. What is your background, Kotaro? I'm very curious because you appear to be much more knowledgeable than me in the field of cancer research. And I'd also like to ask you what you think of my coverage here, if I've done a responsible job here. Affects the treatment strategies. The same would be true for these inflammatory processes. The malignancy itself would contribute to screwing up the situation. Yeah, I agree with all of that. Isn't that kind of like what they're talking about with the non-mutagenic materials causing cancer as the rods, but not in the other form? Yeah, maybe, but I didn't like quite... I see. So the chemically inert... I think that's an argument from sufficiency, right? So disrupting the tissue structure is sufficient to cause a cancer, but it doesn't necessarily... Because that itself is an artificial situation, right? Like... Okay, perturbing the environment is sufficient, but are we... The thing that bothers me is not the sufficiency of the stroma, but rather the idea that the link is inflammatory and not a matter of genetic perturbation in the system somewhere. How do you decide what the threshold for healthy stroma is? Because logically, stroma may be different in each individual because different genotypes. Looks like it would be a very specific treatment. You've identified a hairy, complex... precision medicine-esque kind of problem here that yeah, when I'm referring to these, I'm referring to them as experiments done in pretty much identical mice. So if we could show like what is acceptable in a mouse to be healthy, it's a much more easy situation to manipulate, and then we can see what a non-cancer inducing healthy stroma in a mouse looks like and compare those biomarkers to that of humans and make a comparison, an imperfect comparison, but maybe when that moves the needle, but maybe not. As you are rightly pointing out, like, and again, a lot of resistance in chat is because this is such a hard thing to test, and the authors don't really, from my perspective at least, from my reading of it, are not really offering a lifeboat. We've been thrown off the ship of genes causing cancer. Now we're kind of treading water, trying to grasp at a piece of plywood to keep ourselves afloat. Yeah. Yeah. I think the argument that the authors want to make is, yeah, what Kotaro is saying is that if more people went for this idea versus looking at cancer driving genes, then we might... And just because I'm not clever enough to design an experiment doesn't mean that they're not clever enough. I'm out of the research game now. Maybe there's a common pathway by which the stroma could cause cancer. I would argue that the authors would say that it probably looks like a stupid, complicated mess like what you see on the board. It doesn't rule it out, but because genes are so complicated that it might not even be helpful to think of common pathways, but rather effectors, maybe. I don't know. You can imagine, okay, so here's like my experimental pitch, and I had this idea, I thought it was a brilliant idea, now I don't know. Now I'm not entirely clear as to whether or not I like this idea or not anymore. But here's my pitch, right? You have a very high throughput system where you have like a huge plate or many plates, like 364 plate wells or whatever they are, and each contain some cell system where you can graft some stroma with a cancer cell, and you perturb the system with all sorts of different factors, one at a time, and you try to create this model and give each peak and valley some weights. So even though what you see on screen is conceptual map, I'm wondering if with high throughput methods and with AI potentially, whether or not you could actually draw this for a real cell. Like if there is a future in which we can see like, okay, for this particular strain of mouse, we know that this myeloid progenitor cell, the cell that eventually gives rise to a neutrophil, we know what its peaks and valleys look like in this particular mouse. And we know that when we feed it this diet, that this peak increases, that peak decreases, at least in a very simple cellular context. And if we do that, then we might have, you know, and maybe the argument that the authors would make there is that, okay, you're doing the reductionist thing again and thinking too concretely in terms of molecules and stuff. But, you know, it's hard to escape from that paradigm, because how else are you going to do research here, right? That's, I'm not clever enough. I would want to make a map, yeah. But again, I think the authors might be like, stop trying to do the concrete thing and think more at the organismal level. But then I return by saying, well, what the hell do you want me to do? If you don't approve of looking at cancer in the view of things that we can actually test, then what the hell do we do about it, right? So I'm sympathetic to the paradigm, but it is quite challenging to think of good experiments that we could do that don't fall into some experimental pathway of simple cells and molecules, right? Because even in my molecular experiment, it would be in a dish. Given it's a dish with cancer cells and their stroma grafted in some kind of way, I wouldn't know how to do it, but that's an idea, right? That's an idea. The landscape model might get to the same place by affecting differentiation state. Epithelial mesenchymal transition happens to healthy cells as a normal part of development, so it's reasonable. Yeah, I mean, I think so at least. This paper does that. Try to map out the thing. That's cool. Is there something wrong with my example? I don't know. Let's take a look. I'm unaware of what this paper is, so let me quickly take a look at that. And if I have the APM in my brain, maybe I'll take a look. I don't know if you've already posted this in the Discord, but I don't. Oh, okay. So yeah, I just didn't read this paper. I think you posted this, but I haven't had time to take this paper and actually read it. Yeah, okay. So Marlon did point out that people have already done this. Great minds think alike. Calling myself great here. Very, very cool. Yeah. So yeah. Okay. So this is a paper doing that exact thing where they are trying to map out and define this landscape. So the question then becomes, can we translate that into a therapeutic, which is ultimately the goal of cancer research to begin with? You see, the thing is I don't really think in this modality, right? Like I like thinking about cells and molecules. I don't really think about hard high throughput problems here. Gotcha. I'm sorry, Marlon. Yeah, I don't always have the time to read what's posted in the Discord, especially when there are things out of my field that I'm not ready to think about yet, like cancer. I forced myself to be ready for this because of the spiciness of this on social media. If you get a truly batty signaling environment, having cell mobility active isn't out of the question, especially if it's some of the tumor cells already mutated into the stroma as being bad. Yeah. Hypothetically speaking, yeah. Whether or not that's a thing that is observed in the normal human tumor microenvironment, I'm actually unsure. The stroma signaling could stand in for a mutation insult and achieve the same impact. Yeah, again, from a theoretical perspective. But I'm wondering how to actually test that. Or to test that it is sufficient, and B, test that it is necessary to drive tumor formation, or at least contribute to it. Yeah, so does it happen in humans? And if we put it into an experimental system, is it sufficient to do the cancer thing? We can administer medicine at low concentrations over time, so your question, maybe. This venture is like dangerous territory of like nutraceuticals, which has a lot of grifting going on. Not to say that it is not the future, right? Uh, it's just like the idea of taking medication as a preventative for cancer is a sales model that works very well for when you are trying to sell supplements. And I am not in the business of doing that. And so that's why it's like shaky. Not to say that it won't work, but you would have to have, yeah, I mean, for something like that to tame a tumor, maybe that really would be the future, where you would have to have very good evidence that that is necessary, required, sufficient, etc., etc., etc. So I don't like that idea in principle, because again, it is, that modality is populated with people who just want to separate you from your hard-earned cash. All right. I am exhausted. How long have we been going for? Oh my gosh, nearly 5 hours. That's pretty insane. I will take a couple of minutes at the very end here to wrap up final thoughts, answer final questions. But to summarize my thoughts, is cancer genetic? I think after reading this paper, I think the answer is not always. And I don't think this is incompatible with previous models. And I think that the authors are a little too spicy at times. But I think ultimately it would be, in my estimation, given my current knowledge and not being an expert in cancer, I think it's probably a good thing that we have a more complex view. It just, I'm not clever or creative enough to think about ways in which we can translate this into medicine in a very easy and impactful way. And I think if you adhere to the gene theory of cancer, it doesn't necessarily make you a bad scientist. It's just, maybe that's a function. I'm my like roller coaster on this paper is just like ping pong back and forth. Like, again, without that life raft, you threw us off of the boat that we're comfortable on, but you didn't really offer something in exchange. And we have a couple of ideas that we've generated in chat, but I'm not like in love with any of them, especially because there are cancer drugs out there that work on the SMT model that are pretty good. Maybe not perfect, but pretty good. And this paper itself, I don't believe cites a treatment that is very good using this model. I think because it's really hard to get medicines to work in this way. So even if this advances our understanding of cancer, even if we do paradigm shift away from a genetic model, we're not guaranteed to make the corresponding advancement in science and development in medicine if it is fundamentally too hard to wrangle cells to do what we want to do. Because of, as any biologist can attest to, when you try to get living things to do something, they often fight back. So I wonder who wins in the end, right? Geneticists versus, I don't know, organists, maybe we could call them, who emerges victorious. So I'll say that, like, as an idea for how cancer develops, I think I side with the authors. I think it's pretty convincing, other than the whole like weirdly laser sharp focus on inflammation. I'm on board with this idea. But in terms of the evidence to make a therapy that works, a strategy to develop therapies, I'm going to lean towards the geneticists here, just because I don't see a super obvious way out of this hole, apart from the few ideas we've generated in chat. But again, I'll say I'm not convinced at this certain period of time. Like I'm like 80% won over, and then 20% is my reservation about cell cycle, and I think genes are still pretty important. But again, I'm open to changing my mind. Just right now, I'm not totally sold. Um, would it be possible to figure out what bad stroma is signaling to each other or what genes are activated and thus test those molecules on cancer-prone tissues and see what happens? Absolutely. So that is a very good strategy that you could potentially do. You can achieve this using a couple of high throughput methods. So what you can do is you can force stroma to be bad. You can confirm that it's forcing cancer to happen. Then you can separate the stroma and grind it to dust and do something called mass spectrometry, where you figure out all of the different proteins that bad stroma are making and compare them with good stroma. Then what you can do is you can take the difference. So what are proteins that bad stroma are making that good stroma are not, and figure out what those are, and seeing if you dump those onto if you dump those onto good stroma, do they become bad stroma? If you dump those proteins onto benign tumors, do they become tumorigenic? So that's an excellent approach there. That's assuming that the signals are a soluble factor. That is true. That is my bias, that whatever thing, well, is that assuming the signals are soluble? If you grind them all into dust, you know, that's taking care of cell-cell contacts as well, right? Because you still get membrane proteins. Well, I'm assuming that you do cell fractionation, right? So I'm assuming that you separate cytoplasmic proteins and membrane proteins, and you do mass spec on everything. Just see what proteins the cell makes. So I don't. Hold on. This is my lack of expertise. Is I don't know what the nature of the cell-cell contacts between stroma and cancer cells are. If they are directly connected, then maybe you can take the. Okay. I've resisted doing this, but I should look this up. Stroma cancer. What does this look like? What does this look like? Not stoma. We want stroma. Okay, so what do we have here? Sorry about the momentary pause in the content. I am trying to figure out if there is a way. Yeah, it looks like there is a direct. So yeah, it looks like they do touch each other, right? So I don't think solubility is an issue here. Something like differences in ECM organization wouldn't show up, for example. That's true. So ECM being the space between cells. Could you I'm not really familiar with how to study the ECM though. Yeah, I don't know if you could wash out the cells and just keep the ECM proteins. I'm not familiar enough with those methods. Yeah. I mean, that's where you would start, right? You would start with the hypothesis. Let's assume that the stroma are spitting something into the cancer environment. Then we can do that process of spinning it down and feeding it into the mass spec. You could also do something like FISH, which is fluorescence in situ hybridization, where you can label cells based on what mRNA they are making, and you could do it for a whole bunch of mRNAs that you might suspect are having a role, and see, the profile of stroma that are cancer-causing versus not cancer-causing, and that could give you hints as to what's happening. That might be a little better for ECM stuff because it can give you an idea of what genes are expressed that might be extruded into the ECM space that you would not catch from the experiment that I just talked about. So maybe that's better. I don't really know. to be honest, the synapses in my brain are shutting down. Mark different proteins, molecules, and see their fit configuration in the ECM. The problem is how to practically do that in a high throughput way, because I mean, maybe you could do it. I'm not knowledgeable enough how to do that in the ECM. In the cells, it's super simple. You just take the cells, you break them open, you separate membrane from the guts, and then you separate the protein from all the other garbage, and then you feed it into a fancy machine. With the ECM, I'm unaware of a method that lets you do it to just ECM. Although anybody in chat can feel free to correct me. Yeah, I'm a biologist. I'm still learning myself, you know. You're always learning as a person in science. Okay. Really interesting ideas. You can definitely tell that my energy level has slumped a lot since starting the paper. I'm so I'm so tired, like spiritually tired. My brain is still like super on fire because of the coffee I had, but like I'm winded talking 5 hours here. Okay. Master Yi. Oh, you are invoking something that might happen in my future that is maybe uh closer than I'd like to admit, is playing League on stream. Interesting life updates that I've teased before, but I'm not quite ready to talk about openly. But the nature of this channel will be changing this year due to life things. Maybe do an ELISA for the ECM proteins. The problem is how to do an ELISA high throughput ELISA. I mean, yeah, I could see that kind of approach working where you like dump a ton of antibody and just see what's around, but like I don't know if there exist antibody panels where you could do that, if that makes sense. Especially because antibodies are so expensive that like dumping every antibody known to man onto the ECM, I don't really know if that is likely to yield a result that you can interpret. I'm currently doing a master's thesis related to cancer treatment. Thank you for offering your expertise again. I'm a cancer hobbyist, but having your input has been very, very helpful, informative, and keeping me grounded is also very good. So thank you for your contribution, Longil. That's very nice. Lungil, I guess. You can correct me if I pronounced that wrong. Really nice stream discussion. So yeah, thank you for hanging out here. Finally caught up live after watching the whole stream on 1.5 speed for 40 minutes. Well done, Aiden. Very strong. I am about to call it quits, but if you have any thoughts having watched the entire thing, I'd be very happy to stay around and chat for a bit longer. Since I miss talking to all of y'all. And if I were a better yapper like Cider A and Myelin are, I would do a lot more yapping. But yeah, I think the kind of streamer I am, I have to have like a topic, otherwise I find myself at loss of what to say. 11 p.m. here. I'm also like very happy to have such an international audience. I think and that's why I chose St. Jude's as like the cancer organization of choice, because you know, no country owns science, and for those of you who are students in other countries, I applaud you all, and I hope you all do great things. And you know, join the Discord, especially if you are in biology. Like, you know, I don't expect you all to contribute or talk regularly, but like I would genuinely love to hear kind of where your careers take you, because you know, you're not the only like master student in chat, and I would just love to see. You don't have to like tell me where you study or whatever, but like if I don't know, it sounds desperate, but like if the content I've made helps you stay in a field of study, and you persist through the terrible pipeline that is academia or industry, and you go off to do some of the things that I sing about as a bard of biology, that would be awesome, right? That'd be incredible. Like and I've only been doing this for a short amount of time, so this is more of a long-term thing for me. Like I don't know, it'd be cool to have like a board, like a physical representation of how many by the neutrophil viewers like passed their A levels or finished their bachelor's in biology, or or a related field, or became doctors, became nurses, finished their theses, you know. Yeah, I mean, for my own selfish self-actualization too, right? Because while I might overestimate how much I like to yap, but like in my teaching role, I actually do realize that like I've been undervaluing the impact I have on students, just because in my particular current teaching job, I don't get to know what students do after they graduate unless they email me, and they're unlikely to email a lab teacher, I think, because we have them for so little, you know. Whereas professors, they have a much greater impact on average, I think, because you know you do research under them, you go to their lab meetings, they mentor you over your entire academic career as an undergraduate, sometimes into your master's and PhD. But as a lab teacher, I get you for a semester, and then you go, and then I don't really know what happens for you. So if any of you, you know, have victories, you can post them in the victories post, you know, and I think it'd be so fun to have a little thing, and I'll put a star for every graduate of the it's not right to call it the School of Fire, right? Because you're not really learning biology from me. You're learning around biology or about biology, but I've always considered my content to be more entertainment than education. You're keeping engaged with the content, and I hope that I am deepening a love for the content, but I do not replace your teachers here, because you know, it's not my job. Thank you for linking the Discord, Marlon. What topics do you plan on covering next, Fi? The next video is a secret, so I mean the next video is not a secret. I really hope to get this stupid influenza video out by the end of March, beginning of February, beginning of April. And then after that, I have a video idea that I have been holding on to since the channel was like 2 months old that I've been dying to get to, and it is about microbial evolution, and I will say no more because I think I think I want to build up hype for it in my own little way, right? Because I don't want to be known as just the immune guy. I want to be known as the cell and molecular biology guy, and sometimes that sometimes that means not making a video on a thing that hurts you. Sometimes that means just talking about cells and molecules like I've always wanted to do. I'm still a bachelor's and find such specific videos a lot more fun to watch than my basic molecular biology lectures. Thank you. Thank you so much. And again, you know, join the Discord. Tell me when you graduate, or tell me if you switch majors. That's also important to know too. Because I would say it's not my goal to keep everybody in biology, because I admit, like, not everybody should be biologists. Not everyone should be doctors or nurses, etc., etc. But I would like for my content to inform you as to whether or not you would like to stay. And if I played a role in helping you find out where you are meant to be, that would be very meaningful, right? Because myself, I've had a pretty awkward path through life, just in general. Right? Like, I thought I was going to be a medical doctor, and that did not end up happening. And it took a lot of input from a lot of different areas in my life to kind of figure myself out. So if watching my content helps you figure it out, that'd be very cool to know. Okay, with that in mind, I am absolutely pooped, and I want to thank you all for sticking with me till the very end. I forgot to mention this at the beginning of the stream, but congratulations to Cyre for reaching 1,000 followers on Twitch, showing the growing power of V-Theorem. I'm so honored and blessed to be a part of this group, and you know, I was reflecting upon this yesterday when she actually hit that milestone, that like we are doing good, and I'm excited to do more of it. All right, goodbye y'all. Thank you so very much for sticking around, and I will see you in the future. Bye y'all. Goodbye. Have a good day. Bye bye.